Ma'am, good morning, ma'am. மேடம் கேக்குது மேடம். ஓகே ப்ரோ, பாருங்க மேடம். ஒரு நிமிஷம், இது கட் பண்ணட்டுமா? ஹலோ ஹலோ மேடம், ஹலோ ஹலோ
ஹலோ சார், கேக்குதா?
கேக்குது மேம், கேக்குது.
ஓகே ஓகே ஓகே, தேங்க் யூ சார்.
ஓகே மேம்.
சார் ஒரே நிமிஷம் அப்படியே லைன்ல இருங்க சார்.
ஓகே மேம்.
சார் இப்ப கேக்குதுங்களா?
கேக்குது மேம்.
ஓகே ஓகே ஓகே, தேங்க் யூ சார். எனக்கு ஒரு சின்ன ஒரு இது வந்துருச்சு, அதான் ஹெட்போன்ஸ்ல. ஓகே ஓகே, தேங்க் யூ சார்.
ஓகே
ஜஸ்ட் வெயிட் பண்ணுங்க சார், நம்ம 10 o'clock போல வில் ஸ்டார்ட்.
நோ ப்ராப்ளம்.
ஓகே ஓகே, தேங்க் யூ சார், தேங்க் யூ.
தேங்க் யூ.
A very good morning to all our participants. We'll be starting a session at 10 o'clock. Hari sir, shall we start the session?
Okay, ma'am.
Yeah.
If you need to wait some more time, also no problem.
Yeah, can we just wait for 5 more minutes, sir? Let all of our participants
Okay,
join
no
us. Okay, thank you, sir.
Okay, no problem. Okay.
Okay, sir. Hari sir, shall we start?
Okay, ma'am.
Yeah. A very warm good morning to all our dear participants. We are indeed really very happy to welcome you all for this second day of our international conference on innovative strategies in chemical science and technology. We may now move on to our first session. I may request Dr. V. Karpagam, Assistant Professor, Department of Chemistry, Bhaktoshala Memorial College, to welcome our guest speaker. Karpagam, ma'am?
Yes, ma'am. Sure.
Yeah.
Good morning, honored are present here. I am really honored and privileged in welcoming the speaker of our today's session, Dr. J. Haribabu, JSPS Postdoctoral Fellow, Faculty of Tokyo University of Science, Japan. He has completed his Doctor of Philosophy in Department of Chemistry, National Institute of Technology, NIT Trichy. His area of interest is EVA protein and anti-cancer activity of nickel, palladium, and ruthenium complexes bearing heterocyclic biosemi-carbazone ligands. He completed his internship program in Kosing Medical University, Taiwan. He has published more than 45 reputed journals. He has received many awards. Postdoctoral Research Fellow Award from Japan Society for the Promotion of Science, Japan, in 2008 to till date. National Postdoctoral Fellowship from Science and Engineering Research Board, Government of India in 2019. Young Scientist Award from Dr. K.V. Rao Scientific Society, India for the year 2016 to 2017. Rajiv Gandhi National Fellowship Award from University Grants Commission, Government of India in 2013 to 2018. International Intern Fellowship from Kosing Medical University, Taiwan, 2017 to 2018. We can say more about his achievements, but due to time constraint, I am finishing this now. I may now hand over the session to Dr. J. Haribabu, sir. Thank you. Welcome, sir.
Thank you, ma'am. So, good morning to all. I am Haribabu, a JSPS Postdoc Fellow, Tokyo Institute of Science. We know cisplatin and their derivatives are well-known anti-cancer agents for treatment of various cancers. So, due to their several side effects, researchers are searching for new metal complexes with low toxicity and high activity in cancer cells. So, in this connection, so we design and synthesis of novel metal complexes for cancer applications. So today content of today talk. So introduction about a drug. So drug discovery, a structure of cell and biomolecules. So importance of metal-based anti-cancer agents and importance of heterocyclic thiocyanocarbons. And design and synthesis of palladium complexes with the thiocyanocarbons for the biological applications. Synthesis and anti-cancer mechanical pathway of water-soluble ruthenium complexes for biological applications, molecular docking studies on therapeutic agents for the COVID-19 and summary. So what is a drug? So drugs are the chemical substances which may be say natural or synthetic, so it's also called as medicine or pharmaceutical drug. So natural drugs are derived from the natural sources like plants. So synthetic drugs are derived from the chemicals. These drugs are used to treat and cure, prevent or diagnose a disease or to promote well-being. So how these drugs will work in body? So the drugs interact with the biomolecules of the biomolecule in the body and affect the physiological functions. This is the general idea behind all the medicines. So for developing one potential drug, it will take around 15 to 20 years. For this diagram explain the development of one potential drug from the various stages. In stage 1, drug discovery, synthesis of bioactive compounds. This bioactive compounds subject to in vitro and in vivo biological applications. After successful of these compounds, so enter into the preclinical development. Those compounds significant that compounds are entered into clinical trials. After passing all the stages, the drugs will approved as a compound approved as a drug for treatment of various diseases. So before going to main topic, so there is a small introduction about structure of the cell and DNA, cancer, so types of the cancers, and what is the causes for the cancer, and what is the treatments for the cancers. So our body, so made of trillions of cells, number of cells to form the tissues, the number of tissues to form the organs, so number of organs to form the system, that is nothing but human body. In the cell, the genetic material so present in the cell nucleus and mitochondria, the DNA so present in the chromosomes of the nucleus. So the chromosomes are look like thread-like structure. Each chromosome made of so protein and single molecule of deoxyribonucleic acid. This deoxyribonucleic acid so made of three main parts. One is nucleobases and ribose sugar and phosphate group. There are four types of nucleobases. So adenine, thionine, guanine, and cytosine. The guanine always pair with the cytosine through 3 hydrogen bonds, and thionine pair with adenine through 2 hydrogen bonds. So normal cell division in the human body, generally our body cells undergo cell division to form the 2 daughter cells. So after completing the 4 stages, G1 phase, S phase, and G2 phase, and mitotic phase. So in the start with from G1 phase, so after completing this mitotic phase, one cell will give the 2 daughter cells. This is the difference between the normal cell division and the cancer cell division. Normal cell division, the cells undergoes cell division to form the daughter cells. So further the daughter cell undergoes cell division. During this process, if any cell damage, the damaged cell so did not get any repair, that gets cell suicide. This process is called apoptosis. So in case of the cancer cell division, the damaged cell further undergoes cell division uncontrolled manner to causes the tumors or cancers. So cancer is nothing but so abnormal and uncontrolled cell growth of the cells in the body. So it's spread to other parts of the body also. So damaged cell is nothing but damaged DNA. So damaged DNA undergoes repair in presence of repair enzymes like endonucleases, exonucleases. If the damaged DNA successfully repaired, no hazardous. If a repair is failed, that cause the cell death. If a repair is incomplete, that is cause the cancers. So this is the difference between healthy cells and disease cell. In the healthy cells, the rate of DNA damage equal to rate of repair, and disease cell, the rate of DNA damage is greater than rate of repair. In per day, there are 5 lakhs DNA modification will be occur in the cell. So after repair DNA, so get repair. So in presence of enzymes, again this cells will include in the metabolisms. In repair is failed, incomplete, this cause the cancer and apoptosis. This is a main difference between the normal cell and cancer cells. So DNA modification and so morphology of cell. So what are possible causes for the cancers? Smoking, tobacco, unhealthy diet, a lack of physical activity, so alcohol use, and exposure to radiation, which may be X-ray and UV radiation, bacteria and virus infection. Some drugs also causes the cancers. There are above 100 types of cancers are affected in human body. Some of cancers are shown in the picture. So breast cancer and liver cancer, stomach cancer, pancreatic cancers, and kidney cancer, so and so. So nowadays, so cancer is one of the most and important problem in the world, which claim numerous death in each and every country. So according to World Health Organization, so by 2020 there are 630,000 people cancer death and 1.9 million people are new cases. So these are the cancer treatment options: immunotherapy, radiation therapy, so targeted therapy, chemotherapy, bone marrow therapy, surgery, and hormone therapy. In this, surgery and chemotherapy, radiation therapy frequently used in the hospitals. In surgery therapy, the doctors remove the cancer part from the cancer patients. After that, to give the chemo drugs to prevent the spreading of the cancer one part to another part and stop the growth of cancers. In radiation therapy, the cancer part exposed to radiation and kill the cancer cells, whereas in the chemotherapy, using chemo drugs to kill the cancer cells and stop the growth of cancer on one part to another part. So how these chemo drugs will work? So after entering the chemo drug into body to interact with the cancer cells and kill the cancer cells and stop them to growing or spreading to one part to another part. So cisplatin is the first metal-based anti-cancer agent. So discovered by the Rosenberg, and cisplatin and carboplatin and oxyplatin. These three drugs are commonly used in the worldwide for treatment of 50 to 70% of cancers for the treatment of cancer in the cancer therapy. So nedaplatin, so approved by Japan, used for the treatment of lung cancer, and leobaplatin, used for the treatment of breast and lung cancer, so approved by the China. So eptaplatin, so approved by Korea, used for the treatment of gastric cancers. So when you look into mechanism of cisplatin in the cell, the cisplatin once entering the cell, the labile nature of the chlorines replaced by the water molecule to form the aqua complex. This aqua complex further bind with the DNA to stop the DNA replication and damage the DNA, and further they will stop the growth of cancer. So on one cell to another cell and another part, one part to another part. So a majority of anti-cancer drugs are not cell-specific and high toxicity against normal cells. And this have a new and active research area in medicinal chemistry and bioinorganic chemistry. In recent years, most of anti-cancer drugs are derived from the metal-based therapeutic agents. These are the common side effects of cisplatin and their other derivatives. So vomiting, diarrhea, temporary hair loss, dry mouth and skin, and dry urine, kidney failures. These side effects motivate us to design the new anti-cancer drugs for the treatment for various cancers. Thiazide carbonones are thiourea derivatives. In particularly, so heterocyclic thiazide was a class of highly potent inhibitor of ribonucleotide reductase enzyme. So it is a response for the synthesis and repair of DNA. These four thiazide compounds are under phase 1, phase 2 clinical trials, so used for the treatment of various cancers. Due to their several side effects, so researchers are searching for other alternative compounds. The complexes with the thiazide carbozones is a considerable interest in the medicinal chemistry because of their wide range of biological applications such as antibacterial, so antiviral, anti-cancer, so antioxidant, so and so. These are the various coordination modes of thiazide carbozones so far reported in the literature. The chromones are well-known bioactive compounds. The chromones and their derivatives are used for the treatment of various diseases like cancer and diabetic, so inflammatory. So palladium complexes are alternative agents for the treatment of a cancer because of their structural and thermodynamic similarity to like platinum complexes. So we are searching for new metal complexes should have so strong binding with biomolecules, maybe DNA or protein, and high activity in cancer cells, in vitro or in vivo studies, and less toxicity against normal cells, no side effects, and DNA clearance ability. So this lead to cytotoxicity of the complexes. So inspired by the utility of palladium, chromone, and thiazide carbozone, in medicinal chemistry, we started to synthesis of the chromone-based palladium complex for the biological applications. This paper recently published in the Journal of Inorganic Biochemistry. This is a synthetic schema of synthesis of palladium complexes. The chromone-based thiazide carbozone ligand react with the palladium precursor in presence of methanol room temperature for 10 to 30 minutes to form the unexpected so pyrrole-based thiourea pallade complex than the expected one. The unexpected complex well characterized by the various analytical and spectroscopic techniques. This is the NMR difference between the ligand and corresponding complexes. When you look into this picture, there are A and B protons disappear in the NMR of the complex and appearance of OH and pyrrole CH protons in the complex spectra. And there is an interchange between thiocarbonyl carbon and carbonyl oxygen, which indicate the formation of the complexes. Further, we thought how this complex will form. So we thought first starting the forming of trident complex. So after that, literature, based on the literature, we found that the ligand react with the palladium precursor to form the bidentate palladium complex. After that, the methanol undergoes Michael addition or C2 position of the chromone to form the Michael adduct. The Michael adduct further involved in the ring opening and cyclization to form the unexpected palladium complex. This is the first report synthesis of this type of complexes in the literature for the biological application. Further, the formation of the complex was investigated by the mass and NMR spectroscopy techniques. First, we taken ligand in the RB in deuterium methanol and stirring at room temperature. So after addition of the palladium precursor, immediately the reaction mixture subject to mass and NMR spectroscopic techniques, and the mass spectra show the molecular ion peak around 690, which correspond to formation of the complex. So further, the same reaction mixture continues to stirring for another 10 minutes. Again, this collected the sample and subjected to mass spectra and NMR. So there is no change in the mass spectrum. So compare with the previous spectrum, which confirm the formation of the complex within short time. So further, this NMR changes are shown here. This is the ligand and final complex. So palladium precursor. So after addition of the palladium to ligand, so after 10 minutes, when you look into these changes, so see how this changing from ligand to complex. This changes clearly indicate that formation of the complex. Further, first we thought the pyrrole CH proton appeared as a singlet in the proton NMR, but we observed as a doublet, which may be due to interaction between phosphorus and CH protons of the pyrrole. The similar type of interaction were observed in the CH protons of phenyl in the present in the triphenyl phosphate, which was confirmed by the proton P13-1 HMBC NMR spectroscopy. So after that, the exact structure of the ligand and the corresponding complex were confirmed by the single crystal XRT. So when you look into this complex structure, so this is exist in the square planar geometry, and we've observed intramolecular hydrogen bonding between carbonyl oxygen and hydroxy hydrogen. This type of interaction very rare and very good for the biological applications. So after that, we synthesize successfully compounds, and this before going to anticancer application, we need to find whether these compounds are binding with biomolecules or not. So for this reason, we study the DNA protein binding studies for the complexes. So DNA is an important cellular receptor, and DNA molecular target in the designing new anti-cancer agents. Proteins main molecular targets in the action of the anti-cancer drugs, as the serum proteins perform transport and the distribution accumulation of the drug in the tumor tissues. So therefore, the study of binding properties of the new metal complex with the biomolecules is a great significance for the designing new and anti-cancer agents for the treatment of various cancers. So generally, the cytotoxicity of the complex, cytotoxicity of the anti-cancer drug is related to their ability to bind with the DNA through the non-covalent or covalent bonding. So DNA binding are two types: covalent interaction, non-covalent interaction. So covalent interaction, for example, cisplatin. So when you look into previous mechanism, the aqua complex of cisplatin to form the bond with the guanidine in the nitrogen atom of the guanidine. So after that, non-covalent interactions, these are two types. So intercalation binding mode, group binding. For in the intercalation binding mode, ethylene bromide is one of the example. When you look into this picture, how this ethylene bromide so penetrate into so between the two nuclear bases. This type of interactions are called intercalation binding mode. So whereas group binding, these are two types again. So minor group binding, major group bind. So where compound interact with the minor group binding area, this is called minor group binding. Whereas compound is interact with the major group area that called major group binding. So after that, so the complexes, the intercalation binding mode of the complexes were confirmed by the absorption spectra and fluorescence spectra. So the results also shown here. The absorption spectra of the complex 2 in presence of absence of DNA shown here. After fixing the concentration of the complex, so after incremental addition of the DNA, there is a decrease in the absorption intensity. This spectral changes clearly indicate that the complex interact with the DNA. So this and after that, the changes is plotted and Kb value also calculated and values are shown here. After that, the intercalation binding mode of the compounds with the DNA confirmed by the ethylene bromide displacement study. So ethylene bromide displacement study, the ethylene bromide show less intensity in the absence of DNA. So in presence of DNA, this increase the emission intensity of due to the intercalation between the ethylene bromide and DNA. So after addition of the complexes, there is a decrease in the gradually decrease in the emission intensity of this. This clearly indicates that the complex interact with the DNA through the intercalation by replacing the ethylene bromide. And K quenching value also calculated from the Sternberg plot and the values also shown here. From this value, it clearly indicates that the complex with the phenyl substitution showing good binding with the biomolecules. Then, so we need to know why this complex shown good active binding with the biomolecule. For this, we study the molecular docking study. So molecular docking study is a theoretical study. So it is very important for designing new anti-cancer agent as well as to understand the interaction between the biomolecules and so new drug molecule. And docking attempt to find the best matching between the two molecules. When look into this picture, in the Swayamvaram, we find the best match for the bride from the among the all. So now the compounds are subjected to molecular docking study. So using this this targeted DNA, this targeted DNA taken from the protein data bank from, and we analyze the results and show that results are shown here. The complex 2 showing good binding energy with the biomolecule. The representation figure also shown here. This how this complex penetrate between the two nuclear bases in the DNA, and this is zoom image. This is the dot indicate the interaction between the compound and DNA. So these are the possible reasons why the complex show good binding with the DNA. First, the carbon or oxygen to interact with the nitrogen atom of the cytosine to form the hydrogen bond. So another oxygen atom present in the hydroxy group to interact with the NH atom of the guanine to form the one more hydrogen bond. The 251 number rings show 5pi interaction with the cytosine and guanine. The terminal phenyl ring showed pi interaction with the cytosine and guanine too. And phenyl rings present in the triphenyl phosphine showed hydrophobic interaction with the target DNA. This is a reason why this compound showed good activity with the DNA. Further, so we found the good compound are showing good binding with the DNA, and we studied the anti-cancer activity for these compounds and the 3 different cancer cell lines: A549, lung cancer, liver cancer, and lymphoma cancer cells. And the results also shown here. This graph represented the percentage of cell death and concentration, and based on this, we calculate the IC50 value of this ligands and as well as the complexes, and values also tabulated here. From these values, we found the complex 2 showed good activity in the G2 cancer cell lines with IC50 value 10.4 micromolar, when compared with the well-known anti-cancer agent, is showing good, better activity and similar activity with other anti-cancer agents. And this all the compounds showed so less toxicity towards normal cell lines. So from this study, we conclude that the compounds only selectively active towards cancer cells than the normal cells. So after that, so previously I explained apoptosis is nothing but a cell death process. So there are four types of cell death process. One is apoptosis, necrosis, autophagy, and paraptosis. So apoptosis is nothing but the programmed cell death is mainly due to the DNA fragmentation. So to maintain the cell health, so in the case of necrosis, the swelling of organelles and the rupture of the plasma membrane, which cause several health damages in the cells. So autophagy is a lysosomal degradation. Paraptosis is a cytoplasmic evacuation. These are the few drugs, for example, for the apoptosis and paraptosis. So after that, so we need to find the effect of the complex towards the cell death mechanism. For this, before that, we need to study the morphological change in presence of complex and without complex also. This is a control cells. So after this control cells is treated with three different concentration of the complex 2, there is a drastic change in the morphology. So when you look into this picture, we know the clear how this changes from control. These changes clearly indicate that complex induce the cell death in the cancer cells. So further that we need to confirm the what type of cell death. So generally, anti-cancer drugs used in chemotherapy based on their ability to induce apoptosis in the cancer cells. So flow cytometry is a good technique to find out the cell death mechanism. So for this we conducted a flow cytometry analysis. This is a control cells without treating the complex. This R2 is represented the live cells, and R3 represented the early apoptotic cells, and R4 represent the late apoptotic cells, and R5 the dead cells. So after treating this cancer cells with two different concentration of the complex 2, there is a drastic change in the R3 and R4 and R5. So these studies clearly indicate that the synthesized complex induce the cell death through the apoptosis. So this graph represents the percentage of cell survival versus concentration. So by increase the concentration, there is a cell viability decreased. So already we know the cancer is nothing but overgrowth of cells abnormally in cell division in these cells. It can be strongly limited by arresting the cell cycle. For this reason, we study the effect of the complex on the cell cycle. This is a control cells. So after the F2 cancer cells we treated with the three different concentration of the complex, there is a increase in the G1 phase and decrease in the G2 and S phase. So these results clearly indicate that the complexes arrest in the G1 phase. So it stop the growth of further spreading the cancer cells one part to another part. Now, based on the above studies, we confirm that the complexes induce the apoptosis cell death process. Generally, the apoptosis cell death process acted by the two pathways. One is intrinsic pathway, extrinsic pathway. So in the mitochondrial pathway, the BCL2 box are present in the mitochondria. This is genes are treated with two different concentration of the complexes. It activates the release of cytochrome C from the mitochondria. The cytochrome C activates the CAPS3. The CAPS3 trigger the cell death through the mitochondrial signaling pathway. The proposed cell death mechanism also shown here. From this mechanism, we conclude that the complexes induce the cell death through the apoptosis via mitochondria. From this, we confirm the complexes are actively in the F2 cancer cell lines and trigger the apoptosis through the mitochondrial signal. So after that, we need to synthesize some other compounds. Generally, the water-soluble compounds are very important for a pharmacological response. and the water-soluble drugs are very rare in the literature. So for this connection, we design and synthesis of water-soluble mono and binuclear ruthenium arene complex for the anti-cancer applications. So before going to main topic, there is a small introduction about why ruthenium arene complex are important. The ruthenium arene complex have similar ligand exchange kinetics to that of platinum complexes and capability to interfere with iron metabolism. And with the iron complex are highly active in the cancer cells and less toxicity towards normal cells and structural integrity. This this is the back structure of the ruthenium arene complexes. So this look like a piano stool, and the arene occupied by the sheet of the stool, and three legs are occupied by the ligand and labile nature of the atoms. The labile nature of the atom undergoes the activation like cisplatin. This aqua complex may be interact with effectively with the biomolecule. So this is the how the pharmacological properties of the can be varied using the ruthenium arene complex. So ruthenium arene moiety exhibit hydrophobic interaction with biomolecules like DNA or protein. So and or maybe alkyl or some other functional group which may be stable, which can act as a enzyme inhibitor. So X, Y, and Z. So X and Y is the labile nature of the group which undergoes activation to form the aqua complex. The further aqua complex will react with biomolecules. And YZ is the mono and bidentate ligand. By varying the ligand structure, maybe vary the biological properties of the compounds. So based on this is the evolution of ruthenium arene complexes. In 1965, Clark reported first ruthenium-based compound as an anti-cancer agent. So further, they developed in 1989. So some ruthenium chloro compounds. So under clinical trials, this compound showed the lack of efficiency in the normal cell lines. Further, research are finding the alternative compounds to ruthenium arene complex. So these are some of example under a clinical trial. So next trial I will explain the difference between the cisplatin and ruthenium arene complexes. So what is the primary mechanism of the action of the cisplatin? So we already studied. So cisplatin interact with the DNA, whereas in NAMI, so they block the ion channel, and Kp1339 and this distribution of the cellular redox potential. So in the raptor C is a arene complex. They bind with the histone protein to activate the biological activity of the compound. Some of compounds show good activity in vitro. Some of complex are less activity in vitro. Some other complex shows good activity in the in vivo studies. These compounds are used for treatment of various cancers. So already cisplatin is approved by the and used as a drug. And NAMI is completed phase 1 and phase 2 clinical trial. So but lack of efficiency, there is no further development. The Kp1339 completed phase 1 trial and further is going on other studies. And after that, so Rapta C is not under the clinical trial. So based on this activity of ruthenium complex, we started to synthesis of ruthenium arene complexes from the indole ligands. This is a indole based thiazide carbazide ligands and reacted with ruthenium paracetamine in presence of toluene at room temperature to give the mono and binuclear complexes. Interesting here is by changing the small changes in the N terminal to form the different nuclearity complexes. So where when R equal to H to give the mononuclear complex, where R equal to methyl to give the binuclear complex. So what is the reason behind? So for this, we understood through the molecular docking study. So when look into from the DF study, so when look into DF study for mononuclear of methyl substitution to give the binuclear complex to require 11.5 kilocalorie energy, whereas without substitution it requires 14.7 0 kilocalories. So this is the reason why without substitution giving the mononuclear complex and with the substitution giving the binuclear complex. So after this, these complexes are well characterized by using various spectroscopic techniques, and the structure of both mono and binuclear complexes are confirmed by the single crystal Xrd. So this both the complexes are good soluble in the water. This is very important for the biological applications. So we find again these compounds are, we need to study the efficiency of the compound towards biomolecules, and we studied the DNA binding studies using absorption spectroscopy and emission. So and the results also shown here. The binuclear complex showed good activity than the mononuclear complex. So further, the exact binding mode of the complex with DNA was confirmed by the CD spectra and viscosity studies, and the results are also shown here. The CD spectrum showed the negative bond around 240 and a positive bond around 270, which is due to the helicity and base stacking of the DNA. So after addition of the complex, there is a change in the positive and the negative bond, which indicate that the complex interact with the DNA through the intercalation. So further, which is supported by the viscosity studies. So after that, we need to study the effect of compound towards DNA cleavage. For that, we study the DNA cleavage ability of the complex in presence of complexes. Generally, the mobility of DNA in agarose gel commonly taken as evidence for the evidence for the interaction between the DNA and targeted drugs. So we studied the DNA cleavage ability in two different DNA, and it is also shown the binuclear complex showed good DNA cleavage ability than the mononuclear complex. So after that, both the compounds also showed good binding with biomolecules. Further, so we understand through the molecular docking study. Then results also shown here. The complex 18 with the bionuclear complex showed good binding with the good binding energy with the DNA as well as the protein. These are the possible reasons. The NH atoms of the indole react with the oxygen atom of the guanine and NH atom of the guanine to form the hydrogen bonds, and terminal NH atoms will react with the oxygen atom of the guanidine 22 and guanidine 23 to form the hydrogen bonds. So previously I told the ruthenium arene moiety show hydrophobic interaction. We observed similar type of interaction between the ruthenium arene moiety and the targeted DNA. So after that we understand the interaction, possibility between the protein and complexes. The complex strongly bond with the 22 active sites of the amino acids. This is some of the how this moiety interact with various amino acids in the BSA. This phenyl ring and arene moiety are showing hydrophobic interaction. The NH atom showing interact with the oxygen atom, the glutamate and valine to form the hydrogen bonds, and terminal hydrogen NH atom also showing good interaction with the amino acid in the BSA. So then we find both the compounds are good activity towards good binding with the biomolecules, so especially in the binuclear complexes. Then after that, positive results from this study, we studied the effect of the compound towards the normal cells as well as cancer cells. For this, we selected two cancer cell lines, one normal cell line. So A549, a model of lung cancer, FG2, a model of liver cancer. So and the IC50 values also calculated, and values also shown here. The binuclear complexes showed good activity in the lung cancer with the IC50 value 11.5 micromolar, and compared with the cisplatin is showing better activity than the cisplatin, and less activity towards normal cell, which confirm the compounds are selectively active towards the cancer cells and less toxicity towards the normal cells. So after that, we need to confirm the effect of the complex toward the morphology and the cell death. So for that, we study the staining as a in presence of complexes. This is the staining assay in presence of complex. Without complex, this is a control cells. When treated there, so IC50 concentration of the binuclear complex, there is a drastic change in the morphology. Some of cells are also shown with the arrows. These arrows indicate the apoptotic cells of the cancer cells. So from this, we understand the complex cells induce the cell death in the cancer cells. So after that, we need to confirm the cell death process. Previously, I told mainly the apoptosis is due to the DNA fragmentation, and we studied the DNA fragmentation. So in presence of complex, the DNA fragmented. So from this pattern, we conclude that complex has induced the cell death through the apoptosis cell death process. Then, so after that flow cytometry, also we studied the population of the how this distribution of the cancer cells in the different quadrant, and which was confirmed by the flow cytometry. So after treatment of the control cells, there is a drastic change in the live cells and increase in the R3 and 4. From this overall results suggest that the complexes actively, specifically active in the A549 cancer cell and trigger the cell death through the apoptosis. So this good for the development of water soluble compound. So after that, previously our target is synthesis of chromone based palladium complexes, but we fail to lead to unexpected palladium complex. By changing the reaction condition, maybe we can achieve our target compound. This is a previous results. So we think whether it is possible to trapping the reaction intermediate during the reaction. So it is maybe possible by changing the reaction condition. In this connection, so again we started to synthesis of chromone based palladium complexes. The chromone based thiazimic carbozones react with the palladium precursors, similar condition, but with a slight change under nitrogen atmosphere to achieve targeted chromone based palladium complexes. So additionally, we found the presence of OCH3 group in the C2 position of the carbon. Maybe the complex during the formation of the complex, undergoes the methanol Michael addition. Further, this complex are very new in the literature and is very good for the catalysis as well as biological applications. This Michael adapt palladium complexes confirmed by the various spectroscopic and analytical techniques. This is a difference between ligand and complex. When you look into the spectra, there is a disappearance of the A proton, and there is a change in the B proton into lower frequency, and appearance of OCH3 proton, OCH proton, which confirm the presence of OCH3 in the complex. So further, the OCH3 carbon present in the C13 NMR of the complex. The overall results suggest that the complex formed through the Michael addition. The structure was confirmed by the single crystal oxide. When you look into the structure, the OCH3 group present in the C2 position of the chromone. Further, so the formation of the complex through the Michael addition, the proposed mechanism also shown here, like a similar previous mechanism with a slight change. So first, the forming bidentate palladium complexes. After that, the methanol undergoes Michael addition to form the Michael adduct. Michael adduct, the oxygen atom coordinate to palladium to form the tridentate palladium complex. So with the addition of OCH3, which further was confirmed by the phosphorus NMR. So after that, like previous experiment, again we by to confirm the how this complex is forming. So for formation of the complex, we study the mechanism study through the mass and NMR spectroscopy tools. First, we are taking ligand 3 in the RB in a deuterated methanol, sterile for the room temperature under nitrogen atmosphere. So after addition of the palladium precursor to ligand, so immediately this mixture is subjected to mass and NMR. So further, this sterile for another 5 minutes, and again we collected samples and subjected as mass. Again, this reaction mixture still for another 10 minutes, and this again the collected sample for the mass and NMR analysis. This is the mass results show that after addition of the palladium precursor, we found at a molecular ion peak around 696 and 727, which correspond to formation of the complex. So after the increase the time and collected sample also, there is no change in the molecular fragmentation in the mass spectrum of the complex, which indicate the complex formed very short time. So very short, we are unable to trap the intermediate. So one of this intermediate also, we got the crystals and I sent it to the X-ray analysis. Hope we are expecting maybe it's one of the intermediate in the reaction mechanism. So after this is the difference between the ligand and after addition of the palladium precursor after 5 minutes and 10 minutes, how this change in the NMR. So when compared with the ligand, so this changes clearly indicate that the formation of the complex from the ligand and precursor. Then after, so we successfully synthesis of 3 different types of the palladium complexes. This palladium complex was also subjected to DNA and protein binding studies. So using the absorption spectra and fluorescence spectra and Kb value and K quenching value also calculated. The complex 3 with the cyclohexyl group showed good binding with the DNA as well as the protein. So which further understand through molecular docking studies. The proposed docking models also shown here. So how this compound interact with the DNA and the protein. So in this complex, the oxygen present in the OCH3 interact with the NH2 atom of the cytosine to form the hydrogen bond. So another oxygen atom present in the ring form the hydrogen bond with the NH2 atom of the cytosine and guanine 2. So additionally, the terminal NH proton interact with the oxygen atom of the phosphate group to form the 2 hydrogen bonds, and terminal cyclohexyl ring and triphenyl phosphine show hydrophobic interaction with targeted DNA. So after that, similar interaction also found between the protein and complex. The terminal NH proton interact the carbonyl oxygen of the arginine and cyclohexyl group and triphenyl phosphine show hydrophobic interaction with the target BSA, and additionally the oxygen present in the ring also show the hydrophobic polar interaction with the amino acid in the BSA. Now, so the complex also, the synthesized compounds showed good binding with biomolecules. So after that, so we studied the effect of the compounds on the cancer and normal cell lines. So we selected 4 cancer cell lines, 1 normal cell line, and the values are the IC50 values also shown here. So compare with the previous palladium complexes by changing the coordination mode as well as the functionality of the chromone, there is a drastic change in the activity in the cancer cells. When looking to the complex 3 activity, there's the complex 3 showed 500 nanomolar IC50 value in the cisplatin resistant cancer cell lines and 900 nanomolar IC50 in the pancreatic cancer cells. And all the complexes showed less activity towards normal cells, which indicate the compounds specifically active towards cisplatin resistant cancer cell line as well as pancreatic. This results also compared with well-known benchmark anti-cancer drugs. And these compounds are very better activity than the compared with the well-known anti-cancer agents. So further, we study the morphological changes by host training and AOEB studies, and some of the results are shown here. The early apoptotic cells are shown with the green patches, and late apoptotic cells are shown with the red to orange to red color, and necrotic cells are shown with a large size of containing orange cells. From this study, we conclude the complex induced the morphological changes in the presence of complex and induce the cell death in the cancer cells. So after that, some of complexes showed good activity through the apoptosis. So the numerous anti-cancer agents stimulate this cell death process via reactive oxygen species generation. For this connection, we study the effect of the compounds on the ROS generation using DCFH DA staining method. So and the results are shown here. Without the complexes, the DCFA showed intensity around 54. So after addition of the complex, there is a change in the intensity, which indicate the complex generate the ROS in the during the cell death process. So after that, we study the effect of the cells towards a cell cycle, and we found that the cell compound arrest the G1 phase, and results also shown here. So after that, previous results we conclude that the cell death is through the apoptosis. The pathway of mechanism pathway also we studied through the Western blotting analysis. So from this analysis, we found that the complexes induce the cell death through the apoptosis via mitochondrial signaling pathway. The proposed cell death mechanism also shown here. The complex generated ROS generation and a down regulation, up regulation of the BCL2 bags and bad and bags, which activate the release of the cytochrome from the mitochondria. This cytochrome C activates the capase 9 and capase 3. This capase they activate the cell death through the apoptosis through in the mitochondrial pathway. So we found that by changing the coordination mode, the compounds are very active towards the cancer cell lines. So further that, so we need to study further another biological application. For that, we study the compound towards the coronavirus. We know the COVID-19 outbreak in the December 2019 located in the Wuhan in China. This disease has spread more than 200 countries. So till date, so the number of cases are 99 lakhs and around death are 5 lakhs. This is the structure of the virus. Now, so there is no drug for perfect drug for the curing this disease. For this, we study the effect of compound towards SARS-CoV-2 main protein. So through the molecular docking study, this is the three compounds with varying the substitution, and we selected two reference compound, chloroquine, hydroxychloroquine. These are already used in the as a drug for the treatment of corona. So after results are analyzed, the proposed models are shown here. The complex 3 showed good binding energy with this targeted protein. So and this also bound with the active sites of the amino acids in the protein. So further we need to establish the in vitro studies for the active compound for establishing the good antiviral drug for the treatment of various viruses diseases. I hope we will get good results in future. So summary. So we design and synthesis of palladium and ruthenium arene complexes. Some of complex also soluble in water, and all the complex showed good binding with the biomolecules. Some of complex showed good activity in the cancer cells and less toxicity towards the normal cell lines. And the complex to induce the apoptosis via ROS mediate mitochondrial signaling pathway. And interactionally, some of palladium complex showed good binding with a protein with of corona. So and further in vitro in vivo experiments will be carried out for the developing active complex to establish it potential as a clinical drug in the market. So acknowledgement. I sincerely thank to my PhD supervisor, Dr. R. Karvembu, National Institute of Technology, Tiruchirappalli. So with the absence of him, so I sincerely thank. Without support of my supervisor, it's difficult to achieve of these good results. And he still now is motivates us to designing new anti-cancer agents So for the different biological applications, some of compounds we send it to in vitro applications for corona in Japan. So maybe hopefully we'll get good results with the help of Sin Hoki, current supervisor. And I thank to Dr. Bones for the crystallographic studies, and I thank to Dr. Venilungam in Bharatadasan University for DFT calculations. I thank to Dr. Gayatri, University of Madras for the molecular docking studies. So I thank to my family members and friends. I thank to organizing committee members. I especially thank to Dr. Sangeeta, so head and department of chemistry. So Bhattavachalam Memorial College for the Humans for giving this opportunity for sharing my research thoughts with experts. So I thank one and all. These are some of publications published in the various journals. So still now we publish around 49 papers with the support of my friends and the supervisors. This is my contact details. So my email ID. So I thank to one and all for the kind attention. Thank you. Hello, ma'am.
Yeah, thank you very much, sir. We generally say that research is not the filling of pail, but the lightning of fire. Your talk has lightened the therapeutic effects of metal complexes for cancer, sir. Thank you. Thanks a lot. Sir, we have few questions
Okay, ma'am.
posted by our participants. Can I just read it out, sir?
Yes, ma'am.
Yeah. So the first question, does the presence of functional group affect the therapeutic effect of metal complexes?
Yes, surely this will vary by changing. That's why some of when you look into literature, some functional chromones without metal also showing good activity. So, for example, dextrorubicin that also one of good anti-cancer agent under clinic under as a anti-cancer drug in current society. So by varying the substitution and as well as functionalization of ligand, surely they affect on the towards biological applications.
Okay, so the next question, is there any metal preferences for complexes?
Metal?
Preference for complexes.
So generally base we cannot say like this. So because we know cisplatin, previously we don't know cisplatin is a anti-cancer agent. So after discovery of this Rosenberg only we know this is act as a metal complex. So maybe each metal complex show activity, but we need to without designing synthesis, without screening, we cannot say this metal is suitable for this application. So we need to synthesize and we need to study and we need to change the metal and you need to change the role of the metal as well as the substitution. So after getting the results only we will know that. But in case of this case, the Ruthenium arene complex nowadays, so good because of that arene moiety showing so some of biological activity towards biomolecules. So you know previously I discussed very well in the previous slides.
Yes, sir. So the next question, can any other precursor can be used other than palladium?
So maybe we can use palladium chloride. So we are under investigation. We are studying. We are synthesizing some using palladium complexes. We are getting some different results, and also we are confirm the structure also. So now in future we will develop that so compound also in literature. We will find the good publication in future.
Okay, so the last question, sir. What main factor decides the activity of the metal complex?
So one is based on one is the redox property. So another is kinetics, that is ligand exchange property. When you look into the platinum complex, so when you enter the cell, the labile nature of the chlorines is replaced by the water. After aqua complex, it interact with the DNA in a guanidine, 7th position of the nitrogen, to stop the growth of this replication of the DNA and stop the growth of DNA can be stop the spreading of cancer and kill the cancer cell. I already told the DNA is a main target for the cancer cells. So once we target the DNA, so we can most of, but there are several side effects. So there is some healthy cells also have DNA. They possibly they have so side effects towards the normal cell lines also. That's why still the research is going on for less side effects with the normal cell line, more activity. So that's why still the research is going on still for different metals and a different ligand system for developing the new anti-cancer agents.
Okay, actually, our next question is that actually whether the complexes have any side effects. Anyway, you're giving us the answer.
Yeah,
Okay.
they definitely have. We so definitely have. So there is no drug without side effects.
Yeah,
So
sure, sir, sure.
even though cisplatin is used for around 50 to 70% of cancer in the worldwide. So, but there is no alternative drug. But they have a lot of side effects. So that's why. So researchers are still working on cisplatin systems and other derivatives to with less side effects towards the normal cell lines. So definitely, this every drug have some side effects.
Side effects, yeah. Thank you, sir. Thank you very much for joining us and answering all the questions. Thank you very much. We'll now move on to our second session, oral presentation. I'm greatly privileged to introduce our chairperson of this oral presentation session, Professor Dr. P. Udayakala, Dean Chemistry and Director IQAC, Dr. MGR Education and Research Institute, deemed to be University, Chennai. Professor Dr. P. Udayakala has completed her Master's and MPhil from Manommaniam Sundanar University. She has received her doctoral degree from Pacheppas College, Chennai, affiliated to University of Madras. She also owes a master's degree in value education and spirituality from Annamalai University, and also another master degree in psychology from Tamil Nadu Open University. She started her teaching career from 1997 in Dr. MGR Education and Research Institute and has a teaching experience of more than 23 years. Her area of research includes quantum and computational chemistry, and also science and spirituality. She has guided 2 PhD scholars. Dr. P. Udayakala has more than 33 research publications to her credit. She is currently serving as a reviewer in many reputed scientific journals. Professor Dr. P. Udayakala has also received many awards like Sir C.V. Raman Research Award in 2014 and 16 as Best Researcher, Outstanding Research Contribution Award in 2015, and also Best Teacher Award from Manavar Ulagam 2019. We are very happy to have such an eminent personality as a chairperson of this session. Good morning, ma'am. Now I just hand over the session to Dr. Udayakala. Ma'am, shall I call upon the participants, ma'am?
Hi, yes.
Yeah.
Anyway, I'll just brief a little.
Yes, ma'am.
I'll just brief you. Just please call upon them. Okay, one by one.
Okay.
Good morning, and I'm really happy to chair the 6th session of this great, wonderful virtual conference. The Department of Chemistry, Bhaktavatsala Memorial College for Women. We have to specify the word women, right? So we have all joined together here, and it actually proved this lockdown doesn't knock down the research spirit of this department. And when I went through this abstracts, it was really amazing for me to find papers were from Assam, Mizoram, right to Trichy, Bangalore, north to south. Almost it covers all over India and China as well as Taiwan. So now I open up this session for the participants or the oral presenters to just deliver your presentation here, and we are here to learn from you researchers, and this great country stands upon the innovation of you people. Welcome you.
May I now call upon our first participant, oral presentation 16, Mr. Malswan. Sir, are you here? Malswan, sir? I think he's not here, ma'am. He joined us, but I don't know. Malswan, sir, are you here?
Hello, ma'am.
Yeah, he's come.
Hello, ma'am.
Hello, sir. Can you hear us?
Actually, my signal is not up to the.
So can we just have it at the end, sir? Can I have your presentation at the end? Can I just move on to the next participant? Ma'am, shall we move on to the next participant, ma'am? I think his
Yes.
signal is very weak there. I
Yes.
just now call upon the participant, oral presenter, OP17, Ms. Shainaz.
Yes, ma'am.
Yeah, can you just start with your presentation, ma'am?
Yes, ma'am.
Yeah.
Ma'am, is it visible?
Yeah, it's visible, ma'am.
Okay.
Ma'am, is it visible,
very-
ma'am? Udekla, ma'am? Is
Yeah,
it visible for you?
Yeah.
Okay, ma'am. A very good morning to one and all present here. Myself, Sana Alam. I'm a student of Department of Pharmaceutical Sciences, Dibrugarh University, Assam. Today, I'm going to discuss about podophyllotoxin and its derivative, a brief review on its isolation, biosynthesis, chemical synthesis, properties, stability of derivatives, pharmacology, pattern, and toxicology. Here are the contents of my presentation. Podophyllotoxin, it is a non-alkyl toxin ligandin. It is mainly a aryl tetrahedral ligandin, which is a group of cyanoligandin. It is mainly extracted from the rhizome and roots of Podophyllum species and Barbadiaceae species. The first structure of podophyllotoxin was elucidated in 1930. Podophyllotoxin, it is mainly useful for its anti-cancer and antiviral activity. In UK, around 3.5 ml of medication costs 44.49 pounds. From this, we can imagine how important this podophyllotoxin is. Podophyllotoxin is mainly available in the Eastern Asia, Afghanistan, and China. In India, it is also present in the scrub forest and alpine meadows, and also it is present in some forests of Kashmir. This is the structure of podophyllotoxin. In the structure of podophyllotoxin, we have five rings. Ring A and E is important for its pharmacological activity. In the structure, we have one electron ring, 3 methoxy group, 1 alcohol, and 1 carboxyl group. The carbon 4 is important for binding of podophyllotoxin to tubulin. If we add one bulky group at carbon 4, it will increases its anti-cancer activity and decreases its toxicity. Here are some common identification and analysis tests which is performed to identify the presence of podophyllotoxin in the solution. In chemical tests, we mostly mix 50% sulfuric acid. If it gives blue-violet color, that means the presence of podophyllotoxin. And another test is TLC, SPTC, and SPLC. These are the extraction and isolation process of podophyllotoxin. First, we collect the root and rhizomes of podophyllum, then cut it into small pieces and mix it in methanol. And by the process of oxidation, we collect the methanolic extract. And then by the process of distillation, we prepare a syrupy mass. Now this syrupy mass filtered by using acidic water. Now we collect the filtrate, cool it for 5 degrees Celsius. After cooling, it will produce some precipitate. Then we add hot alcohol in the dry precipitate. Now this solution again filtered and we discard the impurities and collect the filtrate and lastly cool it for 5 degrees Celsius and it will and last step is recrystallization in benzene which will give us podophyllotoxin. One thing is that in 1993 one patent was published by some researchers that which minimize the use of concentration of benzene since it is hazardous to human health. Here the biosynthesis pathway of podophyllotoxin. Since podophyllotoxin is a ligandin, it is mainly derived by cyclic acid pathway. The first product of biosynthesis is coniferal alcohol, and from this coniferal alcohol, by using oxidation, then reduction and dehydrogenation, we prepare podophyllotoxin. These are some chemical synthesis pathways through which we can synthesize podophyllotoxin chemically. These are oxoester route, dihydroxyacid route, dial ender reaction. The main mechanism of podophyllotoxin is it binds with tubulin and prevents cell division and it leads to apoptosis. Another mechanism is some derivative of podophyllotoxin like itiposide and teniposide. They bind with topoisomerase 2 and prevent the replication of DNA. there are around 285 derivatives of podophyllotoxin. Among them, adipocyte, tenipocyte, and adipocyte are the most important derivative from its pharmacological view. This adipocyte and tenipocyte mainly used for testicular cancer, lungs cancer, childhood acute lymphocytic leukemia, and so on. These are other pharmacological activities of podophyllotoxin, that is antiviral, antifungal, immunostimulatory, anti-SIB, anti-inflammatory, antiparasitic, and so on. Podophyllotoxin and its derivative like adipocyte and tenipocyte have many adverse effects. Like the most common adverse effect is drug resistant. It is cytotoxic to normal cell. Then it may also produce nausea, vomiting, then irritation of skin, diarrhea, hair loss, teratogenic effect, and sometimes it produces CNS depression as well as anxieties if ingested. Therefore, because of this adverse effect of the existing drug, some new agents like top 53 and K611 ZL331 azatosins are introduced in the market, which have less side effect as compared to the existing one. And here are the some formulations which are available in the market. It is tenipocyte injection, adipocyte capsule. And lastly, after the study, we can conclude that the full potential of podophyllotoxin is not studied yet. Although podophyllotoxin is mostly the highly soluble in methanolic extract, but other extracted also show some pharmacological activities which are not studied yet. Although we have a good anti-cancer agent that are developed from podophyllotoxin, still a good economic and site-specific anti-cancer drug is in need of the hour. And these are some references of my presentation. And lastly, I would like to thank Bhaktavachana Memorial College for Women for giving me the opportunity to present my presentation. Thank you, ma'am. Hello, ma'am.
Yeah, thank you, Mrs. Shahnaz. On to Dr. P. Udekla, ma'am.
It's a wonderful work.
Thank you, ma'am.
You have studied a lot about this, and I have just one question. And you
Yes,
have
ma'am.
given the adverse effects of this compound. And have you studied any bioactivity and then decided the adverse effects of this drug?
Ma'am, can you tell me again?
That is, my question is, you have given effects, right?
Yes,
Adverse
ma'am.
effects of this compound. And
Hello,
through
ma'am.
bioactivity, you have studied or analyzed those adverse effects.
No, ma'am. Actually, it's not my research work. I'm just writing one literature review on this topic.
Okay, okay, fine, fine. Excellent attempt you have done.
Thank you,
Okay,
ma'am.
great, great. Thank you.
Thank you, ma'am.
Thank you, Mr. Shahnaz.
Thank you, ma'am.
Ma'am, can I just call upon the next participant, ma'am?
Yes,
Yeah.
sure.
I now call upon our next oral presenter, OP18, Dr. K.S. Kiran.
Yeah, madam.
Sir, are you there?
Yeah, madam.
Yeah.
Okay.
So I just want to tell all participants that you'll be allotted a time of 5 + 2 minutes, 5 minutes for presentation and 2 minutes for questionnaire session. And see to that you just kindly stick on to the time. Thank you.
Can I start?
Yeah, you can start, sir.
Can I start?
Yeah, you can start, sir.
Myself, Dr. Kiran. Myself, Dr. Kiran, working as professor in Department of Physics, School of Engineering and Technology, University of Bangalore. My work is crystal and molecular tracking studies of bicyclohexadiols with focal adhesion kinase inhibitor. Here, mainly we are focusing on the crystallographic studies to prepare the ligand as well as the preparation of the protein, as well as the binding site prediction, docking energy calculation, results discussions, and function. First, need to have use bicyclohexadiols. Why you have chosen this one? It is a non-planar molecule having the peculiar property that down exactly at the C9 position of the molecule. One is bis cyclohexide nitrophenol, another is 2,4-cyclohexyl at the C9 position. Why I have chosen this compound is as a significant inhibition towards anti-cancerous as well as anti-tumor activity. There is about better structure, about the molecular packing, as Crystallography studies tells about the molecular packing, better insight to the design of the lead molecule. Stabilizing the structure, you have a better insight to the drug design. That is our ultimate aim.
Got it.
Docking. Hello. Hello. Hello, madam.
Hello.
Sir.
Okay.
Is audible, madam? It is audible,
Yes, sir.
madam?
Yes.
Yeah, it's audible, sir. Can proceed.
Okay. Docking studies provides a better have the drug receptor to the bending site prediction like that. First, you have to conformity about the qualities. Better to have a drug design using the docking studies. This is the second part. This is the audit diagram obtained for the crystallography studies. This is the expected structure obtained 3-hydroxy to hydroxy in the exactly the C9 position. This is a crystal data obtained for the first molecule to around in the monoclinic beta upon N with the R value as around 0.702. This packing diagram is only for the second molecule for this is crystal data molecule monoclinic beta upon C with R value is 0.03. Is there. We can be called as the docking method. What is the use of one is to identify the compounds are there. We need to have the screen pad, called as virtual screening. Further we go for the lead optimization method. This is our ligand in the previous one, receptor docking software. We'll call as a ligand receptor complex. Receptor with a potential inhibitor as a protein. Protein plus inhibitor is a future drug. That is our ultimate aim. How to prepare using the literature survey. In that the cofactors are removed, they convert to the PDQ format. This is the protein sugen for the compound. It is towards the agonist activity. It is called as a focal adenosine kinase. Why I have chosen is it is a cell addition, migration, population like that. This is the structure of the molecule. This is the ligand binding sites. You might have seen the crystal structure has been bind with the residues of the amino acids. This is called as a binding force. Same has been compared with the another binding site with the same molecules. You can see the binding site with the friction. This is a pharmacophore interaction about the molecule, exactly the ligand, how exactly residues of the amino acids are there with the surrounding molecule. This is to study various interactions, van der Waals, covalent, water. We'll call this one as using the distinguished scale that does study about the various interactions. These are the residues of the amino acids. This is called a molecular surface of the body interaction of the leaf plot. This is the residues of the amino acid. This is the crystal structure. We are calling the binding sites of the crystal structure. This is the energy. This is the result obtained minus 10 minus 6.13. Higher the energy, better the affinity of the molecule. With the residues of the amino acids. This is a dark score obtained around minus 10.62 with RMSD value around 2.41. These are these of the amino acids seen in the previous diagram. Similarly for the second structure, this is a crystal structure, binding site how exactly they predicted. Same has been compared with the plastics. This is the pharmacophore interaction for the second molecule. Similarly, second molecule minus 6.4 is a binding obtained for the molecule receiving the amino acids. Dark score around minus 7.8. We have compared that ligand A as well as ligand B. Binding energy is minus 0.42, minus 6.13. Dark score is minus 7.18, minus 10.62 with the inhibition of 18.76 plus 32.02. Better, what is the better version is after the pharmaceutical data is obtained, better the binding site prediction. Here, around -6.42 for the second one. Similarly, for inhibition around 19.68. Further co-crystallization has been taken with the animal trials. It is a challenging job that is called protein ligand complex. Future drug delivery discovery identify the disease, obtain that 2 to 5 years to take final approval, human clinical trial, finally approval like that. These are the stages of the genomics, a combination of crystallography, that is molecular modeling, in vitro and in vivo studies. Now we are doing with some animal that to isolate the protein. Further, we'll go for the preclinical trial, which is called as in vitro as well as the study. It is called as virtual screening. The intramolecular interaction between the ligand and protein was investigated. One of the bioactive compounds is protein inhibitor to block the FAK protein. The selected compound is taken as a wet laboratory to act as an effective anti-cancerous drug. These are the various software used by prediction. List of publications. Thank you, madam. I kindly thank Dr. Salam College for giving an opportunity to the conference. Thank you.
Thank you very much, Dr. Kiran sir. Now on to Dr. Udekla ma'am.
Professor, just for
Yeah,
clarity, I'm having a question. In this docking studies, why is it always hydrogen bonds and van der Waals forces and no other bond is interacting? Just for clarity.
Madam, that why means when doing the diagram there, we'll omit the cofactors in the CHO interaction earlier only. That's why we'll remove that PDB format to whatever the cofactors are there. Why means if you didn't move one, we don't get as a binding force, will cause a binding force with a better inhibition constant. Because of that, we will retain it, madam. About will call as a force like that. Force.
Thank you, Professor.
Okay.
Thank you very much, Dr. Kiran sir. Ma'am, shall I proceed with the next participant, ma'am?
Yes.
Yeah. I may now call upon our next presenter, OP19, Dr. Manigandan.
Yeah, ma'am, I am here.
Dr. Manigandan, you can just start with your presentation.
Yeah, sure. Just a moment, ma'am.
Yeah.
Yeah, ma'am, just unfortunately, I could not share my screens. Just a moment now.
Actually, initially it popped up, sir. Your slide was visible initially.
Yes, I know, but that's not a correct version.
Okay.
So may I start the presentation?
Yeah, you can start, sir. It's a pleasure.
Yeah, ma'am. Very good. Very good morning to all. Ma'am, could you show my presentation clearly?
Yeah, we are able to see it, sir. We are able to see.
Okay. Good morning. Yeah, good morning to everyone. I am Dr. Manigandan from Nanjing Medical University. So working as a postdoctoral researcher in Professor Chen group. So I am going to share one of the research topic is one-step preparation of polyurethane film with a silver nanoparticle coated with the glassy carbon electrode towards detection of the heavy metal ions and the antibacterial some biological applications. So first we're going to start about the heavy metals. As we know, the heavy metals it has a high density and a toxic. It's a poisonous in the lower concentration, even though it's present in the many environmental samples such as drinking water, soil samples, and air samples, etc. So these are all the normal components in the earth crust, and they cannot be destroyed or degraded. So these are all the basic definition of these heavy metal ions. As we know, some examples. As we know, the mercury is a highly poisonous liquid metal in the periodic table, which has a high specific gravity, is a 13.5 gram centimeter cube. So these kind of heavy metals we need to detect by using the several analytical techniques. For example, some chromatographic techniques or some other techniques, which is ICP MOS or ICP OES or some other spectroscopic techniques. There is several analytical techniques available, especially in the heavy metal detection. In my topic, it covers that the detection of heavy metals by using electroanalytical techniques, especially stripping voltammetric techniques. Why I am choosing the stripping voltammetric technique in the sense, because this stripping voltammetric techniques has a very sensitive and selective detection of the target heavy metals. So these are all the main advantages I am choosing for the electroanalytical tool for the detection of heavy metal ions. So after that, I am using a heavy metal detection in the environmental sample. What is the motto to detect these heavy metals? Because as per the WHO guidelines, if the as per the WHO guidelines, the heavy metal present in the drinking water, it can produce a severe effect in the human health. For example, if lead is excessively present, for example, if 0.05 ppm is excessively present in the lead in the drinking water, means it can produce kidney damage, neurodegenerative disease, and some other disease. So these are all the some disadvantages where lead or some heavy metal present in the water samples or some other environmental samples. It can be entered in our human body. It can produce a severe health problem. So these are all the main drawbacks to detect the heavy metal ions. So after that, the chemically modified electrode, as I mentioned earlier, I can use for the electroanalytical tool to detect the heavy metal ions. Here, I used for the stripping voltammetric technique by using the chemically modified electrode. The chemically modified electrode is nothing, the electrode which has been modified by using suitable, which has been used for the suitable modifying materials, which the modified material, it should have a higher electron conducting property, because the electrode normally has the conducting behavior. So this conducting behavior will be enhanced by adding some other substances, which means the modified material. So here I used for the polyurethane film and silver nanoparticles. So as we know, the ruthene is a good flavonoid. We can take it from our human, and then the silver nanoparticles also, it's a good antibacterial and some biological application. So these two materials have been coated on the modified electrode, which means the glassy carbon electrode. This electrode has been used for the two different applications. One is the heavy metal detection, and the second one is the biological application. So only one material used for the two different applications. So here the chemically modified electrode, basically it enhances the electron conductivity nature, and the electron transfer process, it will be enhances. As I mentioned earlier, these electrochemical techniques we can detect by detecting the target target the analyte with the lower potential. At the same time, we can improve the sensitivity, selectivity of the modified. We can improve the sensitivity as well as selectivity of the targeting analyte. And here, my main motto of my main work is the electropolymerization of the routine. First one is electropolymerization of routine, and second one is the electropolymerization of routine with silver nanoparticles, and third one is the electrochemical conformation of these modified electrodes, further the analytical parameter studies, and then on anodic stripping voltammetric detection of heavy metals towards lead and cadmium detection, and then biological application for the hemolysis assay and the antibacterial application. So first, I used for this polymer, which means the polyroutine has been prepared by simple one-step electropolymerization method by using a chronoamperometric technique. The chronoamperometric technique is nothing with respect to time, with respect to potential, with a different time, we can apply a constant potential. The analyte, which means the routine, has been deposited on the electrode surface. So here, with one potential, we used for the two material, which means the routine and the silver nanoparticle. By applying a single potential, these two material has been attached on the glassy carbon electrode. This glassy carbon electrode has been used for the detection of the heavy metal ions. So first, I used for the single step deposition of the polyroutine with silver nanoparticle with a different deposition time in the presence of 1 millimolar of routine and 1 millimolar of silver nitrate in the 0.1 molar sulfuric acid with a different potential, with a different time as a fixed deposition potential in the deposition potential of the 1.3 volt with a different deposition time, for example 5 minutes to 30 minutes. And then by different deposition potential, I obtained that by different deposition potential by applying a different deposition potential, I obtained 1.2 volt is the maximum and the optimization of the polyroutine and the silver nanoparticle formation on the glassy carbon electrode has been optimized. Further, as we know, the routine is a flavonoid. That routine has a many hydroxide group. This hydroxide group can easily oxidize to undergo oxidized form of the routine. But here in the case, when we apply a higher oxidation potential of the routine, it can be undergoes a free radical polymerization mechanism. This mechanism is proved with a suitable example. In this mechanism, the free radical, the oxygenated free radical has produced. This oxygenated free radical has initiated the reaction and then propagates the reaction. At the same time, the silver nanoparticle also attached on the silver nanoparticle with the polyroutine film. So this mechanism has been explained. Further, the single step deposited polyroutine with silver nanoparticle was further confirmed by several surface analysis tools. For example, the figure shows that the SEM images of the silver deposited, the polyroutine and with the silver nanoparticle deposited modified electrode with a different time variation. The A is a 5 minutes deposition, A is the 5 minutes deposition, C is the 10 minutes, and E is the 20 minutes and 30 minutes respectively. And the corresponding elemental morphing images also shown here. And then the figure I is the TEM images of the polyroutine with silver nanoparticles, where further confirms that the polymer film formation, the simultaneously silver nanoparticle also form. Further, we can confirm that the polyroutine and the silver present on the electrode surface by using the EDAX technique. Further, the elemental morphing images of the as prepared modified electrode where clearly shows that the each and every element, for example, the elements with the different each and every element, for example, oxygen, carbon, and silver were confirmed by using a separate elemental analysis tool. Further, the TEM images of the elemental morphing of the polyroutine film with silver nanoparticles were confirmed by the elemental morphing technique. The A shows the TEM images of the silver nanoparticle, which are shown in the 500 nanometer, and the B is the cation of the silver nanoparticle, and C is the L shell of the silver nanoparticle, D is the carbon atom, and the D represents the carbon, and E represents the oxygen. Further, we can, as we said earlier, the mechanism is followed by the free radical, which means the O dot has initiated the reaction, which has been confirmed, which has been further proved by the ATIR spectroscopic technique. As I mentioned earlier, we use the glassy carbon electrode for the detection of the heavy metals. But instead of this ATIR spectroscopic technique, we use the ITO, which means indium tin oxide film. We used for recording ATIR spectrometer, because we cannot do the FTIR, because this film can be damaged by using a FTIR. So that's why I used for the ATIR technique to confirm the polymer formation to confirm the polymer formation. In this ATIR spectra, the figure A and B shows that the monomer of the routine and the polyroutine, which means the monomer has a several OH group, as I mentioned earlier. But in the case of that polyroutine has a COC, which means the COC bond formation. This formation, which has been confirmed, and at the same time, the hydroxide, the OH stretching peak, and the COC and the other functional vibration peaks has been shifted in the figure A, which has been clearly shown in the figure, the A in the next slide. And then further, the polyroutine with silver nanoparticle formation were further confirmed by the XRD spectrometric technique. This XRD, the X-ray pattern method, were confirmed that the silver nanoparticles were present in the polyroutine film. Also, the polymer formation also confirmed. Further, the polyroutine with the silver nanoparticle modified electrodes were confirmed by using the electrochemical technique, which means the 5 millimolar of the potassium ferrocyanide and ferricyanide act as a redox probe in the 0.1 molar KCl solution. We use the different time interval deposited glassy carbon polyroutine with the silver nanoparticle deposited modified electrode. The 30 minutes deposited modified electrode shows the higher oxidation peak current with a lower deposition potential with a delta EP of 0.1648 millivolts. When compared to other modified electrodes, the 30 minutes electrode deposited modified electrode shows the lower oxidation potential with a higher peak current, which means this electrode has a higher surface area. This higher surface area can be used to detect some electroanalytical applications. So this is the one of the another parameter to detect the heavy metals. The same time we used for the detection, electrochemical impedance spectrometer with a different modified electrode, with a different time deposited modified electrode. The 30 minutes deposited electrode shows that the lower oxidation. The 30 minutes electrode deposited electrode shows the lower RCT potential, which means almost around 1222 ohm. 22 ohm. But in the case of the bare electrode shows that the 11,972 ohm. So when compared to bare and 30 minutes electrode deposited electrode, the 30 minutes electrode deposited electrode shows the lower RCT value, which means we know as per Ohm's law, when the resistance increases, the current will lower. The current will increases, the resistance will higher. So here, the lower resistance, the higher current has been produced. So this electrode can be used for the higher electron transfer application. It can help the higher electron transfer application. So we used for the 30 minutes electrode deposited.
Dr. Manikandan?
Yes.
I think
yes.
your time is getting over, sir. Can you just wind it up?
Okay,
fast?
just a minute, just 2 minutes now. Yeah. So I used for the detection of these heavy metals towards the detection of lead and cadmium ions. We detect we optimize some analytical parameter. For example, we optimize the pH study and the different electrolyte study and a different pre-concentration time. We study the optimized pH range is pH 5, which means the active buffer, and then the optimized deposition, optimized applied potential is minus 1.2 volt for the detection of lead and cadmium ions. The pre-concentration time also we detected in the time of 240 seconds. Further, these optimizing the analytical parameters, the lead and cadmium ions were detected in the concentration range range from 29 to 140 nanomolar for the lead ion, and the cadmium ion concentration is 57 to 203.5 nanomolar in 0.1 molar buffer solution. Further, these modified electrodes were studied for the different heavy metal ions, but in the case of the polyroutine with the silver nanoparticle electrode as showed only the selective detection of lead and cadmium ions as showed. The stability and the other analytical parameters have been studied, but at the same time, we used for the detection of these lead and cadmium ions in the presence of copper. This copper will be interacted to form a lead and copper intermetal alloy formation. So we protect by using the ferricyanide solution. This ferricyanide solution will form a copper hexacyanide solution. This copper hexacyanide solution is it will protect a copper ion in the precipitate formation. Then we can detect the lead and cadmium ion. Further, the zinc also we can detect it. This zinc ion by applying a potential in the minus 1.4 volt, which means it's a higher potential. It shows a higher concentration, which means 500 to 700 molar. Further, the analytical application, the practical applicability of the modified electrodes were applied for the different environmental samples, which means the tap water samples and the human hair samples and soil samples were detected, and the satisfied results also found. Further, as I mentioned earlier, we used for two different applications. The second application is the antibacterial application and the hemolysis assay application. Here we used for the antibacterial studies by using a E. coli with a different concentration, with a different mole ratio in the with a different material, which means routine, polyroutine with the silver and silver. But the polyroutine with the silver has shown the higher antibacterial activity. At the same time, we used for the hemolysis assay, which means the RBC cells. The RBC cells will interact with this nanocomposite, which means the polyroutine with the silver nanoparticle. This film has not interacted with the RBC cells, which means this material has been used for some medicinal and biomedical applications. So, thank you.
Yes.
Thank you, Mr. Dr. Manigandan. Now on to Dr. Udayakala, ma'am.
Yes, excellent and fabulous presentation, Dr. Ramalingam. And only one question.
Thank you, ma'am.
Because you have explained elaborately. Only one question is, like you have been telling, like it's sensitive and just a minute, and sensitive and selectivity can be improved.
Yes.
So in what way you can improve the sensitivity and selectivity?
So here I can also improve the sensitivity, because as I mentioned, the selectivity. See, in the detection, in the zinc ion detection, if we go for the higher deposition potential, which means minus 1.4 volts, we can detect the silver. Sorry, we can detect the zinc. So below the 1.2 volt, we can detect only lead and cadmium ions. In the selectivity, in the sense, suppose if our sample has suppose if you have a water sample, this water sample has a lead, cadmium, and zinc, mercury, so many samples, so many heavy metal ions. We can selectively detect. If I want to detect only a lead and cadmium ions, we can using a suitable buffer solution. Suppose if I want to choose only a lead and cadmium ions, I can place a buffer solution pH 4.5 to 5 range, and we can optimize the potential range. Because the lead, suppose if your sample has a mercury, the mercury will be reduced 0 to 1.0 or minus 1.0, this potential range. If I apply a higher potential range, the higher elements, the higher heavy metals only interacted with the electrode surface. Suppose,
excellent.
That's why. Yes.
Thank you so much.
Yes, thank you.
Thank you very much, Dr. Manigandan. Ma'am, may I move on to the next participant?
Please call the next.
I just call upon our oral presenter number 20, Dr. J. Tirupati. Tirupati sir, are you there? Jayaraman Tirupati? Dr. Tirupati, sir, can you just join us? Actually, he's there, but he's not responding.
Maybe your audio is not.
Yeah, he has muted himself, ma'am. He has muted himself. He's there, ma'am. So
Okay.
can you just, yeah.
Okay,
Start with the
ma'am.
presentation, sir.
Okay, ma'am. Hello. So, very good afternoon to one and all. So, I'm going to present my paper on the corrosion. So, the topic is the inhibition effect of phenol, formalin, aniline, melamine, urea resin on the corrosion of mild steel in sulfuric acid medium. So, actually, first we have to tell about what is in the corrosion. The corrosion is we known very well. That is corrosion is when the metal is exposed to the environment and the presence of moisture, it will tend to decay. That is called corrosion. So due to the corrosion, many of the physical properties of the metals will be lost. That is like thermal conductivity and electrical conductivity, and also industrial also affecting by this corrosion. So to prevent this corrosion, so we can use lot of the methods are available for preventing the corrosion and also to reduce the corrosion rate. But in this method, that is we are going to use in this method I'm using the inhibitor. So by using the inhibitor, how we can reduce the corrosion rate, that is we are going to discuss about. So in which I have taken resin, and before that is what are the types of the corrosion? What are the types of corrosion? That is types of corrosion. So a lot of the types of corrosions are available. Acid corrosion, galvanic corrosion, pitting corrosion, and uniform surface corrosion, shallow pit, intergranular corrosion, crevice, and soil corrosion. So these are the corrosions are arising due to the mainly the industrial applications, that is by acid pickling and cleaning the industries and some of the processes used in the industries. So these types of corrosions are occur. First, acid corrosion is mainly caused by, that is we know that is acid corrosion, that is the first metal pipe corrosion. So due to the when the metal is dipped in the lower pH, that is acid, so the metal will tend to corrode. That is the acid corrosion. Galvanic corrosion is due to the difference in the electrode potential. So when the two metals or alloys are placed in the presence of moisture or some acid media, so the which metal has a higher electrode potential, that will tend to decay. So metal which has a higher electrode potential will be affected by the corrosion. That is called galvanic corrosion. Pitting corrosion is a localized corrosion. So it will in which it is also a differential aeration corrosion. That is the pit will be formed in the pipe or any other metal in the center post. That is it will become an anode. So less aeration will be received. That's why difference in aeration there is a cause of the corrosion. At uniform surface corrosion, it will applicable to both uniformly to the entire surface of the metal. And shallow pit corrosion, intergranular corrosion is nothing but that is it will be due to the presence of impurities present on the metal. Due to the presence of the impurities present on the metal, so the intergranular corrosion will be occur. Crevice corrosion. So when the metallic or non-metallic object is touched to any liquid media as a moisture, the crevice corrosion will occur. That will be shown in the figure itself. That is the last one is the crevice corrosion. First one is the acid corrosion. Second one is the galvanic corrosion. And third one is the pitting corrosion. And fourth one is a uniform surface corrosion. And fifth one is the intergranular corrosion. And last one is the crevice corrosion. So there is also available the soil corrosion also. So these are different types of corrosion. So how we can reduce the corrosion or corrosion rate? By using the inhibitor. The inhibitor is nothing but what is the inhibitor? The inhibitor is forming the insoluble precipitate on the metal surface. It is nothing but it is forming the insoluble precipitate on the metal surface. That is called inhibitor. Three types of inhibitors are available. First one is a cathodic inhibitor. If we apply the cathodic inhibitor, it avoids the cathodic reaction itself, and the anodic inhibitor it avoids the anodic reaction itself. And if you are applying the mixed inhibitor, it will affect both and cathodic and anodic reaction. So from the TAF apply itself, we can predict whether it is a cathodic inhibitor, anodic inhibitor, or mixed type inhibitor. We can predict from the TAF polarization. So first we have taken here the mild steel. We have taken mild steel here. So mild is also used in the construction purpose and many other industrial purposes using the mild scale. So in the mild scale, the high carbon content of 0.2% to 2.1%, and other things are manganese, copper, silicon will also be available. So the next one is so how we can, in which we can use the mild steel. So we have taken the mild steel, the 0.5 millimeter thickness, and also the 1 centimeter square surface area. So before we are going to discuss study the corrosion rate, we have to know about the weight loss studies method. So in this weight loss studies method, we are using, that is first we have to dip the solution in with acid itself. First we have taken the mild steel 1 centimeter square. It will be dipped in the sulfuric acid solution. So another one is we have to immerse with both an inhibitor and that is we call that's sulfuric acid, that is sulfuric acid. So we dipped both into sulfuric acid and inhibitor in the another case. So from this we can calculate the weight loss method. We can calculate the corrosion rate. That is average weight loss without inhibitor and average weight loss inhibitor. That's a formula will be given. It's inhibition efficiency can be calculated from this formula. Average weight loss CR0 is average weight loss without inhibitor and average weight loss with inhibitors. So from the average values we can calculate the that is a corrosion rate and also inhibition efficiency also we can calculate. So when we are adding the inhibitor, the corrosion rate will be decreased. When we are adding the inhibitor, this corrosion rate will be decreased. So how we can prepare this resin? That is we actually prepare the resin like that is a polymer. So it is a very long process. That is first we have to prepare the phenol sulfonic acid. It is undergoing the condensation process. From the condensation process, we can obtain the condensate. It is mixed with the homogeneous solution of previously prepared sodium bisulfate, which is mixed with the aniline solution. And this first containment and second one is another containment. We have to prepare urea, water, formalin, so and melamine. So both are mixed thoroughly, so we can obtain the solution, that is resin will be obtained. So main risk will be here, that is how we can get the solubility. That's why we are adding the sodium bisulfate compound. It will come into sulfonate compound, so the solubility will be it will be readily soluble in water. So due to the preparation of the solubility in water, so we can prepare various molar concentrations by using the various molar concentration. We can, that is by using the various molar concentration, that is starting from 0.5 millimolar to 1 molar, then up to 2.5 millimolar. So every addition of the millimolar concentration of the resin, the corrosion rate will be decreased and the inhibition efficiency will be increased. So that is we found out the efficiency of the inhibitor. So these are some ingredients of the inhibitor. So next one is weight loss studies. That is we obtain the weight loss from the weight loss method. How we can obtain the inhibition efficiency? So due to the bigger molecular size and it is a polymer, it consists of 5 electrons in the aromatic staining system. It is used as an excellent inhibitor. So that's why we obtain the corrosion inhibition efficiency very lot here. By the addition of that is a 0.5 millimolar, that is 1 millimolar, and up to 2.5 millimolar. The next, we also support the these functional groups. We have taken the IR spectroscopy. That is a range from 750 to 4000 per centimeter. It is a IR spectroscopy. So in which that is obtain the various frequency range. That is from the infrared spectrum of the compound. The broadband 3352 per centimeter is attributed to both OH and NH groups, and 1047 per centimeter is attributed to either compound is formed. That is 1047 per centimeter, and 1639 per centimeter is attributed to carbonyl functional group, and similarly 2360 is attributed to carbon-carbon group. So the functional groups are confirmed by the if taking the infrared spectrum. So next we are going to discuss about the to support the inhibition efficiency. First we taken the infrared spectrum to confirm the presence of the functional groups. Here we are going to support the inhibition efficiency of the inhibitor. We are going to discuss about the potential dynamic polarization measurements. So what is my potential dynamic polarization? So in which we supply the potential and we supply the potential and monitor the current. monitor the current rate. That is a potential dynamic polarization. We change the potential and apply and identify the what is happening in the current. That is called potential dynamic polarization method. So by using this method, we can take the that is a TAFL plot itself. So here the inhibition efficiency is obtained from the corrosion, that is a corrosion current density of mild steel without and with the inhibitor. The inhibition efficiency are calculated from the equation. That is a corrosion current density of mild steel without and with the inhibitor respectively. So from the potential dynamic polarization method, we can obtain the that is here both there is a wells in both in the cathodic and anode direction, the TAFL plot we obtain. So the plot of the potential versus current here. So by increasing the inhibitor concentration, the inhibition efficiency are here also increased. It is also after by the that is we can say there is a corrosion current density, that is the values of the corrosion current density decreases. So from this we can identify that is a corrosion rate is decreased.
விளம்பரம்னு சொன்னா நம்ப மாட்டியாயா? படம் எதுவும் இல்லையா, சும்மா கத்தினுக்குறானே.
So next one is beta C and beta A. That is a cathodic TAFL slope and anodic TAFL slope. So both it will affect, it will prevent the corrosion reaction by preventing the both cathodic and anodic site of the reaction. Both it will prevent the both anodic and cathodic side of the reaction. That's why it will be acting as a mixed type inhibitor. And also the corrosion potential is obtained. It is not much change in the corrosion potential. So that is also obtained from the TAFL plot itself. So TAFL plot is, that is a TAFL cathodic TAFL slope and anodic TAFL slopes are obtained from the extrapolation of the.
Hello.
Yes, madam.
Sir, I think we are running out of time.
Okay. So last 2 minutes.
Yeah. Just.
So yeah, next one is electrochemical impedance spectroscopy. So electrochemical impedance EAS.
7% and
electrochemical impedance. There's an impedance. What is the impedance? Impedance is resistance to flow of the electric current. That is the impedance. From the electrochemical impedance also we can support the inhibition efficiency by taking the charge transfer resistance and the double layer capacity. What is the charge transfer resistance? So RCT values are here increasing due to the charge transfer process are taking place, and also that is the RCT is controlling the speed of the metal dissolution. So that's why the values are increasing. Double layer capacity, CDL is nothing but double layer capacitance. So here the double layer capacity values are decreased due to the 2 layers are formed. It is like a charged capacitor. That's why it is called a double layer capacitor. So charged capacitor. So it will prevent the corrosion of the reaction from entering into the solution to metal surface, and it will so that's why it is an important one. So next one is by supporting the by taking the scanning electron microscope. It will be that is before that is it is dipped. If it is dipped in the sulfuric acid medium, there are a lot of pits are observed. But if you are taking by adding the inhibitor, maximum amount of the inhibitor, and after 1 hour immersion, it will take under that is by taking the SEM. So it will show that there is no pit or observed in the SEM electron microscope. So that is also confirming that is this inhibitor will prevent the that is a corrosion reaction taking place, and also reduces the corrosion rate. And also it is another one is EDS spectroscopy. So it is also confirming the elemental composition of the inhibitor. Next one is what is the mechanism, main mechanism taking place. So what's mainly we know about how the inhibitor will be acting as a to prevent the corrosion rate. Mainly that is a protonation of inhibitor. So any inhibitor which is dipped in the acid, it is dissolved in the acid solution, it is the protonating of the inhibitor. Due to the protonating of the inhibitor, it will be sharing the electrons pair between the metal surface and inhibitor. So because the metal on the medicinal surface, that is negative charge, on the solution side there is a behind the metal, that is a positive charge. So due to the electron sharing between the metal surface and inhibitor, protonate inhibitor, there is a due to electrostatic interactions. So that's why it is forming it prevent the corrosion rate. And also the metal will also release the electrons to the pi electrons to the bonding orbital, that is antibonding orbital of the inhibitor itself. So that's why due to these 2 types of mechanisms, so there is a inhibition will be higher. So whenever the molecular weight is high and also the presence of rich pi electrons, it will be acting as an inhibitor. So due to the release of the electrons. So that is a okay.
Excellent presentation.
Okay,
And
thank you.
see your conclusion slide also. And
Okay.
I just have only one question.
Okay,
You the mixed inhibitors of phenol, formalin, melamine, urea, and how you will just conclude with the sharing of electrons between metal surface and inhibitor. In all these chemicals, which chemical is going to share the electrons with the metal surface?
So inhibitor actually it will be actually protected by the protease of inhibitor. So it is due to the protease of inhibitor. So the inhibitor molecule is sharing the electrons between the metal surface and inhibitor.
Okay, okay, sir. Thank you.
Donor acceptor interactions. Okay, thank you. Thank you.
Thank you very much, Tirupati sir. Ma'am, shall we move on to the next participant, ma'am?
Yes, sure.
Yeah. I just now call upon oral presentation number 21, Dr. A. Kalaivani. Kalaivani, ma'am, are you there? Kalaivani, ma'am, we are not able to hear you. Can you just unmute yourself, ma'am? Kalaivani, ma'am, we are not able to hear you.
Hello.
Hello.
Ma'am, am I
Kalaivani, ma'am?
audible now? Am I audible?
Yeah, you're audible now, ma'am. You can just proceed with your presentation.
Yes.
Yeah. Thank you.
Good afternoon, ma'am.
Good afternoon.
Good afternoon.
I'm Dr. A. Kalaivani from Savita Engineering College, ma'am. First, I thank the organizers for giving me the opportunity to present my research work in this conference. My research work is synthesis of nanocomposites based on cadmium selenide quantum dots and graphene oxide. This nanocomposite is utilized for sensing L-DOPA. And in the introduction part, the quantum dots are zero-dimensional nanomaterials having 0 to 10 nanometer size. The unique electrical and optical properties make them attractive to use in the field of electroanalytical chemistry. Graphene oxide is another carbon-based nanomaterial used for the sensor. Graphene oxide is a single atomic layered carbon material that is formed by the oxidation of graphite. It is an ideal material for electrochemistry because of its excellent electrical conductivity, high surface area, and low cost. The nanocomposites are formed by mixing 2 or more nano-sized material. Here, for the preparation of nanocomposites, I've taken cadmium selenide quantum dots and graphene oxide. Fabrication of sensor on nanoscale leads to greater sensitivity and conductivity. The analyte chosen here is L-DOPA. It is the most widely used drug in the treatment of Parkinson's disease. Prolonged use of this disease leads to nausea, vomiting. So it is necessary to determine the amount of L-DOPA in the blood sample. Coming to the experimental part, initially I have synthesized cadmium selenide quantum dots by using the already reported procedure. Here, L-cysteine is a precursor and the color of the quantum dots are red-orange. The synthesis of graphene oxide by modified Ammas method, and I've chosen this procedure for the synthesis. Next, for the preparation of nanocomposites, I've taken 60 microliter of cadmium selenide quantum dots and 0.5 milligram per ml of GO in ethanol, and it was ultrasonicated for about 30 minutes. At last, I got a nanocomposite. For the fabrication of modified electrode, I've taken about 5 microliter of cadmium selenide nanocomposite. It was drop casted at the paraffin impregnated graphite electrode surface, and it was dried and it is used for further applications. In the results and discussion part, initially the cadmium selenide quantum dots was characterized by using UV visible spectroscopy. The lambda max at 254 nanometer confirms the formation of cadmium selenide quantum dots. Next, it was characterized with the help of transmission electron microscopy. It shows the particles are spherical in nature and the size of the particles are 3 to 5 nanometers. In the EDAX spectrum, the appearance of cadmium, selenium, sulfur, carbon, nitrogen, and oxygen peaks confirms the formation of cadmium selenide quantum dots. Next, the prepared graphene oxide was characterized with the help of field emission scanning electron microscopy. It shows that the graphene oxide sheets are very thin taper, and it is having larger surface area. And the next image is the HRTM image of the nanocomposites. Here we can able to see the quantum dots are accommodated on the graphene sheets. The modified electrode was characterized with the help of cyclic voltammetry. And this figure shows the cyclic voltammogram of A bar electrode, B cadmium selenide modified electrode, C GO modified electrode, and D nanocomposite modified electrode. Compared to all the electrodes, the modified electrodes, that is the D electrode, shows the greater current response because the nanocomposite has good electrical conductivity and higher surface area. Next, it is characterized by electrochemical impedance spectroscopy. The charge transfer resistance value of the modified electrode is low. So here the modified electrode shows higher electron transfer property. So we have taken this as a good electrode for further applications. Then the effect of scan rate was also studied. The modified electrode shows increased current response when we increase the scan rate. This shows the stability of the modified electrode at all the scan rates. Next, the electrocatalytic oxidation of L-DOPA was done with the help of cyclic voltammograms, and this figure shows the CVs of bar electrode and the modified electrode with the analyte L-DOPA and without the analyte. Compared to the bar electrode, the modified electrode senses L-DOPA at a very lower potential of 0.1 volts with increased current response. This is the differential pulse voltammograms of the modified electrode for the successive additions of L-DOPA in 0.1 molar PBS buffer. And finally, we have done the sensing in stirring conditions with the help of amperometry. And this figure shows the amperometric response of the modified electrode for the successive additions of the analyte. And the modified electrode shows increased current response for each addition. And coming to the conclusion, I have prepared a novel modified electrode, and the modified electrode was studied by HRTM and FESIM, and it was electrochemically characterized by using CV and DPV. The proposed sensor was used for the determination of L-DOPA, and the nanocomposite shows increased current response in amperometric studies. The modified electrode exhibits very good sensitivity, selectivity, and reproducibility towards the determination of L-DOPA. Thank you.
Thank you very much, Dr. Kalaimani. I may now pass on the session to Dr. Udekla, ma'am.
Yes, it's an excellent presentation, Dr. Kalaimani.
Thank you,
And
ma'am.
one question with you.
Yes, ma'am.
And this L-DOPA is an amino acid, right?
Yes, ma'am.
So, how it is possible, or just explain the synthesis of this L-DOPA, you just combine with the CDSC, how you do that?
Ma'am, I can't get your question, ma'am.
Actually, you are just, it's a determination of L-DOPA by CDSC, right?
Yes,
Quantum
ma'am.
density.
Yes, yes.
And do you mix this amino acid with this cadmium CDSC?
No, ma'am, no, ma'am. First, I've synthesized the cadmium salide quantum dots and I poured it on the surface of electrode. And the analyte was taken in the solution form.
Fine, fine. Okay, good.
Thank you, ma'am.
Yes.
Thank you, Dr. Kalevani, ma'am. Ma'am, shall we move on to the next participant, ma'am?
Yeah, sure.
I may now call upon our oral presenter number 22, Mr. Aravindan Santan. Sir, are you available?
Hello.
Hello. Yes, sir, you're audible, sir. You can start with the session.
Can I start it up?
Yes, sir, you can start.
Yeah, sure. I'm just sharing on my screen. So, ma'am, is it evident to you? Can you see me?
Yeah, we can see you, sir. We can see you.
Sure.
Yeah.
Thanks. So, okay then. This is Aravind. I'm from Taiwan, National Taipei University of Technology. So, okay. So I just going to present about the design analysis of 3D flower like CuA4, which is anchored on nickel foam, and its electrocatalytic determinations for environmental hazardous 4-aminophenol. So I'm a student of Professor Hua. She is the director of Department of Molecular Science and Engineering from National Taipei University of Technology. So this would be my outline, a schematic illustration how it can be, and importance of cerium vanadate and nickel foam, where it's basic characterization like morphological characterizations of SEM and TEM, and electrochemical studies. Electrochemical studies varies with DPV, concentration, scan rate, and different pH. So how we have done, I'll be showing in this presentation. So in brief, as I said about schematic illustration, here you can see we have chosen a nickel foam which we have purchased. From that nickel foam, CuA4 is being designed. So I'll get back to this later, where I also means why we have chosen about CuA4, what's the importance of CuA4. Among rare earth metal vanadate, CuA4 is considered one of the most important facilities because cerium comes under 4 electronic configurations varying with a large atomic radius of 181 m, where the electronic duty is 1.12 angstroms. Multiple oxygen states goes around +3, +4, +2, and +1, where cerium vanadate is considered as a theoretical capacitance is comparatively high, and it has also enhanced, and it is also applicable as a good biological compatibility with high electric conductivity. Above all these, when 3D dimensions, 3D architectures are very good electrochemical behaviors as they carry large number of transportations, and specific high charge and condensivity will be very high, and comparatively the enhanced electron transport efficiency will also be quite large. So in this aspect, we have chosen cerium vanadate, and after vanadate, electrode selection was one of the major tasks in electrochemical analysis. So we have chosen nickel foam as a first work of this. We have chosen nickel foam. Commercially, we have purchased nickel foam from Tavian, means a place from Taipei, by Wang Ha Industries. Through Wang Ha Industries, we have purchased this, where we used to have a collaboration, so we got from them. The nickel foam is high electronic conductivity, and it has good 3D structure, where it acts as an electrode substrate material. In this particular topic of choice. So the diffusion resistance among the electrolytes will be very high. It also enhances the ion transportations and also maintains very smooth electron pathways, which is very rapid. And whereas the charge and discharge reactions will also be very high. So we have chosen nickel foam. So how we synthesize CuO4 and how we have anchored them on it. So in a typical synthesis, sodium ammonium metavanadate and cerium nitrate. Both we have mixed in 35 ml of distilled water, making it as a stirrer, making it stir, and when the stirring is continued on, it turns into gold. And then were added drop by drop to this mixture, and after that, at certain temperature, Ag plays an important role. After uniform mixing, the whole golden level was transferred into a hydrothermal autoclave for about 12 hours. So after that, calcination is being done, where the material is being dried. Later, nickel foam has taken, and nickel foam is inserted into grid and autoclaved for another 140 degrees centigrade for about 6 hours to form a material. So how this material is formed, we have I have listed here clearly, saying that how CuO4 is anchored onto the nickel foam, and how the electrochemical detections of 4-aminophenol we have done in bovine serums as a real samples in bovine serum and distilled water and tap water. So to this, before I go into the electrochemical determinations, first in this, yes, you can see A and B are the nickel foams. A is a simple nickel foam, and C is a nickel foam with which cerium vanadate is anchored onto it, and D and E continuously. F, you can see about single helical structures of flower-like 3 cerium vanadate, where the TM image of cerium vanadate is given in G and H, showing how the petal-like structures, means the petals morphological analysis, how cerium is being in this. And continuation is this 4-aminophenol. What's the importance of 4-aminophenol? 4-aminophenol is primary and hydraulic degradation product of paracetamol, and it is widely used as an antiseptic and analgesic drug, as paracetamol is an effective and safe agent to relieve mild to moderate pains. Although 4-aminophenol with significant nephrotoxic and tetrogenic effects, it can be detected in pharmaceutical preparations. So the degradations of paracetamol as a synergetic intermediate is more high due to its toxic content. So in this, so in order to sustain the high toxicities, the maximum content of 4-aminophenol in pharmaceutical is limited to 50 ppm, like about 0.005 percentage by the European and the United States, whereas even in the Chinese pharma companies also, it is listed as 0.005, whereas the limit, if it is exceeded, there will be a very serious way of considerations in health regardless. To overcome all these issues, still there is a need of developing a good sensor with all the basic intimidation, basic lens tints. So we have chosen this. We have chosen 4-aminophenol for electrochemical sensing. In this, in figure A, I have mentioned about how the variations. As you can see in this, the dotted line represents the bare electrode, bare nickel foam, bare electrode, and CVO4 with anchored on nickel foam and nickel foam electrode separately, and CuO4 with nickel foam electrode, where the current representations is being given in the chart diagram representing how the variations is happened in this, where the aminophenol peaks gradually started increasing with a separation of 4-aminophenol with a high scan resolution RS, and the determinations with the retention time, where the analytes has been derived with an average peak of WD, in which is statistically increasing at 0.14, whereas the calibration part plot was also being shown for this. And in C, you can see different pH and how the different pH. pH is also considered as one of the major tasks in finding. In context of separations by ion change, the pH is only variable that directly affects the results in the search of a baseline preparation in respective of the chromotherapeutic bands or chromatographic bands. In this way, we have considered pH as an evaluant way, which we have studied in between minus 0.30 to 0.75 with different pHs ranging from 3 to 7, like 3, 4, 5, 6, and 7 variations. So in these, the sample enhancements, the resolution graphs, and the average peak variations is also been calculated, where the pH indicating that the pH has been most importantly separation process, where the range concentrations, as you can see from pink uh, broad peak, which is pH 3 to 7, gradually increased. So pH 7 we have considered as a perfect ambient pH for electrochemical sensor, and further studies were performed in this. I'm sorry for this. As you can see in this slide, we have done the different addition, the voltammetric additions. We have where the additions it has been done. How the electrochemical behavior varies with respect to addition. Like addition, we have taken from 50 microns to 50 micrometers to 450. The incidence of 4-aminophenol is being added with the 0.1 mole of 4-aminophenol onto the CEV of a nickel for modified electrode, where upon the increase of the material, upon increasing of the 4-aminophenol, gradually the current also started to excite on with the variations from about 3 to 9.5 microampere, where for to it how much addition we have been done means up to 440, how the current has been increased, the concentration of 4-aminophenol versus the reduction current, how it has gradually increased with is given in figure B. Whereas to that, the same scan rate is being performed with the different scan rate ranges from 0.02 to 0.20 volts per second at 100 microliters of 4-aminophenol in pH 6. As we mentioned earlier, in pH 6 we have taken, and the current is being mentioned. The current is being gradually increased from for 0.2 to 0.20, where the plot is also been given in figure D, which is so the mechanism in which the linear proportional to the scan rate, the linear proportional between current is being expressed over here, where this really indicates that the electrochemical reaction of 4-aminophenol onto the prepared film is a surface controlled process at a high scan rangings, and this with the linear regressions APA and EPC with an algorithm scan rate says that a perfect scan events is been uh, evident in this. And whereas continued, the effect of the solution and the effect of the rangings has been continued for this study, where the how the redox reactions of 4-aminophenol has been happening with the two proton coupled under two electron process can also be studied out of this, where the electrochemical, the electro-oxidation mechanism of 4-aminophenol can be understood, where due to the time restraints, we are not presenting much into this. Whereas to confirm for the ultrametric studies for DPV, differential pulse voltammetry, is being used, is being carried out in this detection, where despite the DPV, various concentrations of 4-aminophenol is being spiked onto the material, means the peak currents increases linearly against the concentration of 4-aminophenol within the range from 4.99 micrometers to micromole to 2949, where the increased current shows the calibrations between how a 4-aminophenol has segmented. In this, it is evident that the current has been gradually increased with the combinations, showing that a good sensitivity, as I could not mention over here, the sensitivity conditions. Hello, ma'am, can you hear me?
Yes, sir, we can hear you, sir.
Okay,
Sir, actually, we are, your time is over. Can just conclude it,
sorry
sir.
then. I'll just continue. I'll just
let's just conclude it,
the,
sir, please.
So thank you. Sorry for that. Means I could not see. Sorry for that. So continuing on, the real sample analysis was carried out in industrial water and tap water and a bovine serum, where I have given the comparisons correctly. And in a brief conclusion, like a simple hydrothermal procedure is being used for the preparation of CUF or nickel foam to form a nanocomposite onto the nickel foam for electrochemical determinations. Electrochemical determinations for the hazardous 4-aminophenol, where the detection limit is 0.005 micromole and with a wide linear range of 2949 with a compromising sensitivity of 5.2. So furtherly, we are thinking about to continue on this for photocatalytic activities and extensive detections in paracetamol and as a simultaneous detection. And that's it. And thanks very much.
Okay. Fine, sir. Excellently presented.
Thank you.
That
Thanks a
we
lot.
don't have any questions. That much clarity you have presented. And I think, Sangeeta madam, we can proceed with next participant.
Yeah.
Okay.
I may now call upon Dr. Naresh Muthu, oral presenter 23. So
yes,
kindly do that you just stick on to the time, sir.
Okay, ma'am. Sure, ma'am. Sure, ma'am.
Thank you, sir. You can just carry on.
Okay. Good morning to everyone. Today, now I'm going to present my part of the research work. This work is contains two major objectives. One is to optimize the electrode, and another one is optimize the cations of the electrolyte for supercapacitor application. This work was carried out at IIT Bombay during my postdoc. First, my major introduction of my project research is the major challenge the world is the increasing the partial pollution and the decrease of the fossil fuels. So in this solution, we have to record to find out the renewable energy sources. So we can use solar and wind, but the major challenge is storage of energy. So this is the Rahan plot, that is, it's contains the different types of energy storage devices, that is the lithium-ion battery, pseudocapacitors, and capacitors. From this figure, we can see the battery having the high energy density, but it's having the low power density. But in the case of capacitors, it's having the high power density, but it's having the low energy density. But the supercapacitors are bridging the gap between the capacitors and lithium-ion batteries. So the researchers are focusing on the on supercapacitor. And also the supercapacitors also classified into two types. They're based on the working mechanism. One is double layer capacitance, another one is pseudocapacitance. Double layer capacitance means nothing but the charge will stored on the surface of the electrode. That is the figure 1 showing the charge on the surface of the electrode without any charge transfer reaction. But in the case of here, that is another capacitor, that is a pseudocapacitor. Here the charge storage mechanism, that is the electron will be transferred between the electrode and the electrolyte. So this is called as the pseudocapacitance. So this is also called as pseudocapacitance. So based on this, my objective is to prepare initially I prepare the CO304 RGO nanocomposite, because this is a schematic diagram for the preparation of my electrode material. This is the structural analysis, that is initially confirm the formation of CO3 RGO nanocomposite. That is the RGO express the two peaks, one is the 002 and another one is the 004 peaks. So these two peaks confirm the formation of RGO, and the CO3 will show in the major peak of 6 or 7 peaks. These peaks belong to the JCPDS number of 421467, that is the phase centered cubic phase. And also in the case of the composite showing the exhibit the both RGO and CO3 nanocomposite peak. So it's a confirm the formation of RGO CO304 nanocomposite. This is the XPS spectrum. This is the XPS spectrum, the high resolution spectrum of RGO exhibits the carbon and oxygen peak alone. But in the case of CO304, it's showing that CO2 status, that is 3/2 and 1/2, that is 2 multivalent ion status of carbon dioxide. This is the high resolution spectrum of the CO2p. So it can be fitted with the origin software. So it's showing the difference between the two ion state, that is a valence ion state, is at around the 15 electron volt. So it's showing that the single phase formation of the CO3O4 nanocomposite. This is a different functional group, that is during the graphene oxide, different functional group also attached to the surface of the graphene. This also confirmed from this high resolution spectra. So based on this, we can confirm the formation of CO3O4 orgio nanocomposite. After that, we are the estimation for the supercapacitor application. We have to prepare the electrode. But here I use the solution stopping method for the using the electrode, and also use the 3 electrode system for this. So initially, this is a CV spectrum of CO3O4 RGO and CO3 RGO in KOH electrolyte. Here we can see the RGO, there is no, that is orange color indicate there is no in the oxidation peak. But in the case of CO34, we can see the 2 oxidation peak and 2 redox, that is 2 redox peaks are observed, that is A1 and A2. But the case of CO3O4 RGO, we can see the double A, that is the extension of green color in the range, and as well as we can see the 2 peaks, that is the combination of pseudo capacitors and double layer capacitance. Why these 2 peaks are arising? That is the oxidation of the CO3, that is the metal ions. It's getting the 2 multivalent ions, that is that CO3O4, which absorbs on the OH ions. So it's the form the COOH ion. This is the figure A. It is due to we are using the KOH electrolyte, but in the figure we are using the NaOH electrolyte. Both looks like similar because both are the same anions. Only the cations is the different. The anions is the charge transfer reaction will be happen the same. But this is that is the galvanic charge discharge curve. It's showing the 2 region for the case of RGO and CO3O4 RGO. That is the first region. When we increasing the potential, the time also the increasing in the steady state. After that, it's not constantly increased. That means it will be some oxidation reaction will be happen. That will be the coincide with our CV. So the same case will observe for the both KOH and NaOH. So, but the capacitance also we are estimated, but we can estimated the maximum specific capacitance observed for the case of KOH as compared to the NaOH. That is the KOH showing the specific capacitance 396, but in the case of NaOH showing the 277. So it's may why, and also you are studying the stability. The stability of the RGO also showing the maximum stability of 87.8%. And as compared to the pure CO3O4, because pure CO3O4 only showing the stability of 69%, because the materials will be maximum charge transfer reaction will be happen in the case of pure metal. But when we composite with the double layer capacitance material, the stability will be increased. And also we can see the KOH is the more specific capacitance as compared to the NaOH. That's maybe it's due to the low hydration spheres of the K plus ions. And also this is the Nyquist plot. The first figure is Nyquist plot, and another is the product plot. This is the Nyquist plot. We can see the high resolution peak. See here the RGO showing that the orange color is RGO is there is no semicircle, but in the case of CO3 we can see the broadness of semicircle, and another and the RGO CO3 nanocomposites very less semicircle. That means what? The semicircles indicate the charge transfer reaction. So in the case of RGO there is no charge transfer reaction. There is no material. There is no ions in this case. But in the case of CO3, here the more multivalent ion, so there is the more charge transfer reaction happen. When we composite both will them, so the charge transfer reaction will be less. So these also we are estimated numerically with the fitting the ZB software, that is the equivalent circuit, and then we are estimated and the calculated the RGCT value and solution distance, and it's a tabulated. And also this show it is the so this is the body plot. It's giving the relaxation time. That is the recharging of time of supercapacitor, but the composite is very less as compared to that CO3O4. So this is the same case for the NaOH we are also observed. So there is no difference between both of them, but the numerical value of charge value and the solution resistance is very less as compared to the KOH. So why this?
Dr. Naresh Muthu sir, yeah, just kindly conclude it, sir.
Yeah, it's concluded. Yeah. This is the Rocken plot. This is the Rocken plot. We can fitted with the we are estimated the energy density and power density. So it is in the gap between it's having the higher power density as well as its energy density. And also we are expecting for its the practical application. And also we are now fabricated for the device fabrication. Also it's also going on. So the major conclusion of this work is the supercapacitor is basically depends upon the electrode as well as electrolyte, because we are optimized electrode that is RGO, CO3 for RGO, which is better. And the same thing we are also optimized a different kind of electrolyte, that is KOH and NaOH. And also we are estimated what is the role of the KO cations. that is Na plus and K plus ions. So from this, I concluded that KOH is a more suitable electrolyte, and also CO304 RGO is a more suitable electromaterial for supercapacitor application. Thank you.
Thank you, Dr. Naresh Muthu, sir. Now on to Dr. Piyudekla, ma'am.
Okay.
Excellent, sir.
Thank
You
you,
have been
ma'am.
clearly explaining about all the electrolytes and electrode interactions.
Yes, ma'am.
And of course, I just had one question. Have you used any software for simulation study here?
Here we can use my stimulation study, but in institute, in my department, we are only focused on the practical application, because now here I use the 3 electrode system. So here, so we are now we are considering the 2 electrode system also for the practical application. 2 electrode system means nothing but we are fabricating a supercapacitor device in our lab for the practical applications. So we are only oriented to the practically, because initially we are checked out which electrode and which electrolyte more suitable. After that, we are going to forming the electrode. That is a practical applications. So we are mainly focusing on electrode systems.
Thank you, Dr. Naresh Muthu. Ma'am, can I call upon the next participant? Yeah. I may now call upon oral presenter 24, number 24, Dr. K. Rajkumar.
Hello, ma'am.
Yeah, yes, sir. We can hear you, sir. Rajkumar, sir? Yeah, you can start with your present. So kindly stick on to time, sir.
Yeah, sure.
Yeah, thank you.
Is my slide visible, ma'am?
Yes, sir, it's visible, sir. You can proceed.
Good afternoon to all the participants. My title entails engineering the composition of sodium titanates for high capacity sodium ion batteries, which is done under the mentorship of Dr. Sudhakar Chandran, Associate Professor, Department of Physics, IIT Madras. The aim of this work is to engineer sodium titanates to obtain low voltage, high capacity anodes for sodium ion batteries. The outline of my talk goes like this. Increasing demand of energy consumption and depletion of fossil fuels has led our mankind to think of harnessing the renewable energy resources like solar, wind, and tidal power. Major challenge in harnessing these renewable resources is that they are scattered, unpredictable, and intermittent. Therefore, the energy produced by these energy resources has to be stored in the form of chemical energy. So, while the lithium battery research is still on to achieve higher performances, a question arises why we need sodium ion research. Lithium ion source is very scarce, therefore costly. Sodium, being the second abundant element in the Earth's crust, is also cheap, and the electrode potential is close to that of the lithium metal. Therefore, sodium is an alternate for the lithium. So, working of a battery consists of anode, cathode, which is separated and dipped in an electrolyte. During charging, when you supply electrons from to the cathode, the lithium ion from the cathode deintercalates and intercalates into the anode. While discharging, the lithium ion deintercalates. As a result, the electron flows to the outer circuit. The main battery parameters are listed here, which are specific capacity, charge discharge rate capability, cyclability, etc. Coming to the material of my study, which is sodium titanates, it has a general formula of A2TiN O2N plus 1, where the high alkali metal content possesses layered structure and the low metal content possesses tunnel structure. That is, Na2Ti3O7 possesses layered structure, which is shown here, and the Na2Ti3O13, which is tunnel structure, is shown here. These are the experimental details. Sodium titanate were prepared by hydrothermal method using TiO2 and NaOH. The experimental conditions are shown here. And there are different experimental conditions: pure sodium titanate with a layered structure and tunnel structure are obtained, and under different pH conditions, composition of these sodium titanates were also obtained. The obtained pure phases and its composition are tabulated here. The active material is coated on the copper foil and is cut and fabricated into coin cell. The coin cell fabrication details are listed given here. So the powder XRD pattern matches with the standard patterns of layered and tunnel structures, and it is shown here. The ritual refinement of the powdered XRD pattern confirms that the structure and the phases estimated are as given in the above slide. So the pure phase tunnel and layered structure and its composites with the different compositions are obtained successfully. The transmission electron microscopy images shows that the morphology of the obtained samples are in rod morphology, and the SED pattern confirms that the patterns are dot pattern, and it reveals that they are highly crystalline in nature. From the cyclic voltammetry curves, the redox potential of layered phase is low compared to that of the tunnel phase. The redox peak of the composites shows that it consists of both the redox peaks of layered and tunnel structure. The charge discharges at various C rates were performed for all the samples, and it is plotted here. Here, this graph shows the specific capacity versus cycle number for the tunnel and layer structure, where it can be seen that layer structure possesses higher capacity. Overall study of the specific capacity versus cycle rate for all the compounds, it is seen that 20% layered sodium titanate and 80% sodium titanate, which is tunnel structure, has higher capacity for all the different charge discharge rates. When you see the cyclability, when you check the cyclability, the pure at low C rate, the tunnel structure possesses higher capacity. When you compare overall all the samples, the layered structure with 20% composition and 80% of sodium titanates, which is tunnel structure, possesses higher capacity and cyclability compared to all the samples of pure, even with pure phase. So this is the cyclability at a higher C rate. Even here, the composites with the 20% layer structure and 80% tunnel structure possess higher capacity and cyclability. So coming to the conclusion, pure phases of sodium titanates and its composites with a different composition were successfully prepared by one-part hydrothermal method. Ritual refinement carried out estimated the percentage of tunnel and layered phases in all the samples. The rod morphology was observed in the TM samples, and it is found to be highly crystalline. From the charge discharge results, individual phases depicted less capacity, while the composites with 20% layered Na2Ta3O7 and 80% tunnel structure, which is Na2Ta6O13, has high capacity of 180 milliampere hour per gram, which is about the theoretical capacity, and has good cyclability rate of 87% at low C rate and 96% at high C rate. So this can be this composite with 20% layered and 80% tunnel structure can be a good candidate for a low voltage sodium ion batteries. And now I take this opportunity to thank the organizers of Bhaktavakchanam College for giving me this opportunity. I also thank my mentors, Sudhakar Chandran sir and Professor R.T. Rajendra Kumar sir. I also thank my funding agencies, DST, SERB for providing NPDF, and DAE, SERB, DAE BRNS for providing me JRF and SRF. Save energy and store energy. Thank you for your attention.
Thank you very much, Dr. Rajkumar sir. Now on to Dr. Odekla ma'am.
Sir, it's an excellent presentation. And I just have one question. You have studied charge and discharge results you have given.
Yes.
I just want to know which is the charge carrier here in the sodium titanate.
Yeah. The sodium ions is the charge carrier, ma'am. The sodium from the electrode material, which is sodium titanates, it gets released from the sodium structure, sodium titanate structure, and it intercalates into the other counter electrode. While discharging, what happens? it releases from the counter electrode and it gets back into the structure.
Okay, excellent, sir. Thank you.
Thank you very much, Dr. Rajkumar. Can I call upon the next participant now?
Sure.
Yeah, just call upon oral presenter number 25, Dr. Revati. Revati ma'am, you're not audible. Revati ma'am. Revati ma'am, can you just unmute yourself, please?
Okay, ma'am.
Yeah, we can hear you, ma'am. You can proceed.
I'm Revati, former National Postdoctoral Fellow, did under the mentorship of Dr. R. Selvakumar in PhD IAS Coimbatore. My work is preparation of MPS modified manganese dioxide based nitrate sensor and their future perspectives. Why we need to detect the nitrate? There are two types of nitrogen in drinking water in terms of nitrate and nitrate. So nitrate interferes with the oxygen carrying the capacity of blood and leads to the shortness of breath and the blueness of the skin due to the presence of carcinogenic product. There is an additional concern that more serious bacterial or viral pollution may be likely to occur in the future. Therefore, the development of accurate and sensitive and simple methods are required for the nitrate detection for our environmental applications. There are a number of standard level of nitrate and nitrates available who announced that less than 3 ppm was permissible level of nitrate intaking in the daily life. So we have to concern this value in our daily life. There are many number of detectors are available commercially to date, which based on the colorimetric and the spectrometric methods. These commercially available detectors are worked by mostly grease reaction using toxic components such as sulfur-lanolic acid and aniline-based component. These components give the excellent detection limit against nitrate, but they are still suffer from the interfering capability, such as sulfate and other heavy metal ions. So in past decades, researchers replaced some components using bio-nanomaterials. Therefore, we have some challenges here in the area. We need to replace this kind of toxic material with biocompatible and low-cost material, and need to improve the selectivity and also produce the sensor which capable of simultaneous determination of nitrate and nitrate. Nowadays, there are a number of materials including metal oxides and other inorganic components are used for nitrate sensing, but still that could be suffer from simultaneous determination of nitrate as well as nitrate. Here we choose for that we focused on in this work. Here we choose the manganese dioxide, one of the abundant material in the earth and having the excellent electrocatalytic activity as well as the molecular adsorption characteristics. So here we believe that in this work, we believe that polymer modification may enhance the electrochemical activity. Further, we choose one of the polymers such as mercaptosuccinic acid, which contain the hydroxyl and diol functional group, which may act as a charge carrying transfer agent during the electrocatalytic activity. Further, here we functionalize the surface of manganese dioxide via chemical precipitation method without using any binders or mediators. There are a number of techniques are available, but they are suffering from some stability issues and the sample procedure and then easy to use in real life, daily life. So to overcome these issues, we choose the electrochemical method, which is a simple and low cost technique. At first, as prepared piston nanorods were characterized by XRD Raman and FTR analysis and confirmed the formation of alpha MnO2 nanostructure within the crystalline range of 9 to 30 nanometer. And also, to find out the surface area, BET analysis was done. That was the type 4 mesopause with a diameter of 14.87 nanometer. Then morphological was evaluated using the same also HRTM analysis. Here notice that pristine MnO2 was the C ocean shaped MnO2 nanorods. After modifying with the MPS, that shows the some agglomeration was found, and also they lose the some Uchin like structure. But in TEM image we observed the rods itself. This may confirm the MPS will covered over the surface of MnO2. Further, the electrochemical analysis was started with cyclic voltammetry analysis using pristine nanorods. This will be done at different scan rate up to 200 milliwatt per second, and noted that this can be stable over the high scan rate. And also here noticed that, interestingly noted that pristine MnO2 stores a sensing ability against NO2 than nitrate. But however, this gives favor to the nitrate, thus will be over the higher potential range. Further, we used the modified MnO2 and compared with the pristine MnO2. Here noted that polymer modified MnO2 shows the 2 times higher peak current value than the pristine MnO2. So this may be increase the electrocatalytic nature against the nitrate. Further, to analyze the trace level detection of nitrate, we move on to the pulsed voltammetric technique. At first started with the pristine MnO2. This gives some changes upon addition of nitrate, but there is no increase in the peak current value. Also that gives the higher potential value, nearly 1 volt. So further tested with functionalized MnO2 cyclic voltammetry. Here notice that we observed some little changes in the peak current value, but not much peak separation was observed. So again move on to the pulsed voltammetric technique. Here noted that on increasing the successive addition of nitrate, the peak current values are increased. Some peak separation also observed. This will be give the detection limit up to 0 to 5.5 into 10 power minus mole. So here in this work concluded that electrochemical detection of nitrate was tested using MPS functionalized manganese dioxide nanorods through pulsed voltammetry analysis, and confirmed that detection ability was higher than pristine and bad GCE. This may be due to the presence of diol and hydroxyl group. This helps to improve the dispersion ability as well as the sensing ability. So future work will focus on to test functionalized manganese dioxide against nitrate with reproducible test and also the sensing mechanism. I would like to acknowledge Professor Radhakrishnan, Director PhD IAS, Dr. R. Selvakumar, Associate Professor PhD IAS, Professor R.T. Rajendra Kumar, Badhir University, and DST SERP for providing the fellowship, and organizers and team members of who conducting this conference, ISCST 2020. Thank you, ma'am.
Thank you, Dr. Revathi. Now on to Dekla, ma'am.
Yes,
Dr. Revathi, you have been suggesting like this method is non-toxic. Whereas you have been comparing in the initial stages, few other methods are toxic.
Yes, ma'am.
Just give a brief thing about where you have tested its non-toxicity.
Still, I'm not tested, ma'am, but from the literature review, observed that, ma'am.
Okay, excellent, madam.
Okay,
thank you.
Okay, ma'am.
Thank you very much, Dr. Revathi.
Yes, ma'am.
Ma'am, shall I call upon the next participant, ma'am?
Yes, sure.
Yeah, can we have oral presenter number 26, Ms. R. Manimori?
Yeah, ma'am, I'm here.
Yeah, you can just start.
Yeah.
Kindly stick on to time.
Sure.
Okay, thank you.
Is it visible, ma'am? Hello?
Yeah, it's visible, ma'am. You can proceed.
Okay. Good afternoon to everybody. Today, I am myself R. Manimoli from Mysore University. Today, I plan to discuss my topic, influence of oxidizer to fuel ratio on zinc oxide for visible photocatalytic degradations. So this is the content I plan to discuss: introduction, experimental section, characterization of nanoparticle, resultant discussion, and conclusions. So now nanoparticle, we know everybody that we can prepare either chemical route or physical route. Among all wet chemical routes, solution combustion process have many advantages like time, energy, efficiency, because we can prepare within few seconds to few minutes, and easily affordable porous materials. The powders are voluminous, foamy, and large surface area, and no need of any sophisticated laboratory equipments also. In solution combustion method, the product mainly depends upon the type fuel, oxidizer fuel ratio, and calcination temperatures. This type of fuel, oxidizer fuel ratio, it is depends mainly main parameter for changing the morphology of the products. So if the oxidizer fuel ratio is maximum, that is more than 1, means we can say that is a fuel lean condition or oxidizer rich conditions. If it is a less than 1, it is a fuel rich conditions. If equal, that is equal to 1, means it is geometrically balanced. Next, coming to the environmental issue, the 4-nitrophenol and its derivatives are used in productions of insecticides, herbicides, and synthetic dyes. And up to some threshold conditions, the nitrophenol in water can cause the irritations of skin, nose, throat, and eyes. And it is hardly degradable because of its solubility and stability in water. So we can choose the photocatalytic degradation, that is advanced oxidation process. It has a main advantages like complete mineralization and no wastage disposal, and we can say it is environmental friendly. So here in the synthesis process, I am taking corresponding metal nitrate and carbohydrate as a oxidizer and fuel. So I am changing the oxidizer fuel ratio from 0.25 to 1.25 in the increment of 0.25. So corresponding metal nitrate, I took zinc nitrate, and carbohydrate, I choose a sucrose as a fuel. So this metal nitrate and fuel we can mix with the water and keep it in a stirrer for 30 minutes to get the homogeneous solutions, and we can keep the solution in preheated muffle furnace for 450 degrees Celsius. After what happened, first it started boiling and it is fully dehydrated and formed the gel position, and further increasing the temperature, it is combustion started. So the first figure shows the combustion time, and second one is the final product. It is a foamy powder. So the stoichiometry of redox mixture of combustion is calculated on total oxidizing and reducing valencies of the oxidizer and fuel using the concept of propellant chemistry. So this is the formula I used. The oxidizer and fuel ratio is total oxidizing and reducing valencies of oxidizer and divided by corresponding of fuel also. So according to this formula, we denoted the valencies of oxidizer and fuel. Small m is the mass of oxidizer and fuel, and capital M is the molecular weight of oxidizer and fuel. So now after preparing this zetano nanoparticles, we can undergo the photocatalytic behavior of 4-nitrophenol. So here we are taking the 20 ppm of 4-nitrophenol and 25 mg of the catalyst. We can mix it and keep it in magnetic stirrer in the dark for getting the absorption desorption equilibrium. After that, we can keep it in solar light for 150 minutes. Every 30 minutes, we can collect the sample, and the concentrations was measured by UV visible spectrometer. The efficiency of degradations was calculated by C0 minus CT by C0. That is C0 and CT is the initial and final concentrations of the nitrophenol. So these are the basic characterization techniques for structural and morphological behaviors. So from XRD result, the powder have a highly crystalline and wet-side structures. When we calculated the crystallite size using the Scherer formula, the crystallite size is varying inversely with the oxidizer fuel ratio, because when we increase the oxidizer fuel ratio, the disintegration of agglomerate happen as more gases are released and heat is taken away from the system, which hinder the particle size. But after the stoichiometrical ratio in the oxidation fuel ratio were 1.25, the crystallite size is increased to 14 to 31. That is because of the excess of flame temperature, which assists the crystal growth also. According to FESM, we can find the porous like morphology of our sample. So next, the band gap energy was calculated using the UV DRS spectra. Here I am using the Kobalka-Monk method to calculate the band gap energy. So the band gap energy for a stoichiometrically balanced one is 3.1 electron volt, and for other ratio it is 3.2. And according to the PL spectra, there is a broad peak visible in the PL spectra, which enhances the photocatalytic activity, because the broad peak denoted for oxygen vacancy is more. So the higher degradation efficiency of nitrophenol was achieved 1.25, that is a fuel lean condition or oxygen rich condition, due to high absorptions of visible light. Also large porous surface area enhances the electron hole separation and increases the yield of hydroxyl radical. For other ratio, there is a very small variation only we can found for O/F ratio, that is for stoichiometrically equal, that is 95% only. There is no much difference. So finally, I will come to the conclusions. The nanoparticle that is prepared by the solution combustion method have a highly porous nature because of excess gas released during the combustions. So these large porous surface area enhance the whole electron hole separations and increase the yield of hydroxyl radicals. And one more thing, because of more yield and less for a stoichiometrically balanced conditions, we will get the more yield also and less band gap, and crystallite size also very less. So these sample we are using for our further work. Thank you, ma'am, for giving me the opportunities. Thank you.
Yeah, thank you, ma'am.
Okay.
Yes.
Now on to Dr. Udaykala, ma'am.
Yeah, it's an excellent work.
Thank
And I was about to ask you about this porous morphology of your structure, and that was explained in your last slide. Of course,
yes.
good.
Okay, thank you. Okay.
Ma'am, shall I call upon the next participant? Yeah. Can we have our next presenter, oral number 27, Mr. Vignesh? Ma'am, I think he is not there. Can we move on to the next participant, ma'am? Yeah, I just now call upon oral presenter 26, Mr. Mani Mohan. Sir, are you ready?
Yes, ma'am. Good afternoon.
Yeah, yeah. Good afternoon, sir. You can proceed with your presentation, sir.
Yes, ma'am, sure.
Yeah. So kindly stick on to time.
Sure, ma'am.
Yeah, thank you.
So good afternoon to all connected here. I am Mani Mohan, doing PhD full-time chemistry under the guidance of Dr. Abubakar Siddiq, Islamic College, Autonomous, Vaniambadi, Tirupattur. I elected the catalysis in the broad field for the virtual conference. Now I am going to present the topic is synthesis and characterization of novel copper-2 anchored biopolymer complexes as reusable material for photocatalytic degradation of methylene blue. And these are the contents we are going to discuss one by one. Introduction. Untreated dyes expelling from the industries causes severe environmental disaster, and this leads to various pollution, at last cause desertification. Therefore, the dye containing hazardous substance should be treated before the dispersal. So it is very important to treat dyes before the dispersal. So many physicochemical techniques are utilized for the effluent treatment process, which are shown in this figure. However, these techniques often low efficiency with a high capital cost. On the other hand, using biopolymer metal complex as photocatalyst works more efficiently towards dye degradation, also often completes eliminations of hazardous substance from the environment. This is the polymer chitosan and chitin, and we are getting the chitosan from chitin by deacylation process. Scope and review of this work. Biopolymer ship base is a type of ship base which differs from the usual ship base. Possess advantages such as biodegradability, low cost, easy availability, biocompatibility, non-toxicity, water soluble, and eco-friendliness due to the carboxymethylation. And these aspects of these biopolymer ship-based compounds are quite unique from the usual ship bases. The literature reveals that no considerable work seems to have been done so far the biopolymer photocatalyst for methylene blue dye degradation. Hence, we decided the biopolymer ship-based copper complexes can be used as novel photocatalysts for methylene blue dye degradation process. And these are the materials and the reagents we are used for the synthesis. These are the spectroscopic techniques were employed for the preferred samples. And the synthesized biopolymer copper-2 complexes were utilized as a photocatalyst, anti-inflammatory agent, and anti-diabetic agent. An experimental part, the experiment, the preparations of ship-based ligand and the copper complex. This slide showing the preparations of biopolymer photocatalyst. And next part, results and discussion. First, I am going to explain about elemental analysis. In this table, showing that DCS carbon-nitrogen ratio is 5.19. And this is the DD of the polymer is about 98%. Degree of deacylation is 98%. In ship-based, the carbon and nitrogen percentage values are carbon percentage values are which are higher than the complexes, which confirm the formation of metal complexes. FTIR analysis confirmed the presence of functional groups, amide and amine, and formation of ship-based ligand and the complexes. H1 NMR and C13 NMR were results. The results reveals the formation of ship-based ligand. So here we have a ship-based imine at the delta value, canonical delta value 5.96, and hydrazide, secondary hydrazide, means the canonical delta value 10.18. Similarly, for the carboxymethyl group, whose ppm value in C13 NMR 191.36 ppm. For ship-based imine 171.20 and 164.8 ppm. And these are another spectroscopic analysis for non-identification of metal complexes. Here we have UV visible analysis and VSM analysis to confirm the formation of complexes and as well as identify the geometry of the complexes, and whose magnetic moment value 1.87 and 1.83 for those metal complexes respectively, and molar conductivity measurement for the metal complexes 19, and as well as for another one more metal complex is 16, and which conducts very poor in conduction, and it is non-electrolytic in nature, and which confirmed it is a square planar geometry. And next, various XRD peaks reveals the presence of corresponding compounds from the values. And next, thermal degradation, that is TGA DTA analysis. Various thermal degradation curves reveals the presence of respective compounds. These are the same images, and these images prove the respective compounds. And this is redox spectra, which reveals the presence of respective compounds based on their atomic weight percentage. And this is ESR spectra, which reveals the presence of copper metal ion and confirms the geometry of the complexes. And next, photoluminescence spectra. So photoluminescence spectra is also proved the formation of complexes, and after the absorptions of the dyes. Next, photocatalytic degradation of methylene blue. When dark, no considerable work has been done by the catalyst. In contrast, under UV light at different mass dosage, different irradiation time, and different pH, the photocatalyst degradation of methylene blue was quite better. This shown in the figure. See, the figure shows in the different absorptions when compared with the before in the dark reaction. So after in the presence of UV light, it will absorb in the area, and it shows better in the absorption studies, absorptions of dyes. And these are the percentage degradation of catalytic dosage, irradiation time, and the pH range. And the figure showing the photocatalytic mechanism of in dye degradation of methylene blue. After the 4 cycles of dye degradation, the same image of catalyst shows plain structure, plain morphological surface, and XRD peaks are aggregated. EDOX shows the predominant copper ion. Figure G shows the percentage degradation of catalyst up to 4 cycles, and the catalyst 2 showed better degradation than catalyst 1 from the figure. And these are the comparisons of various catalysts on degradation. The present work shows better catalyst than the reported catalyst. And these are the anti-inflammatory activities and anti-diabetic activities of the compounds, which shows the compound 1, the catalyst 1 shows better anti-inflammatory agent, and compound B, that is another one catalyst, which shows better anti-diabetic agent. And conclusion, the complexes showed better photocatalytic degradation result than the reported compounds. The degradation of methylene blue and different irradiation types of catalysts exhibited better results at 90 minutes with 87.62% for catalyst 1 and 69% catalyst 2. The catalyst at different dosage showed better degradation of methylene blue, and 20 mg of catalyst 2 exhibited 92% and 15 mg of catalyst 2 exhibited 86%. The catalyst in basic medium exhibit better photocatalyst degradation of methylene blue at pH range 12, about 95% for catalyst 1 and about 96% for catalyst 2. So most of the ship-based metal complexes are non-biodegradable. The prepared biopolymer ship-based metal complexes are biodegradable and give more advantage to the environment, such as removal of organic pollutants, treatment for dieffluent, etc. From this, we come to realize the proverb that diamond cut diamond. And these are the references I followed for the work. And I thank the management of BMC and organizers of this conference, virtual conference, and head of the Department of Chemistry, BMC. And I thank the reviewer, Dr. Uday Kala ma'am. And thank you one and all.
Thank you, sir. Now on to Rekla ma'am.
Sir, it's an excellent presentation. And you just mentioned McHugh and Shetty method of analysis.
Yes, ma'am.
And what is about that McHugh and Shetty method?
There is anti-diabetic method, ma'am. Amylose inhibitory method, ma'am. Amylose, it is an yes, amylose inhibitory method.
Okay. Thank you, sir. Thank you.
Okay. Thank you.
Thank you, Mr. Manimohan, sir. Ma'am, may I call upon the next participant?
Yes.
Yeah. I just request OP number 29, Ms. Jeevita, to start with the presentation.
Good afternoon, everyone. I'm Jeevita, studying MSc Chemistry, LNG College. This presentation is a one part of my PG project work under the guidelines of Professor Satish Duriraj, Chemistry Department, LNG College. My presentation is about synthesis and characterization of mixed ligand metal to complexes of E2,2-hydroxyl 3-methoxyl benzalidene, methyl hydrazine carbothiamide, and 8-hydroxyquinoline. We will discuss about the following titles: background of the work, aim and objective of the work, experimental results and discussion and conclusion. Its background of the work is about thiosemicarbosones and the metal complexes have been showed broad spectrum of applications in a heterocyclic compound synthesis, catalyst, electrochemistry, the biological and pharmacological activities such as antiparasitic, anti-malarial, anti-inflammatory, antibacterial, antifungal, and etc. Aim and objective of the work is synthesis of SCIF-based ligand, proton NMR characterization of SCIF-based ligand, synthesis of 5 new mixed ligand-based metal to complexes. We are still in progress in FTIR study of ligands, molecular docking, and biological and catalytic activity. Experimental methods to synthesize SCIF-based ligand are it first 1:1 equimolar of 3-methoxysalicylaldehyde and methyl thiosemicarboxide were taken in a round bottom flask. Then 20 ml of 4:1 ratio of acetonitrile and ethanol mixture were added to this solution. In a magnetic stirrer, then the reaction mixture heated at 80 degrees Celsius for about 1 hour. Then the mixture is allowed to evaporate slowly, and the skip-based ligand obtained is washed well with ethanol and recrystallized in acetonitrile. And it is the schematic diagram of proton NMR characterization of the skip-based ligand. It shows the methane proton showed chemical shift at 11.42 ppm. Phenolic group showed broad spectrum at 3.82 ppm. And all aromatic compounds showed chemical shift in the range between 2.73 ppm. And it is the schematic diagram of C13 NMR characterization or showed. Thiocarbonyl group showed chemical shift at 177.47 ppm. Methane proton showed chemical shift at 149.46 ppm. In C13 NMR, all aromatic compounds showed chemical shift in the range of 30.74 ppm to 177.47 ppm. Next, the synthesis of metal to complexes. Its experimental methods are 1:1:1 molar ratio of metal to chlorides and skip-based ligands and 8-hydroxyquinoline were taken in a round bottom flask. Then 20 ml of 4:1 ratio of acetonitrile and ethanol is added to this mixture. It is heated at 80 degrees Celsius for about 4 hours. Then the obtained mixed ligands complexes were washed with ethanol and dried over. Then this metal to complexes yielded as 55 to 78%. Then copper to complexes gave us a high yield. And it is the formula for the skip-based ligand, synthesis of skip-based ligand from the methyl salicylaldehyde and methyl thiosemicarbazine. And also the formula for the synthesis of metal 2 complexes from skip-based ligands and 8-hydroxyquinoline are showed. And the result and discussion of this project is the skip-based ligand were synthesized have been characterized by proton and C13 NMR analysis. 5 new metal 2 complexes were prepared using this work. Metal 2 complexes were obtained a moderate to good yield. Stannous and mercury showed 5 coordination number complexes due to the previous literature, and also manganese, nickel, and copper showed 6 coordination number complexes. It is also due to the previous literature. And the conclusion is skip-based ligand obtained with a good yield of 96%. 5 metal 2 complexes were also obtained good yield. Skip-based ligand have confirmed by proton and C13 NMR spectral studies. We are still in progress of FTIR study, molecular docking, and biological studies. At last, I acknowledge and I would like to thank the organized committee members of Iscst 2020, the principal and HOD of LNG College Quaternary, my guide, faculty members, and co-researchers in my own department, SIF VIT University Belur for spectral analysis, and all the participants. Thank you. Thank you.
Thank you, Ms. Jeevita.
Thank you.
Now on to Dekla ma'am.
Yes, it's a nice attempt, and it's about synthesis. And I just have only one question. You don't like the ligands are washed with ethanol. Why not with water? Any idea?
Sorry, ma'am.
You told the ligands while preparing,
Yes,
right? So the ligands are washed with ethanol, right? Why not with water? Why it cannot be washed with water? Any idea?
Because its quantity is less.
Okay, no issues. And it's a good trial when half of your work is in progress, you're presented. So that's excellent. Thank you.
Shall I call upon the next participant, ma'am?
Yes, sure.
Yeah. May I now call upon oral presentation 30, Mr. Vasanta Kumar.
Yes, ma'am. Can you hear me?
Yeah, we can hear you. So you can start with the presentation.
Good afternoon to one and all. Vasant Kumar, MPhil College, PG and Research Department of Chemistry, Allen Government College, Autonomous, Pune. My work is part of my MPhil work now I presented. Facin synthesis and proton NMR characterization of 8-hydroxyquinoninium salt with 3,5-diaminobenzoate ferrolate and nitromonophthalate as organic ions. Outline, background of the work, aim and objective of the work, experiment and result and discussion, conclusion, acknowledgement. My background of the work, 8-hydroxyquinonium based ionic liquids or molten salts have been well known of the wide application in superconductors, semiconductor, non-linear optical device, laser technology, organic emitting diodes, and ion chelator. The future, the biological and pharmaceutical activities such as antiseptic, anti-tuberculosis, etc. Antifungal used for the control. You see the picture. Control for grey mold on vines and tomatoes. Listen the picture. The aim and objective of the work. 8-hydroxyquinonium based organic crystals. Synthesis of 3 new 8-hydroxyquinonium with 3,5-diaminobenzoate ferrolate, 5 nitromonophthalate.
Screen is not visible. It's not moving.
Ma'am?
Your screen is not visible. It's in the
So
first.
you have not opened your PowerPoint yet, sir.
1 minute, ma'am. Can you see my PowerPoint? Hello?
No, sir. It's your screen which is being displayed.
Ma'am, can you see the picture?
Yeah. We can, sir.
Okay, ma'am. To calculation by using analytical and only proton NMR studies. Further in progress in FTIR study, biological activity, catalytic activity, crystal structures are going on. Experiment and synthesis of 8-hydroxyquinoninium salts, 1:1 equimolar of 8-hydroxyquinonine and aromatic carbolic acid are taken in clean RB flask. Then 30 ml of 2:3 ratio of acetonitrile and acetone mixer were added to this solution. RB flask is closed tightly and to prevent solvent evaporation. Then the reaction mixer was stirred well for about 4 hours. Then the mixer is allowed to evaporate slowly and obtain homogeneous crystal of the given below 8-hydroxyquinonine, diaminobenzoate, 8-hydroxyquinonine ferrolate, 8-hydroxyquinonine. Then will washed and recrystallized. Ma'am, can you hear my voice? Hello?
Yes, sir. We can hear you.
Among 3 crystals, now I explained one crystal, say diaminobenzoate. You see the proton NMR characterization of 8-hydroxyquinonium diaminobenzoate. The shown the picture in a proton NMR spectrum, the aromatic OH group show the chemical shift. The aromatic OH group show the chemical shift at 3.43 the range, ma'am. ppm as broad singlet. The next aromatic protons of 8-hydroxyquinonium and diaminobenzoate showed in the range of between the 8 to 8.17 ppm, ma'am. Result and discussion: we obtained 8-hydroxyquinonium based crystal with excellent yield. The picture given below. 8-hydroxyquinonium diaminobenzoate is 96% yield, and the ferrolate is 97% yield, and 8-hydroxyquinonium nitromonophthalate anions yield 98%. The complete proton transfer is confirmed successfully by proton NMR spectrum. Because the proton shift for the COH group except 8-hydroxyquinonium, you see that the COH group, the ppm value is very high. It's a 0.2 something. Conclusion. In conclusion, simple work procedure used environmentally friendly solvent, high yield at complete proton transfer. 8-hydroxyquinonium based ion salt with organic ions are definitely eye-opener for the young green chemist who is working in green catalysis. We recommended as recyclable catalyst. Maybe have a very good biological industry. I sincerely thank to the opportunity, the organizing committee member of Iscst 2020, the principal of Allen Government College, my guide and faculty member, and my guide Dr. S. Manjunathan, and co-guide Mr. Sadish. Thank you.
Thank you, Mr. Vasant Kumar.
Yes, ma'am.
Now on to Udayakala, ma'am.
Yes, sir. It's a nice, short and sweet presentation, and you're stick on to your time.
due to time convenience, now only I created the PowerPoint, ma'am.
Very good. Okay, thank you.
Okay, ma'am. Thank you, ma'am.
Thank you, sir.
Thank you.
Ma'am, shall I call upon the next participant, ma'am?
Yes, yes.
Yeah,
yes.
we have 2 more to go, ma'am. I just call upon oral presentation 31, Ms. Roshni.
Yes, ma'am. Thank you.
Yeah, you can start with the presentation, ma'am.
Ma'am, please wait. Ma'am, can you hear me? Ma'am, can you hear me?
Yeah, I can hear you, ma'am. You can proceed with the presentation.
Presentation.
Ma'am, the document is missing. Please wait.
What, ma'am?
A document is missing, ma'am. Please wait, ma'am.
Yeah. Ma'am, can we move on to the next participant, ma'am? You can just do it next.
Yes, yes, sure.
As we are running short of time. Yeah.
Yes, yes.
Mr. Malson, are you there? Oral presenter 16.
Yes, ma'am.
Can you do your presentation, sir?
Yes, ma'am.
Yeah, yeah.
Ma'am.
Yeah, I can hear you, sir.
Is my slides visible?
Yeah, it's getting uploaded, sir. Yeah, yeah. It's visible, sir. I can proceed.
Okay.
So kindly stick on to time.
Sure, ma'am. Sure, ma'am.
Yeah.
Myself, I'm Somdong Lena from the Department of Horticulture, Arithmetic and Medicinal Plants from Mizoram University. And today, the title of the presentation will be the assessment of this phytochemical and medicinal properties of Pinnaria aspera, which is a medicinal plant using GCMS chromatography. And introduction, briefly into the introduction. Nowadays, in modern world, people, they often opted for this medicinal plants instead of this allopathic drug because of the ill effect which it can cause. And briefly into Mizoram is a biodiversity hotspots area which is in the Northeast India and is covered by the Indo-Burma hotspots of International Union for the Conservation of Nature. And then that's why we are immensely blessed with a huge richness of this flora and fauna, and among them Pinnaria aspera is one of the medicinal plants which we are using. Hello, ma'am. Hello, ma'am.
Yeah, yeah, we can hear you, sir. You can proceed.
And as reported by Lalompi et al., it was used for the curing of illness in different diseases. Methodology. In this study, we not only study the plant Bitnari aspera, but alongside with that, we compared the leaves and the stem exudates, their phytochemical. We did the comparison of both the leaves and the stem exudates of this plant. And the sample was collected from this Kebran village, which is around approximately 100 kilometers from the state capital, Aizawl. And sample preparation, we collect the fresh leaves, which are healthy, disease-free, and shade dry to avoid loss of compound due to the action of heat, and then powdered it and did a sequential extraction using 3 solvents, PET ether, petroleum ether, chloroform, and methanol. Further, we did Dota evaporator and we got the leaf extract. And in the other picture, we can see the glass plastic containing the stem exudates. And then phytochemical analysis was done using tris carbon and tris Evans and carbon, following their technique, saponins, tannins, flavonoids, cardiac glycosides, alkaloids, terpenoids, And then quantitative study of this phenolic compound and flavonoid compound was also done using McDonald et al. And Chang et al. And then estimation of this antioxidant capacity was also done following DP-page reticle scavenging activity and ABTS scavenging reticle activity, and MTT assay, which is an anti-cancer assay given by most men. It was also done. And for this, you will see in the result section, we use A549, which is a lung cancer cell lines obtained from a Caucasian male back in 1972. And then after growing the cells in well plates, treatment was given from using the extract from our plants. And after that, MTT was added, which is a tetrazolium dye. And due to the action of these mitochondrial enzymes, MTT, which is yellow in color, is converted into a formazone, which is purple. And that means only the viable cells can run, only the viable cells, only the cells which are living can transform this yellow MTT into a formazone. And using a spectro, we got the result. And then GCMS, sample was sent to the State Forensic Department for GCMS analysis using NIST database, and HPLC was also done using this quercetin, ruthenium gallic acid, hesperidin, and nagerin as a standard. And then coming to the result, saponin was present, tannin was also present, flavonoid, alkaloid, and terpenoids was also present, but sadly, however, cardiac glycosides was absent. And then determination of both this phenol and flavonoid, we go for this experiment because when we say medicinal plants, it's majority of the times it's due to the presence of this phenol and flavonoid that the plants have this medicinal properties. And then our result shows the leaf extract contain more higher phenolic and flavonoid contents, which is 1 to 10 for the gallic acid equivalent, and for flavonoid 51.65 mg per gram tissue for routine equivalent. And coming to the DP-page scavenging activity and ABTS, maximum DP-page scavenging activity was obtained from the leaves extract, that leaves methanol extract, which was 63.99% at 100 mg per ml. And in same like that, in the ABTS scavenging activity, also we can see the leaf extract shows higher activity, which was 67.12% at 100 mg per ml. And cell cytotoxicity, here we found that the stem exudates perform better. It could kill around 45% of the A549 cancer lung cancer cells at 100 mg per ml. Well, the leaf extract shows only 33.39% at 100 mg per ml. And HPLC quantification result shows the presence of more quercetin, ruthenium, gallic acid, hesperidin, and nagerin in our studies. And GCMS, as a result, we can see tetradecapretanoic acid and both benzene propanoic acid in the leaf extract, and in the exudates we can see that tetradecapretanoic acid. And coming to the conclusion, Veterinary aspera exhibit both DPPH and ABTS free scavenging reticle activity, which could be due to the presence of this high phenol and flavonoid content, and it also shows a good cytotoxicity activity against A549 cell lines, which could be used for further studies in the plant. Also shows a good amount of quercetin, ruthenium gallic acid, hesperidin, and nagerin, which could be responsible for its several activities. And GCMS also detect a compound like tetradecapretanoic and noic acid and benzene propanoic acid, which are used in some food industries and pharmaceutical industries. And these are the list of my references. And thank you.
Yeah. Thank you, sir. On to Udayakala, ma'am.
Yes, sir. It's a nice presentation. And I just have one question. You told it is, you have compared the BAM leaf extract and other compound. It is not like the whole plant or only the leaf. And one more thing you have given, like quercetin, gallic acid, all these may be the influence for this activity. And all these compounds, is it tested in this research?
Ma'am, in this study, we study both the leaves. We did a comparison study of both the stem and of both the leaves extract and the stem exudates. And this quercetin further study is not yet done. And for the future work, we also did gastroprotective activity, which shows a very good result, but we are not at the point where we can present. Right now, it's going on.
Okay, okay, sir. Thank you, thank you.
Thank you very much, sir. Next, oral presenter 31, Roshni. Are you ready? Roshni, ma'am? Are you there? Ma'am, give me a minute. I'll just check on whether she's there in the session. Roshni, ma'am?
Yes, ma'am.
Yeah, can you just start your presentation, ma'am?
Ma'am.
Yeah. Kindly stick on to time, ma'am. 5 minutes.
Yes, ma'am. Ma'am, 1 second, ma'am. Ma'am, hello?
Yeah, Roshni, ma'am?
Yes, ma'am. Sorry for the misunderstanding, ma'am.
Would you like to start with your presentation, ma'am?
Yes, ma'am. Yes, ma'am. Please.
Yeah,
speak,
yeah,
ma'am.
yeah. Make it fast.
Ma'am, can you see it?
Yeah, yeah, yeah. We can do, ma'am. Can proceed.
Yes, ma'am. Hello. Good afternoon, ma'am. This is Roshni from second MSc Chemistry, Ellen Government College, Poneri. My topic is about synthesis and 1H NMR characterization of 8-hydroxyquinolinium 3,5-dinitrobenzoate. I just skipped the outline and background of the work. 8-hydroxyquinolinium based ionic liquids and molten salts have been known of their wide applications that are superconductors, semiconductors, nonlinear optical devices, laser technology, organic light emitting diodes, biological and pharmacological activities such as anti-tuberculosis, anti-malarial, anti-inflammatory, anti-cancer, anti-HIV, antibiotic, antibacterial, antifungal, anti-herbicidial agents, etc. Aim and objectives of the work. 8-hydroxyquinolinium 3,5-dinitrobenzoate is to characterization by the analytical and 1H NMR studies. The FTIR, physiochemical properties, biological activity, catalytical activity, and crystal studies are in progress. Now, experimental synthesis of 8-hydroxyquinolinium 3,5-dinitrobenzoate 1:1 equimolar or 8-hydroxyquinolinium and 3,5-dinitrobenzoate were taken in a clean RB flask. Then 30 ml of 2:3 ratio of acetonitrile and acetone were mixed and added to the solution. RB flask is closed tightly to prevent the solvent evaporation. Then the reaction mixture was stirred well about 4 hours. Then the mixture is allowed to evaporate slowly and obtain homogeneous crystals of 8-hydroxyquinolinium 3,5-dinitrobenzoate and it's washed well and recrystallized. Here the experimental of 1H NMR characterization. In a proton NMR spectrum for NS proton showed chemical shift at 9.81 ppm as a singlet. The phenolic of OH showed 3.65 ppm as broad singlet. and the aromatic protons of 8-hydroxyquinoline ring, the range of load showed as 7.83 to 9.01, and the 3,5 dinitrobenzoate ring showed as 8.31 to 8.83. And resultant discussions, we obtained pale yellow crystals and a 98% of yields. They completely proton transfer is confirmed by the 1H NMR spectrum because the proton shift of the COH2 of DNB is not showed. In conclusion, we are having a simple workup procedure, used environmentally friendly solvents, highly yielded, completely proton transfer, 8-hydroxyquinolinium, 3,5-dinitrobenzoate is definitely an eye-opener for the young green chemist who is working in the green catalyst. We recommended as a recyclable catalyst, maybe have a good biological industry. And I sincerely thank to the everyone, faculty and everyone. Thank you, ma'am.
Thank you, Ms. Roshni. Now on to Rekla, ma'am.
Yeah, it's a good attempt, Ms. Roshni, and
you, ma'am.
best wishes for your future studies. Thank you.
Thank you, ma'am.
Thank you, Roshni, ma'am. With this, we come to the end of our oral presentation. We would like to thank Dr. Udekla, ma'am, for being the chairperson of this session. We are really fortunate to have your presence here, ma'am. We are sure that all your questions and suggestions will make our presenters to improvise their works. Thank you, ma'am. Thank you so much. Thank you for joining us. Now we'll just move on to session 7. I may now request Dr. R.J. Kavita, Assistant Professor, Department of Chemistry, Bhaktavashla Memorial College, to introduce the guest speaker of this session, Dr. Kirti Murugan. Kavita, ma'am.
Good afternoon to one and all present here. I heartily welcome you all and our speaker. It gives me immense pleasure to introduce our speaker, Dr. Kirti Murugan, Postdoctoral Research Fellow. Dr. Kirti Murugan has completed her master's from St. Joseph College, Bharathidasan University. She completed her MPhil in Analytical Chemistry from University of Madras. She has received her doctoral degree in Electrochemistry from National Taipei University of Technology, Taiwan. Her field of interest includes nanomaterial synthesis, electrochemical sensors, supercapacitor, electroplating and electrodeposition, dye-synthesized solar cell, photocatalyst, nanomaterial synthesis for anti-cancer therapy. She has many publications in reputed journals for her credit. Present now, she is a postdoctoral researcher in the Department of Chemical Engineering and Biotechnology, National Taipei University of Technology, Taiwan. I now hand over the session to our speaker, Dr. Kirti. Ma'am, please.
Hello, can you hear me?
Yeah, we can hear you, ma'am. We can hear you, ma'am. To start with the session.
Okay, thank you for the introduction, ma'am. Very good afternoon to all of the participants, faculty, and the organizer in international conference. Especially, first I would like to thank BMC Chennai, especially Sankita ma'am, for giving me this opportunity to share my research experience with you. In this talk, I would like to share few things about electrochemical sensor and biosensor. This is the overview of this talk. First, we will see the electrochemical sensor, why we need electrochemically modified electrodes, and electrochemical methods and its advantages, history of electrochemistry, cyclic voltammetry, what is cyclic voltammetry, and what are the components present in the cyclic voltammetry and its working principle, and the next biosensor. Then basic principle of biosensor, and then what are the components present in the biosensor, and basic characteristics of biosensor, and what are the applications of biosensor, and then glucose biosensor, finally immunosensor. Electrochemical sensor. Electrochemical sensor is a device which is used to analyze biomolecules, drugs, dyes, heavy metal ions, and pesticides. Nowadays, so many electrochemical sensors have been developed, coming from research laboratory to commercial market. These are the components of electrochemical sensor. First, there are two components present in the electrochemical sensor. First one is chemical recognizing system, and the second one is physiochemical transducer. This chemical recognizing system is the important main part of the electrochemical sensor. Physiochemical transducer. This physiochemical transducer transforms electrochemical information of the molecule into the useful signal by either oxidation or reduction. This is voltammetry. The electrochemical sensor is working based on voltammetry techniques. Voltammetric technique is measurement of current arising from oxidation or reduction at the electrode surface with the function of applied potential. There are several voltammetric techniques are available like cyclic voltammetry, differential pulse voltammetry, amperometry, and linear sweep voltammetry. These voltammetry techniques mainly working based on this voltammetry techniques classified depends on electrode type or material used as working electrode and how the potential is varied. For example, cyclic voltammetry, we can vary the potential with respect to time. But in the amperometry techniques, we put the constant potential, but only the time is increased. And then how the current is measured. These are there's several voltammetric techniques are classified based on this characteristic. And then these voltammetric techniques high sensitivity and fast detection. This here the electrode fabrication is very simple, which low cost and portable, which easy to use, which will consume less time. This is history of all voltammetrics. The polarography is a type of voltammetry. This is the first invented. J. Haruska is first invented the polarography technique. Here, the dropping mercury electrode is used as working electrode. For this polarography, he won Nobel Prize. Dropping mercury electrode introduced in 1920, but he won the Nobel Prize in 1959. And then electrochemical cell. Here I put the electrochemical cell. These are the components of electrochemical cell: working electrode, reference electrode, and counter electrode. Here the working electrode, we can use Pt, gold, silver, and carbon. The main purpose of working electrode is in working electrode, the reaction interest is happen in the working electrode, and a reference electrode. It provides stable and reproducible potential. AgCl is frequently used working electrode and calomel electrode, and then counter electrode. Usually Pt wire and graphite rod is used as counter electrode. These are the main components of electrochemical sensor. The electrochemical sense, this is 3 electrode system: working electrode, counter electrode, and a reference electrode. The working electrode should be inert. First, the main aspect is it should be inert. Usually, the classic carbon electrode is used as working electrode because which is highly reactive and which will not react with any other material, and we can put wide linear potential. So glassy carbon electrode is frequently used other than gold and silver electrode. And then reference electrode is AgCl electrode, and then counter electrode. The main purpose of counter electrode is just for conductivity. The current is measured between the working electrode and the counter electrode. The potential is applied. Here the potential is applied between the working electrode and the reference electrode. The reference electrode should be maintained the constant potential, will not vary, so that we can accurately detect the working potential in the working electrode. Here we can use the supporting electrolyte. The main the supporting electrolyte, it's for conductivity. It should be soluble in any solvent. And then the chemically modified electrode. The chemically modified electrode is main objective in electrochemical sensor. The chemically modified electrode is result from immobilization of the electrode surface through composite formation, electrode deposition, or chemical reaction or chemisorption. These chemically modified electrodes have high reactivity than conventional unmodified electrode. Recently, chemical modified, there are so many chemical modifiers used like metal oxide, metal sulfide, polymer, carbon, graphene, zeolite, clay, and MOF. We will see how to fabricate the chemical modified electrode. First, before we use glassy carbon electrode, we first clean the surface, and then we just drop casted on the modifier and dried. We can use for electrochemical experiment. And then cyclic voltammogram. This is cyclic voltammogram for ferroferricyanate solution. Here the x-axis represents the applied potential, the y-axis is the current response. And here the arrow indicates the steep direction from point when the potential is moved negatively. The Fe plus is reduced to Fe, and when point is reached C, IPC is observed. The IPC is cathodic peak current. When further increasing the potential, the Fe plus reduced to Fe is decreased due to the diffusion layer. The concentration of Fe reduced Fe on the surface of the electrode we called diffusion layer. Due to the diffusion layer, the cathodic peak current is decreased. When reach the switching potential D, the scan direction is reversed towards the positive side. When here the Fe reduce the Fe start to reoxidize into Fe plus. When the point is reached F, then this point is called anodic peak current. This after when increasing the potential towards the positive side, the anodic peak current is decreased. At the equilibrium, point P and E is called equilibrium constant. We will explain with Nernst equation. The Nernst equation is relates the potential in the electrochemical cell with standard potential of the species. This is Nernst equation is E equal to E naught plus RT NF ln oxidized species divided by reduced species. Here R is the rate constant, T is temperature, N is number of electron transfer, F is Faraday constant. This is one of my research work. We have published in ACS Applied Material and Interfaces. One part, a sustainable synthesis of cerium sulfate, gum arabic carbon flower nanocomposite for the detection of insecticide imidacloprid. Here we used cerium sulfate gum arabic carbon as a modifier for the detection of imidacloprid. Here, this is subjective of this work. First, we synthesize the cerium sulfate gum arabic carbon flower, and this prepared composite is confirmed from the characterization techniques, and then we can done the electrochemical detection of imidacloprid by cyclic photometry, and then we have done real sample analysis. Introduction: imidacloprid is insecticide, belongs to neonicotinoid family. The intensive application of imidacloprid in the agriculture and irregular storage is a major source of contamination in the environment. So it's necessary to detect imidacloprid in food samples. Here cerium sulfate is used as the electrode modifier. There are various cerium sulfate is present, alpha, beta, beta and gamma. This cerium sulfate has high electrical conductivity, high thermal conductivity, and has high thermostock resistance. Here, in order to further increase the electrocatalytic activity, here we can choose gum arabic as a carbon source. This gum arabic is natural abundant biomolecules which is commercially available, which is environment friendliness. And then synthesis. First we synthesize the cerium sulfate gum arabic carbon. Here gum arabic is gum arabic and L16 is taken in the beaker, and we just stirred and put it into hydrothermal. We can get the composite cerium sulfate gum arabic carbon composite, and then we just drop custard on the electrode surface, and then we have introduced into the electrochemical detection. We have confirmed we have take the morphological studies. We get the flower like cerium sulfate gum arabic carbon nanocomposite, and then confirmed from the HRTM images we can get the same morphological characteristics, and then the structural characterization confirmed from the XRD, BET, and the Raman spectroscopy, and we have confirmed from the XPS analysis. And then electrocatalytic activity of modified electrode is investigated. First, we have done the EAS analysis with various modified electrode. Here, the cerium sulfate gum arabic carbon servers get smaller RCT value, which indicate the higher electron transfer ability of the cerium sulfate gum arabic carbons. And then cyclic photometry study. This is we have done the experiment with ferropyriscinate solution for various modified electrodes. For comparison, we used only cerium sulfate and bare JCE. Here we get higher peak current, redox peak current for ferropyriscinate and smaller peak to peak separation. This indicates the fast electron transfer and the higher electrocatalytic activity of the cerium sulfate gum arabic carbon. And then this is scan rate study. In order to investigate the electroactive surface area, we can do the scan rate study by varying the scan rate 10 to 100, and using this slope value, we can calculate electroactive surface area by using the Randall-Weak equation. This we have given the Randall-Weak equation. Here, A is electroactive surface area, N number of electrons, D is diffusion coefficient, R is scan rate, and C bulk concentration of the ferricyanate solution. This is electrochemical detection of imidacloprid. This immunoglobulin in the nitro group converted into hydroxyl group with 4 electrons and 4 proton transfer. Here, the electrocatalytic ability investigated with various modified electrodes. Here, the cerium sulfate composite exhibit higher peak current, and also we have done the experiment with different varying the concentration 50 to 50 micromolar, and then electrochemical and then scan rate study. The scan rate study is used for the electrochemical mechanism, whether it's a surface controlled reaction or a diffusion controlled reaction. When the plot, which is the surface controlled reaction, is a phenomenon occurring due to the migration of molecule or particle from the bulk aqueous phase to interface. The diffusion controlled process is molecule are transported from bulk phase to subsurface just below the surface. The molecule then move from substrate to interface. This is diffusion controlled process. We can calculate from the scan rate study. Here we have done the experiment with 10 to by varying the scan rate 10 to 100 microvolt, and from this here the obtained peak current is plotted against the square root of scan rate. From this result we can we know that that electrochemical reaction is completely diffusion controlled process. And then pH study. We have done the experiment, CV experiment with varying the pH range from 3 to 11, and here the pH 7 exhibit higher peak current for immunoglobulin, and the potential is moves when increasing the pH 3 to 11. The potential is moved towards the negative direction, which indicates the equal number of protons and electron transfer. Here, the peak current is the obtained peak current is plotted against the various pH and various potential. Here we get the slope value, which near this. Here we get a value 51 microvolt, which is near to the theoretical value of 59. That is Nernst equation. And then this is the parameter of electrochemical sensor: L-body, linear range, and sensitivity. For this, in order to investigate the electrochemical parameter, we can done the experiment with DPV analysis by varying the concentration of immunoglobulin 0.05 to 1000 to 66 micromolar. And we get a wide linear range. We get 2 linear ranges, smaller linear range, and this is for higher concentration. And then interference study. In order to investigate the selectivity, we done the experiment with various interfering compounds, and then we compared with other analytical we compared the analytical parameter with other sensor we already published. Here we get the lower LOD 32 nanometer and wide linear range, and then real sample. In order to investigate the practical applicability, we have done the real sample analysis with fruit and vegetable samples. We get appreciable recovery and RST values. And then biosensor. The biosensor is differ from electrochemical sensor. Here the biosensor is an analytical device which is used to determine the presence and concentration of the specific substance in the molecule. Here we use biological molecule as a bioreceptor. This is the main components of biosensor. First biosample, that is analyte, and bioreceptor, and then transducer, signal processing. This is display. This is main components present in the biosensor. The professor Leland C. Clark is a father of biosensor. He is first invented glucose biosensor. The device incorporating a biological sensing element, that is antibody, DNA, enzyme, and cells, which is intimately connected to or integrated with the transducer. This is transducer. Transducer is a working electrode, recognizing based on affinity between complementary structure like enzyme substrate and antibody to antigen and receptor to hormone complex. This selectivity and the specificity depends on biological recognizing system connected to suitable transducer. And then basic principle of biosensor. The basic principle of biosensor involved three elements. First, the biological sensing element. First biological recognizing element, DNA, antibody, enzyme, cells, which highly specific towards the biological material. And then second, transducer. The transducer detects and transduce signal from biological target receptor molecule to electrical signal, and then signal processor. Third, after transaction signal from biological to electrical signal, where the amplification is necessary and it takes place. And then the biological material is immobilized and contact is made between the immobilized biological material and the transducer. The analyte binds to the biological material to form bound analyte, which in turn produce the electronic response that can be measured. Sometimes the analyte is converted to product. This product may be heat, gas, electrons, and hydrogen ions. The transducer then convert the product into electrical signal. These main components of biosensor, and these are the components of biosensor. First, analyte, which may be in medical sample, food sample, chemical pollutant, and then bioreceptor. This is bind with analyte, nucleic acid, antibody, virus, enzymes. This is bioreceptor is important part of the in biosensor, and then transducer. Finally, data processing. We will see one by one. First component, biological element. The component used to bind the target molecule must be highly specific, stable, and under storage conditions and immobilized. Which function is to interact specifically with the target compound. The compound to be detected, it must be capable of detecting the presence of target compound in the rest solution. The ability of bioelement to interact specifically with the target compound is the basics of biosensor. The second component, this is the second component, a transducer, which has acts as an interface, measuring the physical change that occur with the reaction at the bioreceptor, then transforming the energy into measurable electrical output. This is third compound. The signal from the transducer are passed to a microprocessor, where they are amplified and detected. The data is then converted to concentration unit. These are the basic characteristics: linearity, sensitivity, selectivity, and response time. Linearity of the sensor should be high for the detection of high substrate concentration. Sensitivity, here the value of electrode response by substrate concentration. Selectivity, the chemical interference must be minimized for obtaining the current result. And then finally, response time, the time necessary for having 95% of the response. Application of the application of biosensor. The biosensor is applied in medical, agriculture, environmental, and food. First, glucose monitoring in diabetic patients and environmental application, that is direction of pesticides and the river water contaminants such as heavy metal ions, and then detection and determination of organophosphates, and routine analytical measurement of folic acid, biotin, vitamin B12, and then determination of drug residue in food such as antibody and growth promoters, particularly meat and honey. Glucose biosensor. A glucose biosensor is one of the known examples of electrochemical sensor which is commercially available. Blood glucose monitoring is a way of checking the concentration of glucose in the blood using the glucometer. Why we need to monitor glucose? It reduces risk of developing complication with diabetes, allow the diabetics to see if the insulin and other medication they are taking are working or not. Gives diabetics an idea as to how exercise and food affect the blood sugar, may prevent from hypoglycemia or hyperglycemia. Low blood sugar is called hypoglycemia and high blood sugar is called hyperglycemia. Here we can see the working principle of a glucose biosensor. First, glucose is converted into gluconic acid. Here, glucose oxidase is used as an enzyme for the oxidation of glucose by molecular oxygen produces gluconic acid and hydrogen peroxide. In order to work as a catalyst, glucose oxidase requires redox cofactor, that is FAD, flavin adenine dinucleotide. And it works as an initial electron acceptor. It reduces to FADH2.
ஆமா நீங்க தான் உடைச்சீங்க இல்லனா அத வேற நீங்க உடைக்காத மாதிரி.
And then electrochemical sensing of glucose. The cofactor is generated by reacting with oxygen, leads to formation of hydrogen peroxide. Here the hydrogen peroxide is oxidized at a platinum electrode. The number of electrons at the electrode surface is directly proportional to the glucose molecule in the blood. There are three strategies used for the electrochemical sensing of glucose. First one is by measuring the oxygen consumption. Second one is by measuring the amount of hydrogen peroxide produced by enzyme reaction, and then using diffusible or immobilized mediator to transfer the electron from the glucose oxidase to the electrode. The types of glucose biosensor. There are main two types of glucose biosensor is available. First one is enzymatic glucose biosensor, and the second one is non-enzymatic glucose biosensor. The enzymatic glucose biosensor classified into first generation glucose biosensor, second generation glucose biosensor, and third generation. The anthropometric enzyme glucose biosensor are divided into three parts. It depends on electron transfer method used for the measurement of biochemical reaction. In all cases, the presence of enzyme is required, and therefore sensor performance relies on different parameters such as working pH and temperature. Here we have the first generation glucose biosensor. The first generation glucose biosensor estimated glucose concentration in the sample based on hydrogen peroxide produced by glucose oxidase, which utilizing dissolved oxygen. Based on this technology, Yellow Spring Instrument Company launched the first commercial glucose biosensor in market in 1975 for the measurement of glucose. Here, the most expensive metal, platinum, is used as an electrode, which restricted the biosensor to clinical laboratory only. Here, we have drawbacks of first generation glucose biosensor. The amperometric measurement of hydrogen peroxide required high operating potential of 0.6 volt for high selectivity. Second, the restricted solubility of oxygen in biological fluids, which produce the fluctuation in the oxygen tension. Third, deactivation of the enzyme due to the production of hydrogen peroxide. Then we will go for second generation glucose biosensor. The second generation glucose biosensor utilizes redox mediator to transfer electrons from enzyme to the working electrode surface. Here, the redox mediators such as ferrocene, ferricyanide, quinine, methylene blue, etc. we are used as used for the improve the sensor performance. Here, the lower redox potential of chosen mediator 0 to 2 volt result no interference from the other electroactive species such as uric acid and ascorbic acid. The redox mediator enhances the electron transfer between the redox center of enzyme and the electrode surface. These are the drawbacks of second generation glucose biosensor. First, high consumption between redox mediator and oxygen. Second one is interference of other electroactive species lead to fall and it give inaccurate results. Third one, small size and highly diffusive nature of mediator possesses problem of leaching the mediator from intermediate region between the enzyme and the electrode surface. Then we will go for third generation glucose biosensor. The third generation glucose biosensor are based on direct transfer between the active center of enzyme and the electrode. The intrinsic barrier to electron flow is the globular structure of glucose oxidase with the active sites, which is containing FAD and FADH2 redox cofactor. The carbon nanotubes immobilized electrode surface provides suitable orientation for enzyme immobilization and establish connection between electrode surface and deeply. Here, next one is non-enzymatic glucose sensor. Nowadays, we are using non-enzymatic glucose sensor, but which is not commercialized. The use of non-enzymatic electrode as a glucose sensor potentially promises a fourth generation to analytical glucose oxidation. The active metal nanoparticle undergo the oxidation step that forms a hydroxide layer that mediate oxidation of absorbed species. And then electrochemical immunosensor. Electrochemical immunosensors are affinity-based analytical devices. This sensor is based on immunochemical reaction compressing of antigen or antibody as the biorecognizing element. Here we have the second two antibody. First one is capture antibody. The second one is the detection antibody. This antibody is connected with nanoparticles. This is act as the chemical recognizing element. And then immunosensors can play an important role for detection of cancerous molecules, proteins, food pathogen, bacteria, and viral due to their high specificity and label-free detection. Basic analytical principle of electrochemical immunosensor. This sandwich type immunoassay, which is a common format of immunosensor, which based on separation of unbound material which do not participate in the binding event. The signal usually comes from catalytic reaction of enzyme molecule labeled as the signal tracer with the detection antibody. Improving the analytical performance of electrochemical immunosensor. Choosing the appropriate electrode along with the proper surface modification is very important for the analytical sensitivity. Such nanomaterial used as electrode must meet the following requirement for enhancing the electrical signal. First, a sufficient electrocatalytic trait. Second one is adequate electron transfer ability. Third one is excellent biocompatibility with biological molecules such as capture antibody. And then signal enhancement via labeling techniques. Here, nano carrier and electroactive nano tracer and enzyme-based approach is used as a label to enhance the electrochemical signal. The label techniques can be categorized as nano carrier, electroactive nano tracer, enzyme-based nanopolymer, and redox cycling of enzyme reactions, which enable an electrochemical signal enhancement. Thank you.
Thank you very much, Kirti ma'am,
Thank you.
for your very elaborated and informative session on variety of sensors.
Thank you,
We have few questions, ma'am.
Okay.
Can you just read it out?
Okay, ma'am.
Yeah. Ma'am, one participant has asked, which nanomaterial is found to give higher sensitivity?
Which nanomaterial?
Maybe I think they
it's,
might have talked about graphene or something like that. Graphene
we
or...
are the graphene and the other carbon materials is just used for conductivity. It enhances activity, but it will not specific to any molecules. It's just for conductivity. We just other metal oxides, metal sulfides, and other nanomaterial are other functionalization, like like other functionalization only they give the specificity in electrochemical sensor.
Okay. Another question, ma'am. How far is the sensitivity of these electrode when you go in for any real-time analysis?
But usually these modified electrode we just only laboratory level only. We cannot introduce into commercialize. But if the sensitivity high, but the selectivity is poor for all the electrochemical. Only glucose biosensor only we can use the commercialized one.
Oh, is there any other sensor used as a commercialized one? No?
No.
Okay. Okay, ma'am. The next question. What is the need of purging nitrogen in the electrolytic solution?
Usually the nitrogen is inert gas, but oxygen is not inert. It will reduce in the potential window. So in order to interference of oxygen, we need to purge nitrogen to eliminate the oxygen from the solution. Usually the oxygen have the characteristics of dissolve in any solution. It will interfere with other which analyte we want to detect. It will interfere with that analyte. So we in order to remove the oxygen, we need
to purge nitrogen.
Yes.
Okay. Next question, ma'am. How far is the stability of material what we use on the electrode surface?
Stability. What? Can you?
How far is the stability of the material used on electrode?
Stability of the material.
Maybe that mediator what you're using.
But usually here usually the enzyme is used as a mediator, but in the enzyme has less stability only. That is the main drawback of all the biosensor. It will degrade or like that in atmospheric conditions. It will, but a nanomaterial compared with other enzyme like that, and the nanomaterial is good stability, gives good stability, but the specificity is low compared with the enzymes.
Then how long will be the life period of this kind of a sensor?
Maybe we can use 1 month.
Okay. That too under a well storage condition.
Yes, ma'am.
Okay. Thank you very much.
Thank you.
Thank you very much, Kirti ma'am.
Thank you.
Now we'll move on to our last session.
Okay.
Thank you.
Thank you, ma'am.
Thank you. Dr. Chandrakumar sir, shall we start with the session?
Yeah, just.
May I now request Dr. R.J. Kavita, Assistant Professor, Department of Chemistry, Bhaktivinoda Memorial College, to welcome the guest speaker of this session. Kavita ma'am.
Yes. Good afternoon to one and all present here. It's a great honor for me to introduce and welcome Speaker Dr. Chandrakumar, Scientific Officer for this session. Dr. Chandrakumar, after completing his PhD from National Chemical Laboratory, Pune, he has been working as a scientific officer since 2004 at Baba Atomic Research Center, Mumbai. His research interests are in the areas of growth mechanism of carbon nanotubes and fullerenes, self-assembly process and development of hydrogen storage materials. Recently, he has made an attempt to design gold-based nanocatalysis for the activation of CO bonds and decomposition of environmental pollutants, especially carbon monoxide and carbon dioxide. His noteworthy contributions include the conceptual density, functional theory-based chemical reactivity descriptors, development of materials for energy and environment, and structure reactivity relationship in carbon-based nanostructured materials. He is also interested in science popularization and basic education program. He is a recipient of the Young Scientist Award from the Indian Science Congress Association in the year 2002, Indian National Science Academy in the year 2005, and the Department of Atomic Energy in the 2007. He has also recently been awarded the Scientific and Technical Excellence Award in the year 2015 from the Department of Atomic Energy. On behalf of all gathered here, I once again welcome you, sir. I now hand over the session to our speaker, Dr. Chandrakumar, sir. Sir, please.
Yeah. Thank you, ma'am. I would like to first of all thank Professor Sangeeta and Kavita for their nice introduction and inviting me to give a talk here for this conference. The title of my talk, as you can see in the presentation file, this is based on the computational chemistry, and I will be dealing with the nanomaterials for energy and environmental applications. So before going into the details of the talk, let me first introduce what is meant by theoretical chemistry or computational chemistry. So the theoretical chemistry deals with the mathematical description of systems of chemical spaces. And we simulate the chemical structures and reaction based on the fundamental laws of physics, especially the quantum mechanics. So based on the quantum mechanics, the chemical structures and reactions are simulated and we mathematically define. And in order to study this chemical phenomena, we use the computers heavily rather than doing the experiments. And what is the role of theoretical chemistry? Let me just explain in a few slides. It says that somewhat the role of theoretical chemistry is indispensable nowadays. The reason is that it almost provides all the information related to physics, chemistry, biology, and material science. For example, some of the important estimates like heat of formation, heat of reaction, entropies, heat capacities, like thermodynamical parameters or chemical kinetical parameters or electrochemical parameters, most of the things, the valuable information one can obtain from this theoretical chemistry. And these informations can provide somewhat valuable insight into the properties of new material, and hence one can design some new materials of the product at the macro level. Having said that, the advantage of theoretical chemistry, there are some chemical complexities are there. The chemistry is not a very simple subject. It's a very complicated thing. Some of the chemical reaction may happen in a very slow fashion, like rusting of iron. But some of the chemical reaction can occur lightning speed. In a fraction of the millisecond, in a femtosecond rather, electron can jump from one atom to the another atom. So how do we study such kind of chemical reactions? And what is the mechanism that we can provide? And in addition, there are many other problems are associated with that. First of all, in a chemical reaction, we use a lot of solvents, additives, catalysts, co-catalyst, and so on. So what is the role of each component in a particular reaction? To solve one by one, it turns out to be that it is a n parameter dependent. It's not a single parameter like reaction. Reaction is going on, A and B, both the reactants are contributing. No, the answer is that it depends on many other parameters that particularly defines the product yield and the type of product that we get it, and so on. So the chemical reactions are not very simple. Although the chemical reactions are not very simple in the sense that it is very difficult to investigate, the applications of computational chemistry has expanded. That means that it has a valuable, good framework to study such kind of chemical reactions, starting from atmospheric chemistry or drug design, catalyst, biocatalyst design, material design, and so on. Starting from, we can even think about how to model the surfactants or lubricants or polymer structures, or even the biomolecular designs also. Now you can see that the Nobel Prize has been given in the area of quantum chemistry in the year 1998, Professor Walter Kohn and John Poppel, and for their development of density functional theory and development of computational methods in quantum chemistry. And you can see the valuable inputs that has been developed since the birth of quantum mechanics. And recently, in the year 2013, one more Nobel Prize was given in this area. That is Professor Martin Karplus, Mitchell Levitt, and Professor Warshall. And they got this Nobel Prize for designing new drugs or materials. They mentioned that computational chemistry allows the very accurate predictions how small molecules will approach and bind to proteins and surfaces. And the real contribution made by these three professors are still used in the area of drug design and biomolecular modeling and chemical reactions. The computational challenge, what we have at present, is in terms of number of atoms. We have 1,000 to 10,000 atoms in protein. For example, if you want to study a small portion of your protein, let us say, and it involves millions of atoms, and we can look at the structure at the microscopic level and find out what is the active site for that particular protein, and one can model the quantum mechanical model as the active site, and remaining portion we can have it as the molecular mechanics. I will give you the details. What about this QM and MM models? So we have now come to the stage that we can handle even such big systems involving 1,000 to 10,000 atoms in a protein. And we can do the molecular dynamic simulations to study the chemical reactions or proton transfer or treatment of excited state and so on. And the recent one, the structure and function of bioactive molecules for coronavirus or drug design, even that can also be studied, and the studies are ongoing now. The success and achievements of computational chemistry expands. Once again, I want to emphasize the fact that it has given a good, valuable insights into the modeling of enzyme catalysts and protein accents, the dynamics of photobiological process, how the vision is happening. So we know the accurate mechanism for that, and the chemical reactions in solution, of course. And in case of macromolecules, what is the role of electrostatic energies and protein folding? And we have also developed a lot of force fields for studying such kind of photobiological process or enzyme catalysts and protein accents. And the last two important things are industrial catalysts and drug discovery. And the last two are heavily dependent on this industrial catalyst in the industrial catalysis. The reason is that for a particular reaction, there might be n number of possible catalysts are available, but which one would be suitable, that we do not know, unless we make an attempt to study it is very difficult to tell that what kind of system would be acting like a catalyst for a particular reactions. So this computational chemistry provides a platform to investigate what are the things can act like a good catalyst for a particular reaction. Now, let me come to the point, very important. What is the basics of computational chemistry? And that is nothing but quantum mechanics. And the entire foundation of computational chemistry relies on the quantum mechanics. So in general, we define the Hamiltonian for n electron systems. That is the H cap is the Hamiltonian. And the first term defines the kinetic energy of the electrons and kinetic energy of the nuclei, electrostatic interaction between the electron and nuclei. Basically, the total energy depends on the kinetic energy and potential energy. And you define each kinetic terms and potential energy terms and sum it up, and that will give you the Hamiltonian operator, and you solve the equation. And we apply here Born-Oppenheimer approximation that separates the nuclear Hamiltonian and electronic Hamiltonian, and we get the electronic Hamiltonian as only the kinetic energy of the electrons and electron nuclear potential, and the third term, that is the electron-electron repulsion term, and nuclear repulsion energy is also included. So the basic question is that we now know that what is the equation that we have and how do we solve this equation. And lot many theories have been proposed. Once you get the Hamiltonian and the how to solve this equation, to answer this question, it has taken almost more than 30 years. In 1927, the Schrödinger equation was proposed, and 1930, we know that to some extent how we can deal with the system. But the detailed calculations and chemistry in chemistry started 1960 and 70 onwards. And one of the foremost important theory what we have in theoretical chemistry is Hartree-Fock theory. The basic concept of Hartree-Fock approximation is nothing but the mean field approximation. And this mean field approximation, we use it to solve the complex many-body problems numerically. The mean field approximation is nothing but is something like this. When you have a cloud of electrons, let us say, the electron at a time sees, one electron at a time sees all other electrons. So, once the one electron sees the other electron, then its count is over, and go to the another electron and see the other interaction. So you sum it up, the electron-electron interaction in this fashion. And you get what you get is the mean field approximation. And this is the essential part of the electronic structure theory. And this approximation we use in Hartree-Fock approximation. And that leads to the two terms, very important terms, and that is nothing but the J term and K term. J term refers to the Coulomb energy, and K is the exchange energy. And this is the Hartree-Fock equation that we have. That is the one. The first one is the core energy, and the other one is the combination of J and K terms, exchange and Coulomb terms. That is the K is the exchange. And once you get this expression, then you minimize the energy that requires the Hartree-Fock equation. And what we get is the Hartree-Fock operator, essentially Fock operator, and you solve this equation, you get the Hartree-Fock energy. And in this model, only one problem is that we normally say that two electrons can be occupied in the same orbitals with opposite spins like this. That means that we have avoided two electrons with parallel spin in the same orbital. But there is a catch. Even though two electrons are occupying in the same orbitals with the opposite spins, but still they can avoid each other. Because the electrons are electrons, they have negative charges, so they should avoid each other, even though they have the opposite spins. So, and this particular problem is known as the electron correlation problem. So the instantaneous repulsion between electrons with opposite spins, that is missing in the mean field of Hartree-Fock theory. And that energy corresponding to the this particular problem is known as the electron correlation. And interestingly, this electron correlation, the energy corresponds to the electron correlation, comes within the range of chemical energy, chemical bond energy. And hence, if you miss this electron correlation part, you will instantaneously going to miss the chemical properties. So it is extremely important to go for highly electron correlated calculation. This is the reason why we go for electron correlated calculations. Now, there are several methods are available for this. We have the wave function based methods for the electron correlation. Basically, we have the variational theory based methods or perturbation theory based method, and we have the configuration interactions and Muller cluster theory based and couple cluster theory based. And the Hartree-Fock theory without the electron correlation, as I mentioned earlier, it is very simple and one can get the basic information about the system and hence the system that we are trying to investigate. But if you start including the electron correlation properties, then computationally the time increases exponentially. And this is a major problem. So if you want to have highly accurate calculations, highly accurate properties, then the time that much time we need it. So if you go for the higher, so there is a balance between the accuracy and that computational time that goes hand in hand. So there is some balance point where we can define that, okay, at this particular, for a particular problem, this method is suitable and one can go for it. For example, normally we go for Hartree-Fock or MP2. If you go for the higher theories like couple cluster theories, we cannot, we are not in the position to handle a particular system more than maybe 25 atoms or 30 atoms. That is the main problem. However, if you have the good computational power, one can handle. The next one theory is the density functional theory, which is very popular among all the experimentalists. And the basic concept behind this density functional theory is based on the two theorems. That is the first Hohenberg-Kohn theorem and second Hohenberg-Kohn theorem. And this theorem actually forms the basis of density functional theory, and which was proposed around 1960s. And this theory is this theorem says that the external potential is determined up to an additive constant by the ground state charge density. That means that the energy of a particular system is exclusively dependent on electron density. Please note that electron density now depends on only 3 coordinates. Whereas in wave function theory, the energy depends on n number of coordinates, all the n particle coordinates. And that is how it complicates the situation. And why density functional theory has become popular and very easy among the experimentalists? It is easy to solve because the energy is now dependent on electron density alone. The electron density depends on only 3 coordinates. And that is the reason why it is very successful and popular. There are some issues concerning this electronic structure calculation. For example, if you look at the table, it says that if you have n number of particles, the Hartree-Fock model, which says that n to the power 4, and DFT, which includes the electron correlation by taking suitable models, electron correlation functionals, you can have the formal scaling from n to the power 3 to n to the power 4. But if you go for the slightly higher accurate calculation, for example, MP2, we need the n to the power 5, or couple cluster n to the power 7. So this is the main problem. So yeah, the order that I have given is in the increasing order of the accuracy. So if you want to have a good accuracy, you need to have a good computational power. That's the thing. Okay, so now we have discussed about the basics of computational chemistry, and now we will discuss about the main theme of our topic, that is on the computational design of nanomaterials for energy and environmental applications. Now, what is the main issue with this? Now, we have a global problem, atmospheric pollution, ocean acidification, ozone layer depletion, and so many things are So many problems are there, and hence we need to have some suitable materials for energy and environmental applications. Now, why? That depends on the energy and environment. The reason is the depletion of fossils, fuel cells, coals, and oils. Now, why it is getting depleted? It is very simple. In this earth, if we need to have that fuel, we just go and dig up the well and get the fuel. It is just like robbing the nature. If you want to get money, go directly to your bank and just take away all the money, whatever, how much ever you wanted. That is how we are doing it, without realizing what the bad effects of this fossil fuels or coals and oils. That is the problem. We need to stop. Otherwise, we are going to have a severe problem of global warming and so on. So that defines that need of alternative fuels, and that comes under hydrogen energy and hydrogen economy. So the need of alternative fuels, what I'm going to speak, is based on the hydrogen energy. Now, why hydrogen energy is there? Why hydrogen energy has become very important? Please note that hydrogen is not the fuel, it is only the alternative energy carrier. And the energy density of the fuel I have given in this table. You can see that the fuel, coal, wood, gasoline, and diesel, and among everything, the hydrogen has the more energy content. That is a 120 kilojoules per gram. And whereas the simple natural gas or methanol, which is far away lesser than the hydrogen. So the hydrogen has the highest energy content per unit mass. That is why the hydrogen has been considered as an alternative energy carrier. Although the hydrogen has a lot of advantages, there are some major problems and issues associated with this. This needs a visionary strategy. Why? Because the first, foremost important problem associated with hydrogen economy is that how do we generate hydrogen? And second most important problem is how do we store hydrogen? So generation of hydrogen and storage of hydrogen, that forms the hydrogen economy. So if we can handle these two problems properly in a suitable way, then we can be successful for achieving the hydrogen economy in a proper way. So we need to have a proper catalyst for water splitting and suitable materials for hydrogen storage. So the hydrogen storage material, what should be the properties? And before that, I want to tell you that if you have a vehicle with fuel cell, which has 4 kg of hydrogen, that can be run 500 kilometer range. That is advantage. That is equivalent to 50 liters of petrol tank. And 50 liters of petrol tank we can store, if you have a nanomaterial, 63 kilogram of hydrogen meter cube can be stored. That is the advantage. You can see that within merely by having 4 kg of hydrogen, we can run 500 kilometers. But however, these are the theoretical predictions. The main problem is that there is no storage technology is currently capable of meeting this goal. So what are the requirements of hydrogen storage material? How we can store it in an efficient way? The point here is that the materials that we are talking about to store the hydrogen, it should have appropriate thermodynamics. For example, favorable enthalpies of hydrogen absorption and desorption. Like whenever you want to get hydrogen from the stored material, you should be able to quickly take it. That means a fast kinetics should be there. And that means the absorption should be fast, and at the same time, desorption also should also be fast. And of course, it should store more amount of hydrogen, and that is determined by the high gravimetric and volumetric densities. And the life cycle of the hydrogen storage material after operating so many cycles of absorption and desorption should be very stable, and the lifetime should be long. And hence the mechanical strength and durability of the material comes into the picture. And of course, finally, the safety under normal use and acceptable risk under abnormal conditions. That also should be taken into consideration. So now, theoretically, we talk about the first 4 points, that is the favorable enthalpies, or kinetics, and storage capacity. So we will talk about now how we can address this particular issue based on some of the very well-known chemical and physical concepts like simple electrostatics, curvature of the material, aromaticity, and charge transfer. So based on, I will use these particular concepts to explain how we can design the hidden storage material. Let us first look at the concepts based on the electrostatics. You look at the alkali metal series, sodium, lithium, beryllium. Now you can see here sodium ion, which is connected to 8 hydrogen molecules. 8 hydrogen molecules, and lithium, which can, the second one, green colored one, you can see that 6 hydrogen molecules, and beryllium that can also be attracted to 6 hydrogen molecules. And these are the stable system. That what I am showing you is the sodium ion can interact with the 8 hydrogen molecules very comfortably. I'm not even talking about after 8. 8 hydrogen molecule can comfortably interact. And we can see that from the IR spectrum, they're getting the vibration frequencies all are positive, and it confirms these complexes are the local minima. And this interaction, particular interaction, that is the alkali metal interaction with the hydrogen molecule, is found to be non-dissociative with weak interaction energy. That means that if you want to desorbs, you don't need to heat the system to a large extent. That is the main point. And the interaction here is essentially driven by the electrostatic forces, means that the hydrogen molecule initially doesn't have the dipole moment. Dipole moment is 0. But however, in presence of ion like sodium ion, beryllium ion, calcium ion, you can see the hydrogen molecule gets polarized, and hence the type of interaction here what we have is the induced dipole moment and quadrupole interaction of the molecules in presence of the cations. And here you can see that I have plotted the number of hydrogen molecules in the x-axis and the interaction energies. The energy varies between -5 kcal per mole to -30 kcal per mole. And the blue line stands for lithium, and red one is for sodium, and green one is for potassium. And here you can see that lithium is found to have more interaction energy. That means that it requires more amount of interaction desorption energy as well. So what we, in the previous slides, what we have seen is the interaction of cations with the hydrogen molecules and what type of interaction that we have. Now we will study the negative ion and molecular hydrogen interactions. And here we can see that fluoride ion, bromide ion, and the interaction energy that we have seen here is that it carries up from -4 kilocalories per mole to 2 kilocalories per mole. So the halide ions, which is having negative charge, they can also interact very effectively. In some sense, the interaction is somewhat more stronger than cation cases. And in sodium case, what I have shown you is that in case of sodium case, 8 hydrogen molecule can interact. However, for halide cases, the number of hydrogen molecules even it turn out to be more than 24 hydrogen molecules. In the first coordination sphere, we have around 12 hydrogen molecules, and the second coordination sphere, 10 hydrogen molecules, and the third one turn out to be 2 hydrogen molecules. So the number of hydrogen molecules that can interact with the halide ion is much more, and hence one can expect that this particular concept can be useful for designing the material based on the anions. And this is the same thing I have shown here. And now let us see the concept of neutral cluster. So we have seen the cation and anions, how the hydrogen molecule interaction is taking place. So if we take the neutral cluster of magnesium, can it happen? Now, why I have taken magnesium in this particular case, it is a very well-known reason. The magnesium is in the bulk space is known to attract hydrogen molecule. That means that it interacts very effectively with the magnesium. And based on that, we have simple metal hydride, transient metal hydride, light metal hydride, and so on. Now, in this particular case, the magnesium is lightweight, which is inexpensive, and high affinity for hydrogen. And the gravimetric capacity in bulk, it can go up to 7.7 weight%. But still, it is not very effective so far. So what we have done is that instead of taking the bulk, we have gone into the nanoscale. That means a cluster of magnesium atom clusters we have considered for hydrogen adsorption. And now here you can see that the activation for hydrogen desorption is lowered by 50 kilojoule per mole. If we take the magnesium cluster and allow the hydrogen molecules to interact, and the activation energy for hydrogen desorption is lowered by 50. This is the most important result. So in case of this is the one specific advantage of considering magnesium for hydrogen storage. The interaction is very strong in all other cases, but the desorption sometimes it is very difficult. It requires high temperature. But if we consider magnesium, the activation energy gets reduced. You can see here the hydrogen adsorption on nano-sized magnesium clusters. We can have number of hydrogen molecules turning to 16, 24, 32, and 36, and so on. And the same thing we can see for other hydrogen molecule, like if we have 4-atom magnesium-based cluster, we have 24 hydrogen molecules. Now let me just show you the desorption, how it happens. And here in this particular case, I have shown dehydrogenation characteristics of Mg4, which has initially have 24 hydrogen molecules. And in the x-axis temperature is given, and the y-axis average magnesium hydrogen bond length. So you can see that even at low temperature, around 150 Kelvin, the hydrogen molecule liberated is 6 hydrogen molecules. And around room temperature, you can see that 300 Kelvin, you can see that the hydrogen molecule, liberated hydrogen molecule is around 6. And the average bond length, which goes up to 5 angstrom. And that is an indication that it is already desorbs. And the same thing it is shown here for another cluster, magnesium 8. Even in this particular case also, we can see that the number of hydrogen molecules attached to magnesium 8 at the temperature, which is having 32, and most of the hydrogen molecule comes out only at 300 Kelvin. However, it starts deabsorbing from 160 Kelvin onwards, and slowly it is getting desorbed. If you increase the temperature up to 300, all the hydrogen molecule comes out. This is the point. This is the valuable information one can get it for magnesium nanoclusters. And the structure that I have shown here at 100 Kelvin, this is a 0 Kelvin, the leftmost, and the middle figure shows that at 100 Kelvin, all the hydrogen molecules are very good intact with the magnesium cluster, and 327 Kelvin, all the hydrogen molecules have comes out. They are the desorbs from the magnesium cluster. So the adsorption and desorption properties that has been studied here. And now next one is that how we can use another concept, that is based on the curvature and charge transfer. So what I have shown so far is hydrogen adsorption properties based on the electrostatics. Now let us go to another concept, that is curvature of the materials. And here we have considered the material is a carbon nanomaterials. It can be fullerenes, it can be nanotube, or it can be graphene. Now, here, what is meant by curvature? Let me just explain to you. And this is the picture. This is the intra-deformed benzene bond. Benzene is a planar molecule that you know very well. Carbon nanotube is having, except at the end, all the carbon nanotube is involving only the 6-membered ring. Now, if you start deforming slowly that 6-member ring, which is very much similar to benzene ring, except that hydrogen is not there in carbon nanotubes. Here I have modeled as the all other hydrogens are carbons are replaced by hydrogen just for simulating purpose. I will give you the detailed result after 2 to 3 slides. How this model seems to be well proven, let us see first. So you take the planar benzene molecule, you bend it. Both the ends you just slightly lift up or down. So we have concave type, convex type. So the amount of bending that has been applied for planar benzene molecule, that defines the curvature. Now, in this picture, you can see that this is a simple benzene molecule, C6H6. When you have the bending angle with 10 degree, the sodium atom still not interacting with the benzene ring, which is having 10 degree. But however, the next right side picture, which clearly shows that the bending angle, if it is 35, that means that it is very well gone up and the highly curved systems. And for this case, the sodium atom seems to be interacting very effectively with the system. That is, in our case, it is a benzene. So the lesson that we learn from here is that the curvature is very, very important. Now let us see how this systematically, if we increase the curvature of the benzene ring, how the charge and interaction energy is increasing. If the bending angle is 0, on the left side you can see, left side of the picture, if the bending angle is 0, that means the curvature is 0. Sodium atom doesn't interact with the benzene ring. There is no interaction, so the charge on sodium is 0 only. Now, you slowly increasing the bending angle, that means slowly increase the curvature, and you can see that the charge on the sodium increases, and if we keep on increasing the bending angle, the charge on sodium atom, it is increasing from 0 to 0.6. We can even achieve up to 0.8 just by increasing the curvature. So what we can achieve is that from 0 charge to 0.6 charge. So that is very important. Now you apply the basics of electrostatics. That means that if you have a cationic structure, then we can increase we can have the hydrogen adsorption at the center. That is the purpose of this curvature now. So the binding strength also, you can see that it is increasing very efficiently. That means that from 0.5 kcal, when the binding angle is 0, we have only 0.5 kcal per mole. That is means that at room temperature energy. However, if you increase the bending angle, the interaction energy changes from 0 to 21 kcal per mole. That is huge amount of energy is applied. So in principle, we can say that if you have a planar structure, then the interaction is very less. And if you have the curved structure, the interaction is very high. And that is why we term this type of interaction as curvature-induced molecular interactions. So let us see now in this pineapple type of structures, you can see that all these systems are all fullerenes. And in all the structure, we have applied the concept of curvature. Each fullerene has different type of curvature. So even if we vary slightly the amount of curvature, the interaction is going to be different. And hence, each fullerene can have different hydrogen adsorption properties. That is what it is shown here. The values are the what type of curvature is involved for each fullerene. Like C20, if you take the curvature value is around 65. That is why C20 structure itself is very unstable. And C28, C32, slightly if you go from C20 to C60, you can see that the curvature value is slightly decreasing. But all the systems are having finite amount of curvature. That shows that the fullerenes are very reactive, can be very reactive towards the hydrogen molecule. And this concept we applied and proposed the hydrogen storage materials, and that is based on the C60, which is having the curvature around 30 degree. And in this particular, we have taken the sodium atom, and when it is interacting with the C60, it becomes ionized because of the curvature. And there are many sites are available, and we have proposed that maximum you can put around 8 sodium atoms, and each sodium atom can have around 6 hydrogen molecules, so that you can have 48 hydrogen molecules for this particular system. And the gravimetric density turned out to be around 9. And whereas the requirement is only 5. Now, this explains why are the fullerenes or carbon nanomaterials are more reactive than graphite or diamond or even graphene. And we can also define or explain what should be the optimum degree of curvature for a particular material to have a desired property of the nanotube. So in this particular case, the hydrogen adsorption density is 9.68%. And what we have seen is the sodium metal doped fullerenes can have advantages because they are the weakly interacting systems, and hence the desorption can take place very easily, and lightweight materials. And this has also experimentally proven recently, and they also that is done for the lithium case. And you can see in this picture the C60 versus lithium, and the weight percent what we have what has been achieved maximum is 4.1 weight ratio. 4.1. What we have proposed is around 9, and for sodium case, for lithium case, it is experimentally what has been achieved is 4.1. And now let us switch over to another system, that is another concept that is based on the charged surface and aromaticity. Now, as a chemist, we all know about the aromaticity. What is meant by aromaticity that we know. And if you take the simple C6H6, it is an aromatic system. But what happens if you take the C5H5 case, which is lacking 1 electron to become the aromatic system? So what we have done is that we have taken the C5H5 radical and bring sodium atom, neutral system, and sodium atom alone, and interacting with the C5H5 case. And we see that sodium atom donates 1 electron to the C5H5 case, and C5H5 dot becomes like a C6H6, like a benzene case. It becomes aromatic in nature, and sodium atom becomes ionized now. So atom has become ionized, and this ion can indeed interact with the hydrogen molecule. So this we have used two concepts, the aromatic nature as well as the electrostatic nature. Both the things one can use to model such system. So the role of aromaticity in designing the materials for hydrogen storage based on the simple organic molecular system, starting from C4H4, C5H5, and C6H6. Okay, now let me just quickly go into the details. Another one, that is the alkali metal cluster aggregation on fullerenes. Now here you can see that the main problem what has been faced in the hydrogen storage material is that the metal atoms that are doped on the carbon nanotubes or fullerenes or graphene, they tend to agglomerate. So the individual atom capacity of hydrogen molecular system will be reduced if all the atoms gets agglomerated. This is what has been seen for transient metal atoms. However, for the alkali metal cases, the cluster aggregation turned out to be almost nil. The reason is that the cohesive energy of transient metal ion is much more than that of alkali atoms. So alkali atom never interact with the other alkali atom very efficiently. So once it gets doped on the fullerene surfaces or carbon nanotube surfaces, they tend to be remain there only, and hence the hydrogen storage properties or adsorption capacities will not be changed even if you go to the high temperature. This is the one important point that we have addressed. in the scientific literature. And let me go into the second topic, that is the nanomaterials for environmental application. Here I will be explaining about the chemical activation of gold clusters towards CO oxidation. Now, here I would like to spend 1 minute. The great chemistry can be done using only the C1 carbon, the system having one single carbon atom. That is the carbon monoxide, carbon dioxide, or methanol, or formic acid, and so on. Just one carbon system that can really make you very crazy, because modeling and designing these kind of systems are very, very difficult. For example, CH activation. If you can activate CH if you can find a suitable catalyst for CH activation, that would be the great work. Or another one is if you can find out the suitable catalyst for CO oxidation or CO2 utilization. Because carbon dioxide is, we consider it as a environmental pollutant that should not be there in our system, in our world. But the CO2, once we have it, you hydrogenate, you can get n number of chemical products, that is formic acid, methanol, methane, water, and etc. So the point here, what we are talking about here is that the C1 chemistry is very important, but at the same time it is very complicated. Now here I will be talking about the CO oxidation. Why it is so important? The CO is a major constituent in automobile exhaust system, and if CO is present in polymer electrolyte fuel cell, even at the relatively lower temperature, then the entire electrolyte becomes poisonous and it cannot function efficiently. So the amount of CO here, what we are talking is only at the ppm level. Even at the ppm level, the catalyst becomes poisonous. Another drawback or another worst point about the CO is that the bond dissociation energy of CO is the highest among all the diatomic molecular systems. This is turned out to be around 1047 kilojoules per mole, and hence you need the efficient catalyst for oxidizing CO. That means that activating the CO molecule is not very, very simple. So the challenge here is that we need to have a good catalyst which should function at the lower temperature. Now, in the literature, it has been shown that the gold cluster can be very effective for CO oxidation even at the lower temperature. And in turn, what we have seen is that if you just modify the gold cluster based on the hydrogen dopant or alkali metal dopant or many other atoms, if you dope with them, the sodium, the gold cluster becomes more efficient towards the CO interaction. Now you can see, for example, if you take the hydrogen dope systems, the absorption energy of CO, it increases tremendously if you dope with the hydrogen atom. Whereas if you dope with lithium or sodium, the interaction energy is very less. So the highest interaction that we can see here is around 25 kilocal per mole. And we took this particular model in our mind and we designed for oxygen activation and in turn for CO oxidation as well. So once oxygen is activated using the gold cluster and CO is interacting, the CO can be oxidized into CO2. That's what we have done. And here we can see that the remarkable role of hydrogen in oxidizing CO molecule in the hydrogen doped gold clusters. The oxygen activation is also done. At the same time, CO oxidation is also done. That we got two aims with the one stone. That is, we simply dope with hydrogen in the gold cluster, and we could achieve both hydrogen oxygen activation and CO oxidation. And the activation barrier in this picture, you can see that if you don't have the hydrogen doped one, the barrier height is 1.13 electron volt. However, if you dope with hydrogen, it is reduced to 0.7. So approximately we could reduce it by more than 45%. And this is the important result. And similar studies have been done for the seeing the curvature, how it is, whether curvature plays any role in activating the CO molecule by the gold cluster or not. And here we see that yes, indeed, it has a lot of, it has significant role in activating the CO molecule. And the other one is that if you have the biomolecular systems like DNA, RNA, how it is getting activated, the same gold cluster, whether it is the same as the carbon nanotubes or DNAs are different. And here we can see that the DNA base pairs like adenine, thymine, guanine, cytosine, the gold particle efficiently interacts with the system and it helps in the CO oxidation. And here in this particular table, on the from third column, you can see that there is a EB and kilocalorie per mole, adenine, thymine, guanine, cytosine case we have seen. And in the table below, whatever it is given here, there is a base pair, adenine, thymine, thymine, adenosine, guanine, cytosine cases, they are given here. And you can see that the binding energy and the activation barrier is very less here. So if we have the hydrogen dope system, there is a remarkable enhancement of CO adsorption energy, and the gold nanoparticle along with the DNA matrix, which can act like an alternative catalyst. So the mechanism that we have provided here is that the local charge distribution around the catalytic active substrate, and that becomes the controlling factor for the better activity of the catalyst. Okay, so last few things I will just shift, that is a hydrogen generation. Let me come to the summary and conclusion. So what we have studied here is so far is the molecule and materials, and we have addressed some of the important questions like, can we have different type of hydrogen, different materials, or different concepts of conceptual approach for storing hydrogen or a catalyst. That is what we have addressed here. And the important point that we have made in the environmental issue is that the CO adsorption can take place on gold nanoparticle, and that can be enhanced. The activity of a gold nanoparticle can be enhanced by the hydrogen dopant. And the gold nanoparticle, along with the DNA matrix, that can also form the alternative catalyst to the conventional metal oxide based catalyst. So the issues and future goals are like this. Although the hydrogen is being very small and simple, but it is very complicated. It is not that easy. Although I have shown all the modeling and the designing of the materials, but we are yet to achieve the desired value of gravimetric density around 5.5%. Although one of the experimental proof that I have shown that they have reached 4.8, but implementation of that particular material for practical purposes still very complicated. And the other important problem that I have discussed is C1 chemistry, which seems to be very interesting, but greater challenges ahead for making fine chemicals. So with this, I thank you, and I would like to acknowledge my colleagues, Dr. Srinivasu, Jana, and Professor Ghosh, and our BRC Computer Center for providing the high-performance parallel computing facilities. And before I end, especially, I hope the students are there at the end of my talk. I would like to just tell you about the opportunities at our DA institutes. And you can see that every year we take chemistry students for scientific officer job, and that is done by the entrance exam as well as the interview. You have to clear the interview, and the third stage is the training school. You have to appear for 1 year training school courses, and then you can get the job here. So I earnestly tell all the students to appear for the BRC entrance examination. If you have the GATE exam, get good GATE score, then you don't need to write the entrance exam. You can directly come for the interview, and you can join in the training school courses. And the other one is the diploma in radiological physics. That is a diploma courses, and this is really a job-oriented courses. And diploma in radiation medicine and training courses. That is for the MTech and PhD courses for HBNI, and that we have recently started Homi Bhabha National Institute. Apart from that, there are many schemes are available for doing science projects like DST Rajasthan and NIUS, that is a TA for institute is there. and VSRP program is there. Then Science Academy Summer Research Fellowship is there, conducted by the Academy Institute in Bangalore, and the other one is the JNCASR, that is also in Bangalore. Other one recently started by the S.N. Bose National Center for Basic Sciences. So they also give the fellowship for 2 months for doing your summer research fellowship. So if you are in the MSc first year, you can apply for this. And if you are in the MSc second year, you can go for this, our BRC entrance exam, and come for the interview. You can appear for the training school courses also. With this, I thank you all for listening this talk patient. Thank you very much.
Thank you very much, sir. You have lightened and expanded our knowledge on the topic to greater heights, sir. Thank you. Thank you very much, Dr. K.R.S. Chandrakumar. So we have few questions. Shall I put it to you?
Sure, yes.
Okay. First question, how does hydrogen fuel car work?
Hydrogen?
Fuel car.
What is the
Hydrogen
performance
fuel
of?
Yes, sir.
Yeah, the efficiency, as I showed in one of the slides, that is if you have 1 kg or 4 kg of hydrogen, you can go up to 500 kilometers. That is the projected thing. And this has been in many companies in Germany. Many companies are trying to make it. And the test car that has been run with 4 kg of hydrogen that are ran for 500 kilometers. So that is the efficiency we are expecting.
Okay. Next question, sir. How can we store hydrogen safely?
How we can store hydrogen?
Store hydrogen safely.
Safely. That is where you need this kind of materials, nanoscale material. Indeed, this is a very important question, and that is the focus of my talk. If we have the normal cylinder, there is a lot of problem associated with that. That all the experimentalists, they know very well. Hydrogen cylinder is extremely important. One should take care of very well. So that is why we look for solid state nanomaterials can store hydrogen, and they won't leak out that easily, and at the same time it solves all the safety issues.
Okay. Next question, sir. For hydrogen storage capability, transition metals must show Cuba's interaction. So what exactly Cuba's interaction involved?
Okay, yeah, this is a very important question. And in case of transition metals, the hydrogen molecule gets dissociated into hydrogen atom, and the hydrogen, so there is a d orbital interacting with the s orbital of hydrogen. Most of the transition metals have the d orbital, so d type interacting with the s type of hydrogen. So this type of interaction is called as the Kubo's interaction.
Okay.
But there is a problem that if you have the transition metal, if we dope it on the carbon nanomaterials or any other materials, fullerenes or graphene, they tend to agglomerate and they form the cluster. So we don't have atoms in a specific places. They all come together because the cohesive energy is very high. But in case of sodium metal or alkali metal, the cohesive energy is very, very small. So they remain to be in the same place, number one. Number two is that you don't need high desorption energy for the case of alkali metal because the interaction is very poor compared to the transition metal. The Kubo type of interaction is not involved in case of alkali metal series, but transition metal cases, Kubo interaction is there.
Okay, sir. Last question, sir. Can any other metal other than sodium can be doped with fullerenes?
Yeah. Transition metals and in alkali metal series itself, we have many other atoms like lithium, sodium, potassium. Even the alkaline earth metal also one can consider magnesium, calcium, and so on. This why fullerene is very effective in interacting with the atom is that fullerene has got an electron affinity of 6 electron volt. And that is a huge value that you cannot expect this much electron affinity with any of the organic molecular system unless it is a radical species. So having the neutral character of fullerene, its electron affinity is very, very high because it requires 6 electrons to complete its electron shell. So that is why it is very, very, very active. So it can interact with any metal atoms.
Hello. Thank you. Thank you very much, sir. Thank you for joining with us. Thank you.
Thank you very much for inviting.
Thank you.
We really enjoy listening all the talks. Thank you very much.
Thank you very much, sir. Thank you. Dear participants, we'll just take a break of 5 minutes and rejoin you for the valedictory session. Good evening, Udekla ma'am. Shall we start the session? Principal ma'am, shall we start the session?
Yes, sure.
Yeah. So welcome back Dear participants, we are just moving on now to our final session of this 2 days international conference, ISCST 2020. Now I just call upon Aparna to take over the valedictory session.
A smile is the universal welcome. With that humble smile, I welcome our respected Madam Principal, Dr. C.N. Ishwari, our distinguished Chief Guest, Professor Dr. P. Udayakala, Dean, Chemistry, Director IQAC, Dr. MGR Educational and Research Institute, our guest speakers, heads of department, faculties, and all our participants to the valedictory session of the 2 days international virtual conference on innovative strategies in chemical science and technology, being organized by the Department of Chemistry, Bhaktavachalam Memorial College for Women. Hope that you all had a great day. All our sessions has ended well. I would now like to invite our Madam Principal, Dr. C.N. Ishwari, Iron Lady of BMC, a rare combination of compassion with intelligence, for the special address.
Respected Chief Guest, Dr. P. Udayakala, Dean, Chemistry, Director, IQAC of Chemistry, Dr. MGR Educational and Research Institute, Chennai, convener of this 2-day international virtual conference and head of the Department of Chemistry, Bhaktavachalam Memorial College for Women, Dr. N.S. Sangeeta, other organizing committee members, resource persons, faculty members, and all other participants. A warm greeting to all of you. At the outset, I express my deep sense of appreciation to the Department of Chemistry for organizing this 2-day international virtual conference on innovative strategies in chemical science and technology, ISCST 2020, to provide a platform for the best brains across the globe to present, discuss, and deliberate important advancements in the field of chemistry. This lockdown initiative has proved beyond doubt that the lockdown may restrict physical mobility, but the human spirit is resourceful and resilient and cannot be curbed or confined. I'm sure this has been a unique experience for every one of you. The tone of the conference was set by our chief guest of the inaugural session, Professor S. Sriman Narayanan, Pro Vice Chancellor, Wales Institute of Science, Technology and Advanced Studies, deemed to be university, and former Professor and Head, Department of Analytical Chemistry, University of Madras. His crisp and informative inaugural address highlighted the importance of chemistry in our daily life and traced the periodical discoveries and developments. As Professor pointed out, chemistry is present in all fundamental aspects of our varied life, and it has improved the quality of personal care products, ready-to-eat foods, computer circuits, television screens, the color of the houses, the code of the refrigerator or the air conditioner, and even the beauty of our faces exist and improve only because of chemistry. Why? Now, when the world is reeling under the impact of COVID-19, we look towards the pharma industry and research in chemistry to come up with a vaccine, which we all pray and hope will happen soon. The presentations were mind-boggling. The kind of research that are being carried out to minimize our woes and improve our lives fill us with a lot of positivity and energy. And I'm very confident that this 2-day conference would take a step forward in the field of research. Once again, I thank the Department of Chemistry for organizing this 2-day international virtual conference, and I thank every one of the participants who, irrespective of their time zones, because it may not have been a convenient time to everybody across the globe, the time you have taken to be a part of us, to be with us in this conference. Once again, I thank every one of you. Thank you so much.
Thank you, ma'am. Look back with a smile. Go forward with courage. Many wishes for all that is good to go with you. With that note, I may now request Dr. C. Chandrakala, Assistant Professor, Department of Chemistry, to give us the summary of this wonderful 2 days international virtual conference.
Good evening to one and all present here. It is my great privilege to give the summary of 2 days international virtual conference on innovative strategies on chemical science and technology. The Department of Chemistry, Bhaktavakshalam Memorial College for Women, Chennai, hosted 2 days international virtual conference on innovative strategies on chemical science and technology ISCST 2020 on 27th and 28th June 2020. The ISCST 2020 brought together the experts, academicians, research scholars, and budding scientists on a single worldwide platform to explore their innovations, even amidst this COVID-19 crisis. The essential objective of the conference was to assist the researchers in discovering the global linkage for future joint efforts in their academic and research careers. Registration for the conference was open to all faculties, scientists, industrialists, research scholars, and students. Due to COVID-19 situation, invitations were sent directly to contacts in relevant fields. In total, 550 participants had registered for this conference. We had received totally 40 abstracts for oral presentation, out of which 31 papers were selected on the basis of the theme provided and their scientific novelties for oral presentation. ISCST 2020 was hosted through Zoom and was also live streamed on YouTube. The 2-day international conference ISCST 2020 was inaugurated on June 27th by Professor S. Sriman Narayanan, Prof. Vice Chancellor, Wells Institute of Science, Technology and Advanced Studies, Vistas, former head Department of Analytical Chemistry, University of Madras, joined by our Madam Principal, Dr. C.N. Ishwari, and our guest speaker, Dr. Shivakumar Ramdas, Assistant Professor, Research, Department of Obstetrics and Gynecologist, David Jeffrin School of Medicine, University of California at Los Angeles, followed by explication about the conference by Dr. N.S. Sangeeta, convener and head Department of Chemistry, Bhaktavakshalam Memorial College for Women. ISCST 2020 comprised of totally 8 sessions with 6 keynote lectures and 31 oral presentations from the participants. On day 1, the special sessions started with a keynote address of Dr. Shivakumar Ramdas, Assistant Professor, Department of Obstetrics and Gynecologist, Los Angeles, on targeted cancer therapy using nanoparticles-based epigenetic drugs. The second session was oral presentation chaired by Professor Priya Murugesan, Head Department of Physics, Savitha Engineering College, Chennai. 15 research papers were presented. The presenters were queried both by the chairperson as well as the audience. Dr. Mahesh Sundararajan, Scientific Officer, Theoretical Chemistry Section, BARC Mumbai, hosted the third session with his keynote address on pumpkin-shaped supramolecules from simple binding to catalysis. The special session was concluded with a talk of another eminent speaker, Dr. V. Balakumar, JSPS Postdoctoral Research Fellow, Department of Earth Resources Engineering, Kyushu University, Japan, on metal-free visible light photocatalysis towards sustainable future. On day 2, the second day of conference started with the eminent scientist Dr. J. Haribabu, JSPS Postdoctoral Researcher, Faculty of Pharmacy Science, Tokyo University of Science, Japan, delivered a special lecture on design and development of novel metal complexes for cancer therapy. The second day of the technical session was conducted in which the remaining 17 research papers were presented. The presentation was reviewed by the chairperson, Professor Dr. P. Udayakala, Dean Chemistry, Director IQAC, Dr. MGR Head, Dr. MGR Educational and Research Institute, Chennai. For both the sessions, best papers were selected by the session chairpersons. Another remarkable lecture was given by Dr. Kirti Murugan, postdoctoral researcher, Department of Chemical Engineering and Biotechnology, National Taipei University of Technology, Taiwan, on electrochemical sensors and biosensors, and followed by the last session on computational design of nanomaterials for energy and environmental applications by the eminent scientist Dr. KRS Chandrakumar, Scientific Officer, Theoretical Chemistry Section, BARC, Mumbai. It is fair to conclude that the conference was a great success. So many people have contributed in so many ways to turn this event into a grand success with many interesting presentations and creating a good ambience for discussion and networking. I thank all supporters for their generosity and interest in the conference. Now, I would leave you with a quote by Stefan Brand, an author and innovative thinker, who once said, "We can see the past, but not influence it. We can influence the future, but not see it." Let us all be guided by all the things we have learned and heard throughout the conference and be able to see and influence our future. Thank you.
Thank you, ma'am. A good teacher is like a candle. It consumes itself to light the way for others. I am greatly privileged to welcome our chief guest, Professor Dr. P. Udayakala, Dean, Chemistry, Director IQAC, Dr. MGR Educational and Research Institute, deemed to be University, Chennai. She has received her doctoral degree from Pachyapas College, Chennai, affiliated to University of Madras, master's degree in value education and spirituality from Annamalai University, and master's degree in psychology from Tamil Nadu Open University. She has guided 2 PhD scholars and has 33 research publications to her credit. Professor Dr. P. Udayakala has received many awards like Sir C.V. Raman Research Award in 2014 and 16 as Best Researcher, Outstanding Research Contributed Award in 2015, and Best Teachers Award in 2019. Ma'am, I would request you to address the gathering with your encouraging words.
Yeah, thank you. Thank you. I'll just continue. Good afternoon to all the researchers who were online enthusiastically all the time online for these 2 days. I could see that enthusiasm when I chaired one session and the department's enthusiasm. Even in this lockdown period, the department was not at all knocked down. They were all doing busy work, and especially I would feel that spirit within that women in this college. And the department head, she rightly understood her responsibility. It is like a responsible person fulfills the assigned duty by staying true to its aim. Duties are carried out with integrity and a sense of purpose. So I think she solved that by bringing all these researchers together for this 2 days conference. And it's really fantastic thing. I just found amazing to see the participants from north to south. Assam, Mizoram, Chennai, Tenkasi, Bangalore, almost all the states in India, and Chennai and China and Taiwan too. So I would see, she connected the whole globe even in this lockdown period, and it's really a great and wonderful job on her part. And coming to participants, It's really amazing to see your enthusiastic participation even on a Sunday, right? So even on a Sunday, it's really, I could see many chatting coming on. Thank you, it's good, well. So you were all like, researchers will never sleep. So there is a quote like, researchers will be always in their own thinking pattern to bring something new to this country. So it's your innovation will take the country forward. So with these few words, and I would like to share one small trick and tips to the participants here. When you present the slide, I would see like sometimes it is like we get many data from the literature review like antimicrobial, antibacterial, anti-inflammatory. This is seen irrespective in any article. So when you come for a presentation just for 5 minutes, it's not needed to display all these data which is not in your hand. So it is something like when I raise a question or someone raise a question, it will be like out of the way. So the final picture will be as if you have not done something great. So it is better just a 5 minutes presentation, crispy what you have done, and 100% the words displayed in the PPT, you should know thoroughly. It can be even 5 words, even 2 sentences or 3 sentences enough. So it is something like you have to present in a way the work is creditorious to me. So it is with this small tricks, it is simply a tricks. Of course, we all do that, and we have learned, and we are standing here now, and you are on the learning process, of course. So I just want to share these few words. With these few words, I thank the management and governing body of this Bhaktarakshalam Memorial College for Women for giving me an opportunity, and Dr. N.S. Sangeeta, the department head, for organizing such a beautiful conference. Thank you so much. I don't want to take a Sunday evening. Let me leave with a few words. Thank you.
Thank you, ma'am. I think we are even more energized after hearing you, and we'll be keep on trying new things like this. Every event comprises of a lot of hard work, and feedback always motivate us to do even more better the next time. So we may now request any of the participant to give the feedback about the conference.
Hello.
Yes, sir.
Really, it was a wonderful conference. Like in virtual way, it was very nice. Like we are attending like normal conferences. There are some mistakes will be there, but in this conference, I throughout all the sessions I have attended, it was really awesome, and all the oral presentation also I have attended. I really thankful to all the organizing committee, Sangeeta ma'am, and the team. Really, it's a teamwork and very succeeded one. Thank you, ma'am. Thank you for giving opportunity to present our work here. Thank you very much, ma'am.
Thank you, sir. Thank you. Thanks a lot.
Hello. Hello, madam.
Yeah, hello, sir.
Hello, I'm Dr. Kiran from Jain University, Bangalore. It was a wonderful sessions. All the 8 sessions are wonderful, madam. I would like to congratulate the organizing committee, especially HOD of Chemistry, Sangeeta madam, for giving the opportunity to present in this conference. Thank you, madam. Thank you.
Thank you, sir.
Hello. Good afternoon, ma'am.
Good afternoon, sir. Yeah, we can hear you, sir.
Yes, ma'am. Myself, Dr. S. Saravanan, ma'am, Assistant Professor, Saranda College of New Technology, Narsapura, Andhra Pradesh. So, thank you, madam. Thank you for this organizing committee, and great privilege to be here and part of this successful event, Iscst 2020. And then again, I'm thanking to the organizing committee because the way they have conducted these 2 days virtual conference with scheduled timing. Next, I'm thanking to the various subject experts around the world who delivered the lectures. And then finally, I'm thanking to the principal, BMC management for their support, and the Department of Chemistry. Thank you, ma'am.
Hello.
Hello.
Hello, ma'am. Myself, Sana Zalam. Ma'am,
yes, ma'am.
Ma'am, I would like to thank the Bhakta Vatsala Memorial College for Women for organizing such an informative session. And I would also like to thank the organizers, respected invited author guest, and also Ma'am Sangeeta Ma'am for giving us the opportunity to present our work in during this pandemic also. And it helped us to enhance our knowledge in various fields of science. Thank you so much, ma'am.
Thank you. Thank you, ma'am.
Hello, ma'am. This is Sachin from Jain University, Bangalore. First of all, I would like to congratulate the organizers for the successful completion of this event. And I must say that all speakers were very informative. And I would like to thank the organizers for giving me the opportunity to present my work as well as to share my feedback. Thank you.
Aparna, I think you can just move on.
Thank you everyone for your valuable feedback. Now it's time to announce the best oral presentation.
Good evening, everyone. I'll take the privilege of announcing the best oral presenters. The chairpersons of both the sessions, they have collectively given us a list of 5 best oral presenters. I just would like to announce the first best oral presenter. It goes to OP2. That is Mr. R. Rajkumar from St. Joseph College, Kovur, Kundrattur. The next best poster goes to OP15, Mr. Ningaraju C. from Center for Nano and Material Sciences, Jain University, Bangalore. The next one goes to Dr. R. Manigandan, Nanjing Medical University, Nanjing, China. The fourth one is for OP23, Dr. R. Naresh Muthu, JP College of Arts and Science, Tenkasi. And finally, we have OP24, Dr. Rajkumar K. from IIT Madras. Thank you, dear participants. You'll be receiving the certificates at the earliest. Thank you. Aparna?
Thank you, ma'am. Gratitude is not in the words, but in the heart that expresses it. I may now request Dr. N.S. Sangeeta, Head, Department of Chemistry, to propose the vote of thanks.
Occasionally in life, there are those moments of unutterable fulfillment which cannot be completely explained by those symbols called words. Their meaning can only be articulated by the inaudible language of the heart, saying thank you. Our most valuable dignitaries and ladies and gentlemen, I'm very humbled and honored to be the one making the closing remarks and giving the vote of thanks in such a distinguished and intellectually stimulating virtual gathering. Gratitude is the healthiest of all human emotions. With that expression, we would like to place on record our sincere thanks to our dynamic Vice Chairman, Dr. K. Maragadamani, whose vision has engraved us to perfection. You cannot cross the sea just by standing and staring it. The ingenious thoughts of our Revered Secretary, Sir, Dr. S.P. Rajagopalan, and his guidance has made us cross boundaries. Thank you, sir. Aim for the moon. If you miss, you may hit a star. It is the secret mantra of our respected Madam Principal, Dr. C.N. Ishwari, Iron Lady of BMC. She's a rare combination of creativity and intelligence, and she has always been sculpting us with her
valuable advice, sheer supervision, and constantly encouraging us in each and every step throughout this endeavor. Apart from the decision making, communication, and overall professionalism, the most important thing that I have learned from you is how to be a good human being, ma'am. Thank you so much. I express my deepest sense of gratitude to Professor S. Narayanan, Vice Chancellor, Wales Institute of Science, Technology and Advanced Sciences, and former Head Department of Analytical Chemistry, University of Madras, for finding time for us amongst his busy schedule and joining with us for inauguration. We are extremely thankful to all our guest speakers, Dr. Shivakumar Ramdas, Assistant Professor, Department of Obstetrics and Gynecology, David Geffen School of Medicine, University of California at Los Angeles, Dr. Mahesh Sundarajan, Scientific Officer, Theoretical Chemistry Section, BARC Mumbai, Dr. V. Balakumar, JSPS Postdoc Research Fellow, Department of Earth Resources Engineering, Kyoto University, Japan, Dr. J. Haribabu, JSPS Postdoctoral Fellow, Faculty of Pharmacy Science, Tokyo University of Science, Japan, Dr. Kirti Murugan, Postdoc Researcher, Department of Chemical Engineering and Biotechnology, National Taipei University of Technology, Taiwan, Dr. KRS Chandrakumar, Scientific Officer, Theoretical Chemistry Section, Baba Atomic Research Center, Mumbai. We really appreciate as they've highlighted the directions for the research to reach greater heights. Thank you all guest speakers for showing keen interest in associating with us. It's my proud privilege to express my sincere thanks and deep sense of gratitude to Professor Priya Murugesan, Head Department of Physics, Savitha Engineering College, Chennai, for chairing the oral presentation. Her ideas and perspectives are the explanations to everything. I may like to express our sincere thanks to Dr. P. Udayakala ma'am, Dean, Chemistry, Director, IQAC, Dr. MGR Educational Research Institute, Chennai, who have joined us today for the valedictory session and was also the chairperson for oral presentation. Today, we had an opportunity to hear your thoughts, ma'am, and this will surely encourage us in our future events. A special mention to our respected Dean Madam, Mrs. S. Mangai, for stimulating us to do our best and always standing as a pillar of strength. My gratitude is due to the heads of departments, dear faculty members of Bhaktavachalam Memorial College for their inspiration and moral support during the conference. An event like this cannot happen overnight. The wheels started rolling weeks ago. I have been fortunate enough to be backed by a team of very proactive and dedicated staff members of my department who were really well versed in their job. Thank you so much, my dear friends. I'm immensely thankful to my student, Ms. Aparna, for her efforts towards comparing the inaugural and valedictory session. Our sincere thanks go to all our participants from all over the world who have made this event to achieve its excellence. Thank you so much for attending the conference. I would like to thank all my family, for all our family members who have stood beside and helped us to successfully complete this conference, even amongst this COVID crisis. And last but not the least, all reserved and unreserved appreciation goes to the Almighty God for making these two sessions a remarkable and memorable one for us. Thank you. Thank you so much.
Thank you, ma'am. Every story has an ending, but in life, every ending is a new beginning. And this is the new beginning for a methodology of sharing our ideas. Once again, I thank you all for the extended support and wish you a great week ahead.
I wish to thank personally all the dignitaries and all the participants who had joined us for this two-day national conference. Thank you so much. Thank you.